per region, and there is one. Okay, there is one on the top. But yes, I can explain it to you later.
Okay, okay.
No, I mean you can keep talking. I just started the recording. We will cut that out just to make sure everything starts at time.
Okay, because it was like, okay, what an elegant way to say everybody. No, but yes, no, Davor, sorry.
No. Yeah. I mean, it's 10:05, but people are still joining. I mean, it is early morning, and I hope that most people look at the time difference if that was a problem. So yeah, we should start so Simon is at 10:15, and I need about 5 minutes, and then all of the founders. Julian, I'm not sure, is he here for
I
your
didn't see him.
speeches?
No.
Not yet.
So yeah, you have 2 minutes each, which is 6. Yeah, we will make it in time, so we can wait just a little bit for people to join.
If you want, we can shorter the welcoming part in case you are struggling with the timing.
I'll let you know.
I don't think I will occupy the 2 whole minutes. I have just a brief welcoming, and that's all.
Yeah, and same here. I think you know, I mean, I'm gonna show a few slides. I think 12 or 10 or so. I don't think that I mean, I usually talk a lot, but so I'm not sure if I actually need 25 minutes. So take your time, I guess.
Perfect.
No.
Good. So yeah, let's wait just a bit. Well, not really, a bit more. Let me just see the timing. Yeah, I think we will start right now, and then we can add another. Well, to be honest, just let me just get another lecture after you. Oh, Ivana is already here. Okay, so we can start, I suppose. Let me just share the screen. Yeah, just confirm that you see the screen sharing and everything. You hear me right? Perfect. Okay. So it's 10:07 and we can start this meeting. Good morning if you're in the Central European time zone. It is my honor to welcome you to the Naturalist Point event. This is a online symposium-like event made possible with a collaboration of Young European Biologists Network, BioBalkan Collab initiative, and Young Biologists Matter Network. This is a culmination of our work and also an icebreaker for our network in hopefully a very good start of our race and future in building this network and building a society of young researchers in the field of biology and biotechnology in Europe. Today, you will listen to 14 incredible speakers talk about different topics in biology, in biotechnology, and career development. I would like to thank the organizers of this event, to today's coordinators, all of the participants that register and hopefully join, and obviously all of the lecturers who agreed to give us a lecture today. The structure of the event. Does it move? Yep, it does. So every lecture will have 20 minutes to give their presentation, after which they will have Q&A segment of 5 minutes that can be extended to 2.5 minutes extra, but we would ask all of the lecturers to keep it in the agreed slots. While you listen to the lecture, feel free to write all your questions in the chat on the side. And after the lecture, one of our coordinators will read the questions to the lecturer and the lecturer will respond. Today's coordinators are colleague Eddie, Lena, and Leila. Obviously, we would like to ask all of the participants to keep their microphones off. You can keep your camera on, it is not necessary. And if you have any issues, you can contact us through our email or maybe even faster through Instagram DMs. And now I would give a couple of minutes of time to founders and ex-presidents of Young European Biologists. In order, I would like to call Anna, Stephanie, and Julian.
Okay, so well, thank you very much, Davor. Welcome everybody for this fantastic journey and this fantastic event ahead. So I'm Ana Horta Bellido. I'm the co-founder and first president of YB back in time. It is a pleasure for me to having this opportunity to be here. Almost in the anniversary when this idea came to life, which was on November 28th of 2015. So almost 10 years ago. Time flies. So just to finish my welcoming speech, I would like to do a reflection about what biology has meant to be. Biology means everything. For me, biology means diversity, knowledge sharing, science, curiosity, and passion. And what better example of this than this fantastic event where 3 different organizations have came here to collaborate together in order to foster different fields. As Davor has mentioned, ecology, they are talking about snakes, about different regulations and dissemination of different strategies that have been developing in the EU context. So thank you very much. Glad to be here and wish you all the very best for the event. Thank you very much, Davor.
Thank you so much, Anna. Stephanie, you have the word.
Yeah, good morning, everyone. Well, I'm Stephanie Lois. I'm also co-founder of YB and a former first vice president. Well, I would like to thank you all for being here. And it's really great to see YB coming back to life and becoming again a community for students and young professionals of this beautiful science that it's biology. So I would like to thank especially Davor and all the organizing association for creating this event. And I hope you all find it very interesting and very useful to make new connections among you and maybe, I don't know, start some new projects. And I would also like to thank the speakers for being here today and sharing their knowledge with all of us. So, well, I won't take any more time and let's get started.
Thank you, Stephanie. I'm sharing the screen so I can see. Is Julian maybe in the chat if he wants to say a couple of words?
No, he's not.
Okay. Well, then we have 3 extra minutes until the first lecture. If Simon, you want to start ahead, you can have extra time. So, oh wait, stop. Nope, yeah, I forgot about something. So thank all of the founders for their kind words of wisdom. And now, before the start of our first lecture, we would like to ask all of you to join us in a moment of silence to honor the victims and all those affected by the ongoing conflicts and struggles in the various regions around the world, including Eastern Europe and Middle East. Now, we would like to take a moment of silence, so 1 minute of silence. Thank you. Thank you for joining us in this moment of reflection. Now, we would ask the first lecturer to start his lecture, and obviously I need to announce you. So, Simon, you have the stage. So today's first lecture, his session will delve into the critical importance of collaboration in biology and sustainability education across Europe. Title: Strengthening European Collaboration in Biology and Sustainability Education, the Role of ICBA and Synapses. It will explore how collective efforts can drive meaningful process in these fields. Please welcome Simon Hossel, who will give us guide us through this important and inspiring topic. Simon, you have the stage. I hope I didn't butcher your last name.
No worries, Davor. Yeah, thanks a lot for the invite, Davor, and the newly forming YEB. I was also happy to meet Anna and Stephanie earlier, as some of you already maybe listened to our background talk. And I'm working for the ECBA for 1 year now. I started working for the German Association of Biologists 2 years ago. So I'm slowly progressing in getting people together in Germany and across Europe. So, and this is also the topic of my talk, strengthening European collaboration in biology, because I think this is really essential. And sustainability education, the role of the ECBA and Synapses. And Synapses is an EU-funded educational program where we start to make teacher training academies all across Europe. And I'm going to say a few things about this as well in a few slides. So in general, I want to say something about the status in biology in Europe. So we do estimate between 750,000 and 1.6 million biologists to be either employed or self-employed in Europe. So already from this number, you can realize that, or the huge variance, you can realize that organization of biologists is difficult. And so we do have different school systems, different studies. Some countries do have chambered biologists, which is super nice. Some do not have that. So for example, in the UK, you are in a chamber, and this also helps biology fields in a professional way to be guaranteed. In other countries where we do not have chambered professions, there's an EU-wide competition also with medicine and engineering. I mean, in Germany, we have really big problems, let's say. So the organizational challenges in biology. So biologists by nature are a very heterogeneous group of people. We usually are very poorly organized because sometimes we think about too much about the differences in our biological fields and not about the common things we should agree on and work together. And so even if they are organized in national associations, they often fight each other. And that's why we don't have a good lobby for our interests in most countries. And this is an invitation, and we see this already from the 3 collaborating groups that are organizing this event here. So there is a huge importance for topic-based regional working groups. If you do not have a national organization at your service. So we are, again, the ECBA, that stands for European Countries Biologists Association. And I didn't put all the logos here, I think. So you do see the blue countries that are members in the ECBA. So we do have from north to southeast and southwest, a lot of members in our organization. And the ECBA is a forum of national biology associations. So we do not have individuals being members in our association, but it's rather that people are coming from their association into the ECBA and use this forum for collaboration and exchange. So one question to all of you attending here, are you a member of a national association? If not, look it up. I mean, being working for 2 associations currently in biology, I think it's really important that we do organize ourselves. And this is just a plea to just look what is in your country. And if you do not have a national association, I was already saying this in the slide before. We can also talk about how you can become a regional working group that can also come under the umbrella of the ECBA. So, and if you are from one of the countries that were just highlighted in blue in the previous slide, so you could tell if your association is a member of the ECBA. So what are we doing? So as I said, it's a network of organizational members, and it's an opportunity for dialogue, knowledge exchange, and collaboration. And we do also share our success models, because the different organizations and associations, they have different usually also fields. Some are very active in accreditation, some are very active in teacher trainings, because we also do need some money income. And even though we are non-profits, we do need models that are also helping us gaining or keeping our members. And this is just a very big forum for this. We do have annual council meetings, so people come together, they meet somewhere in Europe, they come with their reports from their national associations. And so this is also a very fun time to hang around with people from all across Europe and to see what they're doing. It also happened in the past that, you know, there were different topics that were worked on during these council meetings that we published. And these were recommendations for policy and for societal guidance on key biological issues. We do also certify biologists, because even though Europe or the European Union is increasingly developing common standards, it is essential that professional competence of those who oversee the maintenance of standards is recognized, and academic qualifications alone have limited value, because we do have different standards, and that's why we have an acceptable common professional standard, and we do certify biologists, because it requires a lot of experience in applying knowledge, skills, and have enough attention to safety and environmental consciousness. We do have for this the designation European Professional Biologists, and it's a professional qualification, and it accredits biologists as being registered as qualified biologists, so they can work all across Europe. And this register is held by the ECBA. Okay, and then the ECBA is also, this is more for the self-employed people. It's also a nice way to interact with people, because the certification, I mean, if you're working in a big company that is globally active, you don't need a Euro ProBio identity card. This is more professional qualification if you're self-employed, for example, especially in the environmental sciences. This is really important. And so this is also a way to connect with other people and their experiences. So there's a lot of common questions for beginners usually that are self-employed. So how do I start this career? How do I start my own company, for example, insurances, and what else is necessary? And that's also like one of the problems, or let's say challenges, that, you know, we do have a lot of beginners that have a lot of questions, and to keep everybody in the ECBA and everybody happy, it's good that people coming together, even though they progress in their career, because usually you have fewer questions and fewer time, and usually those people get less active in their organizational involvement. And we just want to highlight here that this is really important to keep the ball rolling. And yeah, so biologists have been poorly organized over the past 50 years. And I think, you know, now is also a different time, because biologists are needed. We are all needed in times when, you know, we have global warming, we have biodiversity loss, and we need to be a common voice. And we need to come together for this and start lobbying for our interests. And this is just a question, will you make a difference? I hope. And just come and chat what the opportunities are, what the things are that we can do together. And so this is the ECBA for now. And now I want to briefly talk about the Synopsis project also. We are setting up Erasmus Plus school heads and teacher training academies all across Europe. And we do train for sustainability citizenship. So that's education for sustainable development. This is part of our consortium. So we are 10 different organizations from 7 countries. They're highlighted in orange in the map. So it's partial overlap with the ECBA network. And yeah, so we're teachers, teacher trainers from universities, NGOs, NPOs, researchers, professors, curriculum developers, so all kinds of people. And we also, or I want to have this invitation for you to engage with me. We do have ECBA webinar series that I started when I started working for the Synopsis project. And the webinar series is entitled Global Goals, Local Impact, Sustainably Ever After, hopefully. So we do have a huge focus on environmental issues. And if you're interested in giving a talk between February and June next year, just feel free to reach out. And because I'm starting to organize the program for the upcoming spring and summer. And with this, I'm at the end of my talk. And thanks a lot. And if you can email me, add me on LinkedIn, and I would be really happy to get in contact with you. Thanks. Davor? Yeah.
Thank you, Simon, so much. Now, if there's any questions, it's a Q&A segment. You have extra time, so everybody can ask questions in the chat. We will read them. And until the question first comes, here's my question. So regarding, as you know, there's a lot of people in the chat, and most of them, I believe, are bachelor or master students. So as a young researcher, what are the benefits of participating in associations and some type of either student or young researchers networks in building your career and stuff like that?
I mean, thank you. That's a very good question, Davor. I think people underestimate how important networks are in your career development, because usually, I don't know how it is in your countries, but from a German perspective, you're in university, you're always thinking about the really fantastic scientific details. And at university, there's this one perspective that you have, yeah. So you're a biologist, you do a PhD, and then you become a professor. I mean, there's nothing more usually outside of this. And if I'm also working for the German Association, and I'm doing webinars also with students. So what can you do with your life after having studied biology for quite a time? And a lot of people, they cannot imagine what they can do. And this is where associations come and give you a perspective. Usually you can connect with people, and you know, I'm also doing this, I'm doing webinar series where we show what you can do. And if I invite a lot of guests throughout the years, and if they show their CV, some of the students, they say like, yeah, but this looks like you just gotten lucky, yeah. But that's not how it is, because you can basically prepare for luck, because this is how you set the right things in your career, and this is by connecting with people. This is also, you know, skill development for sure, but this is not something that we lack as biologists, because usually we are highly motivated, we are highly trained, and but what we forget is how we get into this world and how we get into a career, because we have these scientific fascinating details that we're working with. And I think this is something that young people especially underestimate, and I want want to highlight that this is really important, knowing people and having a good network, because then you can jump from one thing to another, which you might not find so easy if you don't start this early.
Thank you. Anna pointed out that your QR code doesn't work, so you can share your LinkedIn profile in the chat, or we can send it however you like it best. Perfect. And another question from this side is, how does one apply to become a certified biologist?
All right, for me it works. Sorry, again, to apply for the ECBA.
Yes.
Just let me check. It's on our website. You can apply online, and you basically we ask you a few questions. What you about your trainings, about your experience, and so forth. And then after you hand us your application, we will handle the rest for you. Yeah, but I can put it in the chat also. Let me check.
Perfect.
So I put it in the chat now here. That's the europrobio.eu. And just to make sure that you can find me, I'm putting the LinkedIn as well. All right.
Perfect. Thank you. Is there any more questions for the first lecturer? Feel free to write them down. Going once, twice. Yeah, perfect. Thank you so much for the lecture. It was really thought-provoking. It is really it's a very nice lecture. And I believe all of us who listened to it benefited greatly from it. So yeah, thank you so much, and hopefully we will collaborate on some other future events.
Yeah, thanks a lot again for the invitation, and have fun during this symposium. I'll take care of my baby boy now. Ciao.
Thank you so much. Goodbye. Yeah, so we will continue with this event with our second lecture. Ivana, you can just, you have extra minutes, so you feel to try the presentation and sliding everything.
Yeah, I just need sharing permissions.
Oh, I think anybody can share. The permission is for, yeah, you should be able to, all the participants can share.
Interesting, because when I click, nothing happens.
Okay.
Okay, just a second. Okay, I think you can see it.
We can see it. Can you slide just one slide? Okay, perfect.
Okay.
Perfect,
so
perfect.
Can you just tell me what do you see?
The first slide is the title and your name on the end.
Amazing. Okay, then I'm ready to start if you're ready to start.
Just let me announce you. Right, perfect. So we will continue our event with our second lecture, Ivana Ostradak. Our next lecture will take us on the journey into the cutting-edge world of long-read sequence technologies. These groundbreaking tools are revolutionizing the way we explore the genome, uncovering hidden regions and complexities that were once beyond our reach. Titled The Significance of Long-Read Sequencing Technologies in Exploring the Unseen Parts of the Genome. This lecture promises to shed light on how these advancements are transforming our understanding of genetics. So I give the platform to Ivana.
Great. Hi. Hi, everyone. I am Ivana. I am PhD student at EMBL in Heidelberg and also in parallel a master student at the KU Leuven in Belgium. But today, in this lecture, I will talk about how long-read technologies very recently enabled us to study especially repetitive elements, structural variations, and centromeric regions that were quite inaccessible with the short-read sequencing. And I will tell you what we have learned from it. And just a little disclaimer, no one sponsoring me for promoting long reads here. It's just my PhD work. So to follow this easier, I divided the lecture into 4 smaller parts. So I will first remind you of the variation mirrored in the human genome. And then I will switch to explain you how can we characterize it using both short and long read technologies. Then we will look at the improvements in understanding the biology we see in the long-read sequencing technologies. And then I will give you the examples of the projects I am working on that, again, focus on the long-read sequencing. But let me first walk you through what I mean by variation in healthy, but also a cancer genome. For some of you, this is going to be a reminder. But so in general, we typically classify genetic variations into 3 main categories. And so we have single nucleotide variants, or SNPs for short. Those involve a change in small nucleotide or a letter in the DNA sequence compared to the reference genome. And those are the most common type of genetic variants we see. Second one are small insertions and deletions, or indels for short. So this means that we have changes involving the addition or removal of short DNA sequences, typically around 50 base pairs or fewer. And last but not least, we have structural variants. So those are really larger scale changes in the genome. So they include large insertions and deletions, but also more complex rearrangements such as translocations, where the parts of the chromosomes are swapped, and inversions, where the segments of the chromosomes are flipped. But then you might ask yourself, what does that mean for me personally? So if we look at the typical human healthy genome, there are around 4 million SNPs, so they're most abundant. There are around 500,000 indels, so it's a smaller proportion. And then we have structural variants that are least frequent, but they really have the most profound impact on the cellular function because they are larger and they often disrupt gene regulation or chromosomal integrity, and really quite often making them the key contributors to human diseases. But let's say this previous slide was easy, because now once we start talking about the variation in cancer, things very quickly become very complicated. So one critical factor is the timing of when we sequence a tumor sample, and this is because the genomic landscape evolves over time rapidly. So tumors, as you can see here, they are highly dynamic. They are influenced by the ongoing mutations, selective pressures, and treatment altogether. So over time, you have different clones with different mutations that support survival or growth, and they will expand, and then you have less advantageous clones that just diminish. So that really leads to significant tumor heterogeneity. And speaking about the heterogeneity, one key thing is to consider how deep you're going to sequence. So to accurately capture this heterogeneity, sequencing deep plays a crucial role. So consider this example. So you have a tumor, and let's say we sequence it the entire thing at the 100x, which is quite deep. And then you have this purple clone, which constitutes only 10% of the tumor content. So that means that automatically, in the end, the sequencing depth for this particular clone will effectively reduce to 10x. And then if you analyze haplotypes, this goes down even to 5x, which means it's extremely challenging to do any high confidence somatic variants. And this problem is further complicated by this contamination of the normal tissue, because it kind of contaminates the cancer, and then it's even harder to say, okay, what is my normal cell, what is my cancer cell? If you have a very shallow sequencing depth, it almost comes to the impossible. And then on top of that, we have to often deal with a very complex rearrangements, and one example is chromatopsis, and it's a quite common thing in cancer where like a large parts of the chromosome just shatter and they randomly rearrange altogether. And this is very difficult to kind of resolve with short read sequencing. So to wrap up this slide, so the key take-home message is then when we need to sequence tumors, we need to sequence deep, but also the consideration you need to take is that the deeper you sequence, the more expensive it gets. So this is what we're dealing with. But despite those challenges, several years back, we analyzed a cohort of 2,600 cancer patients in the study that's called Pan-Cancer Analysis of Whole Genomes, or PCOG for short. So this was a groundbreaking global collaboration involving researchers from numerous nations across continents that characterized around 38 distinct tumor types. This was based entirely on short reads, but even today we keep going back to the PCOG and kind of refining understandable cancer biology, especially because we can integrate long reads. But one of the key takeaways of the study is that the structural variants actually played a majority very big role in driving tumorigenesis. And then on average, each tumor had around 4 to 5 driver mutations, so the key mutations that are important for the cancer development and progression. And also, we very often see complex rearrangements such as chromatripsis, and sometimes this chromatripsis can happen even 10 years before a patient is diagnosed with a cancer. So how does this look like in a day-to-day bioinformatics lab when you have to deal with that? Here I will go back to something my professor at the uni said, is that when you deal with the biology, it's certainly not like engineering, where you engineer a perfectly working cell. In biology, when you deal with sequencing, you deal something that looks like that, like a crashed plane, and you're trying to figure out what the hell happened here. And traditionally, we've been doing that with short reads. So you chop the DNA into very short fragments, you sequence it, you align it, and then you, in the end, get sort of a table like this, where you have different variants, and you're trying to figure out what variation do you have in your genome. But the short reads are fine for only small structural variants, because on the other hand, most structural variants are large, and this led to significant discrepancy over years between the actual diversity of SVs and what we can actually capture. And particularly difficult are, again, those repetitive regions where short reads completely fail to do any accurate estimations. So here you can see how was this progressing over years, where you see that only with the emergence of the long read sequencing, we really got a peak or increase in number of SVs we can characterize. Long reads are still more expensive than the short reads, and the costs are in general going down, so that's a good news, but it's totally something we need to consider when we think about those things. And now, there are two key technologies that we use at the moment when it comes to long reads, and this is Oxford nanopore sequencing and PacBiosciences, where now, in comparison to 150 base pairs fragment from Illumina, we can sequence on average 10 to 50 kb with the ONT or 20 kb with the PacBio, depending on the library prep. But in this lecture, I will only focus on the ONT, which I'm also using at work. So how does nanopore work? So it essentially, this is sequencing in real time. So you kind of have a membrane, and there are thousands of little nanopores embedded in, and then altogether is placed in an electrolytic solution, and then you apply a constant voltage to produce an ionic current, and then negatively charged DNA literally goes through this pore, and it's ratcheted with this motor protein that unwinds the DNA and pushes it through the pore. So what happens when the DNA goes through the pore is that bases that are detected will have a very specific electric shift in this current, and this is basically decoded into bases using computational algorithms. So it kind of in a cartoon, this is how it looks like. So the DNA goes through the pore, causes those shifts in the electric current, and this is essentially what gives you base calling. And for decoding it, we quite often use recurrent neural networks. What is very specific for the nanopore is also that you can detect at the very same read not all the modifications, but like majority of the modifications that are happening on those bases, because as a normal letter would cause a specific shift, a letter with a modification also causes a very specific shift, which is then quite often processed with the convolutional neural networks to give you, for example, methylation readout. So coming back to this biology plane, the question is now, okay, what are the main advantages of using long-read sequencing to decipher what caused the crash? So in my opinion, now since you don't need to chop this original sequence into short 150 base pairs like with Illumina, you can really let several megabases, long stretches through the nanopore. And what that means is that instead of having this thousands and thousands of small short contigs, you have few very long contigs, and it's very easy to then assemble the whole genome. And then that also means that you can just sequence through most of the large SVs and complex rearrangements and resolve them. And what is very, very valuable, you can also resolve them per haplotype. And I will give you an example. So here we have a medulloblastoma case from my lab. It was sequenced the 30X with the ONT. The main focus was to assemble those complex rearrangements. So here on the left, you have a copy number plot that is focusing on specific regions of chromosome 11 and chromosome 17. For chromosome 11, you can see that it starts with a copy number of 1, but then there are distinct amplifications. So one region jumps to the copy number of 4, then it's followed by another amplicon with the same number of 4. And then also on the chromosome 17, we observe multiple regions with a copy number of 4. The thing is that when we examine these amplified regions, particularly when they share the same copy number and are connected with the long-range structural links, with the long sequencing read, we can quite confidently then say, okay, this is actually how chromatopsis in this cell looks like. And what is really cool is coming back to the methylation that we can detect. Paper from 2018, but it still sparks a lot of attention, is they literally used the nanopore sequencing during a patient operation. So they took a piece of the tumor, sequenced it while the patient was being operated, and based on methylation patterns, they could classify tumor type. For the patient, that means actually for the doctors, that means having very instant information how invasive that cancer is. And you can imagine if you get information that the cancer is really invasive, you automatically during surgery cut more of the tissue. And if it's not, then you cut less, which becomes very important, especially when it is some tissues like brain, for example. You don't want to cut too much if it's not necessary. Okay. And the last part, so I will briefly say how I'm using ONT sequencing, exploring centromeres and transposable elements. I will not say much about my projects because work is still very much in the progress, but I will try to give you an idea. I will start with transposable elements. So this is kind of a different type of variation we see in the human genome. And we all know that human genome is much more than just coding versus non-coding regions. We can also classify it as looking at the mobile elements or the jumping genes or TE. And those are repetitive elements that make up close to a half of the entire human genome. And based on the mechanism of jumping or transposition, we say that there are two major types of transposable elements. So type 1, and they're known as retrotransposons, and they rely on copy and paste mechanism, meaning that they first get transcripted into RNA that is reverse transcribed back to the DNA. And this class involves the line signs, SVAs, and LTRs. And then there is a rather simple type 2, so they do something that we call cut and paste. So they will simply excise themselves from the genome and integrate elsewhere. And so I will focus only on the L1 because this is simply what I focus on in my PhD. And I will tell you that in humans, those are the only autonomously active family of transposable elements. There are around 500,000 of L1 loci in the human reference genome. Most of them are truncated inactive elements that cannot retranspose, and then you have a small subset of 100 to 150 loci that can, and interestingly, that increases or almost a half in cancer context. But despite this rarity, they actually have a significant contribution to genetic and epigenetic evolution in both normal development and disease context. So this is something that really draws our attention to explore those elements more. I can tell you that transposable elements really are drawing attention in oncology, but people are really facing the issues of analyzing them with long reads and current tools that are on short reads and current tools that are based on the short reads. And this is particularly because those insertions can be polymorphic between individuals and between the clones in the somatic tissues. Second, they're repetitive, so meaning it's a nightmare for any short read sequencing to kind of really look at it because it's so repetitive. Three, they're quite often affected, this quantification is affected by the nearby genes. They can be easily confused with some noise from transcripts with incomplete splicing in the total RNA sequencing data. And when it comes to neoantigens, it is so the annotations that we have for those transposable elements are very fragmented and incomplete, and it's really, really hard to tell which transposable element derived neoantigen is actually from what DI loci. So I will just quickly tell you that this is something I'm addressing in my PhD, where I'm really trying to look at how we can now capture them with the long reads where they're transposed, let's say de novo, so L1s that just transposed, and also how they are expressing themselves on some transcriptomics level. But I will quickly finish off with exploring centromeres, the other project I am very much working on, and centromeres are becoming really getting a spotlight because with short reads we literally had a gap. We couldn't sequence centromeres. It was so repetitive that all the sequences would just collapse somewhere randomly on the reference genome. And this is because the centromere, so the central part of the chromosomes that is responsible for chromosome segregation, is comprised of the two highly repetitive regions. So you kind of have this pericentrometric DNA that is a bit less repetitive, but then you have those alpha satellites that are very, very, very repetitive, and we often refer to them as higher order repeats because you can literally have thousands and hundreds of the same segments repeated. But what is quite important for the centromere is that one of those high order repeats is what is associated with the kinetochore. And the idea here is to really to explore the centromeres for each chromosome in healthy and disease context, especially cancers. And as you can see here, what we learned is that centromeres between different chromosomes vary in length quite a lot. And if we combine that now with a methylation readout, where you can get from the very same run of the ONT, we can explore how this methylation on the centromere looks like. And why is this important is that centromeres have 1 to 2 those CDR regions, or the centromere dip regions. This is literally a dip in methylation where it's thought that the kinetochore attaches and then pulls the chromosomes onto the different poles of the cell. And you can imagine if something is going wrong either in the length or methylation, this can cause missegregation in the cells and really have a quite high cancerogenic potential. So this is what I'm focusing on. I'm sorry I cannot tell you more, but hopefully you can read in some future papers. So just to summarize, the open questions that are still there for me is to really discover what is the rate of those de novo L1 transposition in human cells, both in health and disease. What is their contribution then on the protein expression in cancer? And regarding the centromeres is how different architectures of those centromeres will contribute to cancer genomics and cell missegregation. So with that, I would like to finish off my presentation. Let me know if you have any questions, and I hope you didn't fall asleep. Thank you.
Thank you, Ivana, so much. A really good presentation and a really cool topic. It's a complex topic, but the visualization really helped a lot. If anybody has any questions, feel free to write them in the chat. Until that, my question. So this is based on long read sequencing. And we had one of the lecturers in BioBalkan a couple of blogs ago that used a similar method. Well, we used long read sequencing to work on spider determination through molecular methods. And the one question that was interesting, someone asked him, does he think that sequencing technologies are going to become even more advanced, more simple to use for everybody, and more cheaper? And his answer was that he doesn't believe so, because he kind of thinks that we kind of reach a threshold of sequencing, that it can't really be cheaper than this, and can't really be more simple, because nanopore sequencing is already quite simple and affordable to a point. So what is your opinion about that?
I disagree regarding specifically regarding the simplicity. So nanopore is really much. So first of all, this is a technology that changes so rapidly that it's also in the lab really hard to keep up with their updates. So they're constantly improving and making it simpler to sequence. And also with Illumina, we've seen over past few years that you're getting larger and larger sequencer, that you just need to press few buttons and all the reagent reactions and sequencing happens for you. And there are also, especially Illumina is improving on delivery in hospitals, where you have the Dragon software directly connected to the sequencer. You just sequence and get all the result analysis out in like few hours. So I would say it's definitely getting more simple. So in terms of also ONT, I've been to their conference in May in London, and they're really trying to make those small devices where you can just plug in and sequence and get the result. So this is kind of where the field is going to. Regarding the costs, yeah, it's relatively expensive at the moment. Illumina is still quite cheap compared to ONT. My personal hope is that this will get cheaper, but for me, it's at the moment hard to tell how and when is this going to happen.
Thank you. One question that a colleague on the side asked me, do you believe that methods that you are using can be used in other parts of research in genome, not just necessarily for oncology research?
Yeah, definitely. So sequencing is very much present everywhere. It's kind of the start of your experiment. I mean, at least this is, we as bioinformaticians, we like to work with sequences to see what we're dealing with. And then you later on go to some experimental validations. So my focus is on oncology, but my colleagues are working on rare diseases, for example. This is highly valuable to have ONT in rare diseases where you have to look at different haplotypes, for example. Then EMBOL now has this track expeditions where they're literally taking boats and going along coastlines in Europe with the sequencing devices where they sequence real time. So it definitely has a very broad application. If that answers your question.
I think it will, yeah, definitely. And another question on the side, they're not in the chat. She asks, obviously there's not one specific cure for cancer. Cancer is a lot of different things, a lot of different subtypes. So do you believe that, so research in genome is obviously important because the cancers are very heterogenic, but do you believe that development of different types of vaccines and different types of cures for cancer would at one point make the difference in the genome not so relevant for their curing?
No. So actually, the more we're going into personal medicine, the more you need to rely on sequences, because you need to learn what's going on with the person. You need to know their genetic makeup. So it's really inevitable that you sequence the individual, you sequence the tumors as best as you can. And then if you want to do any type of personal medicine that is based exactly on that. At the moment, the lab that I was previously on, they're also switching to long-read sequencing to sequence through T-cell receptors, for example, which are very patient-specific. You cannot really replace that with anything else.
Thank you. And the final question, well, this is my question. So how does one get in your field of research? Like, how did you start and how did you arrive to this this field of biology?
I very much like okay, this is my personal experience as a bioinformatician. For me, it was very important that I have a very flexible job. where I can work from, let's say, anywhere. So most of my time I spend in the office, but for me, big part was that I don't have to spend days in the lab. I can work from home. I can work from Croatia, Germany, whatever. I would say now, from a retrospective, if you want to get into bioinformatics, maybe focus less on becoming a good coder, because we see that things like ChatGPT can solve that for you. What you should focus more is getting theoretical knowledge on statistics and biology, with the emphasis of statistics. This is what I would recommend to people if they want to kind of keep their position in bioinformatics.
Thank you so much. If there's any questions, feel free to write them in the chat. If not, thank you, Ivana, so much for this beautiful lecture and the Q&A section. And I hope that you enjoy giving us a lecture, and I hope all the participants enjoyed as well.
Yeah, thank you for inviting.
You have a hi from Varaždin in the chat.
Nice, thank you. That's actually my teacher. Hi.
Yeah, thank you so much. We are 10 minutes ahead. So next lecturer is Federica. Federica, you can try, if you're in the chat, you can try your presentation again. And I would ask you to have a 5-minute break just to get back on the agenda. We ask all the lecturers to join 10 minutes early, but we are 10 minutes ahead. So just to keep everything going.
Okay. Okay, I think you can see it, and I'll wait.
Yeah, we can see it.
Perfect. I'll wait.
Perfect. So yeah, 5 minutes break. Let's say 11:06. We can start again. Okie dokie, it's 11:06, so we can slowly start. Federica, are you here?
Here I am. Perfect.
Perfect.
You should
Let me
see
just,
me.
yeah, we can see the,
Perfect.
okay, so let me just announce you. And right, so now we're moving on our next lecture, titled Map the Giants Seeking Out the Last Monumental Corals. This presentation will take us in the deep waters where some of the oldest and most majestic coral giants reside. We'll explore their significance in marine ecosystems and their urgent need to protect these natural wonders. Simone Montano, Federica Siena from the Map the Giants will share her experience on this topic. You have the stage. Oh, your microphone is unmuted.
It should work now. Does it work?
Yeah,
yes,
it
it
works.
does, but also I'm not sure if we have the slides right now. I'm sorry.
Oh, yeah.
Okay, great start.
No, it's completely fine.
Yes. Now, video and audio.
Yes. Okay, yes. Great. Thank you.
Okay, perfect. Thanks a lot. So my name is Federica Siena, and I am a PhD student of the University of Milano Bicocca. So the title kind of spoiled the presentation, but the first question is about who are these giants that we are talking about? As biologists and also biologists on land, very often we think about creatures and organisms that live on land. So when we talk about giants generally and very long living organisms, we think about, for example, these aspen organisms, plants that have been found off the United States. We're talking about organisms that are
Federica, I just want to, the presentation cannot be seen.
Okay, I don't know what's happening.
When you share the screen, there's the three options, I believe. You just share the entire screen.
Okay, that's what I
think it's the first option.
did. Okay, what about now? Can you see?
Yeah, we can see.
The second slide?
Yeah, it's Panda, right?
Okay, perfect.
Yeah.
So we were talking about aspen, so these very long living organisms that are part of the United States, they're found in the United States. We are talking about organisms that, although they look like very independent and individuals, they're connected by a shared root system, making them the largest living organisms in the world. And they've been around for over 10,000 years. Always when we are thinking about organisms on land, we probably think about the sequoia, the redwood found in California. A lot of people have seen them in videos, and a lot of people every year visit them because they're recorded as the tallest of the trees on the planet. They have been around for thousands of years, and people from around the world make trips to visit these trees. But when we are talking about the marine realm, where we spend as biologists, marine biologists, the vast majority of our time, we think probably about whales as the largest living organisms. Of course, whales are the largest vertebrates that are found in our oceans. But when we think about the biodiversity of our oceans, actually, invertebrates are what make biodiversity in large amount. So up to actually a few weeks ago, Big Mama was the largest known organism, actually not the largest, but the most famous invertebrate in our ocean. It's a coral that has been found in American Samoa. It was not the largest because also in this area there was another coral larger than this one. This one was measuring 42 meters in circumference, but next to it, very close to it, there was another coral over 69 meters in circumference. When we're thinking about invertebrates such as these ones, we're thinking about clonal organisms. Basically, we are starting from a single polyp that keeps splitting itself and reproducing, forming a colony that has been around for hundreds of years. We're thinking about organisms that will be probably born at the same time as Leonardo da Vinci. Actually, just a few days ago, the news broke all over the media and internet and everywhere about the largest coral being found off Solomon Islands. We're talking about a Pavona claus that measures 32 meters by 42 meters, and it's regarded as an organism of 300 years old. a very, very special and majestic organism. So what is in common between all these organisms? Giant corals are organisms that have resisted for hundreds of years to environmental perturbations. They've survived, basically. They preserve unique information from a genetic point of view, but also studying their genetic patterns, but also their skeletons, they harbor important information about the past oceanographic conditions of the area where they are found. Actually, research and protection of this organism can change the approach that we have to research and protection of other organisms and actually coral reef in general. We want to change the narrative. We want to make them at the forefront of marine conservation, basically talking about how important they are rather than talking always about how fragile and what we are losing in our ecosystems. So the question for my PhD and the question that the research group is the questions that we are trying to answer are, how many of these giants are there? What are they? Are they actually the key to coral reef resilience? Is there anything that we can actively do to protect them? That's how Map the Giants was born. So the aims are multiple. We would like to discover the biggest coral colonies in the world. We have chosen a measure, 5 meters, as a proxy to determine the size and the age of these organisms. We would like to study them, reveal the unique characteristics that make them so resistant to environmental perturbations. And finally, as I was mentioning before, we would like to turn them into new symbol of marine conservation, basically into new models. Marine monuments. How do we want to do so? We started the project less than a year ago in January 2024, and we cannot search the oceans by ourselves. We need the help of citizen scientists of any wealth of knowledge. We rely on tourists, diving centers, students, and biologists like you that have a network of researchers next to you that can help us find these giants of our ocean. We have launched a website that allows people to learn something about the corals, but also to input and report their sightings to us. What do we ask our participants? We ask them to estimate the size. Actually, you can see in the photos 2 biologists which are trying to measure colonies, but not everyone has these tools at hand, and we don't want to force people to collect data. So basically, we ask them to estimate their size with very, very basic tools like a photo of themselves, for example, next to a colony, so that we can define more or less their sizes and then eventually validate them afterwards. We ask people also to give us an idea of how the colonies are doing, because it's important to know how they are doing today, but it's also important to know how to resist to eventual bleaching events such as the one that you can see in these photos. How much of the alive tissue is present? Are the corals bleached? What happens after the bleaching event stops? Do they survive? How do they thrive? We do so also by asking our people to collect photos for us. We're not only talking about very detailed photo through photogrammetry, which allow for 3D models, but we're also talking about very simple photos, which allow us to have a first step in the knowledge of our colonies. Basically, we allow to have a time zero of the colony. And actually, if they have the opportunity to photogrammetry, this allows us to have more detailed information in the long term about our corals. Drones are nowadays more and more common, so it's very often that people are surveying areas with drones. So drone surveys offer an incredible opportunity, not only to have more or less like an accurate size of the colonies, but also to have orthophotos and basically collect more detailed information about the corals. But what we have achieved so far. So to date, in less than 1 year of the project, we have received over 95 entries. Of these 95 entries, 80 are considered, according to our standards, as giants. The remaining were either inaccurate or smaller sized colonies. And finally, we have received entries from 14 countries. If we have to be very honest, the project was born in the Maldives. Our university has a campus in the Maldives, Mare Center, and that's where we started receiving the vast majority of the data. But in less than a year, we have already received data from many different countries, from Seychelles, Egypt, Thailand, Australia, and so on. But let's see at some of the colonies that we have identified. This was one of the first colonies that we bumped literally into. It was next to a place that we have been several times. It's an Heliopora, so technically not a Scleractinian coral, but yet a reef-building coral, measuring 7 meters in length, only the portion that you can see in the photo. But there are other portions, possibly of the same colony, making the colony even bigger than this. Then one of the first colony that was reported to us by a fellow citizen scientist, it's a Porites, so a long living organism, an organism that grows very few centimeters a year. And it was 8 meters in length, 5 meters in height, and it resisted during the bleaching event. We have reports before and after the bleaching event. Finally, discoveries of the last few days, actually, of the last scientific expedition, a very large turbinaria, 11 meters in length, 35 meters in circumference. Truly a majestic creature. And again, Pavona clavus, which was discovered in the same day that the other colony came out in the news, a Pavona measuring 65 meters in circumference and 20 meters in diameter. So again, a very large coral colony. Again, from the Maldives, a colony of Goniopora, so a different type of coral in the appearance with larger polyps, measuring 29.5 meters in circumference. All these are proof that an area that was not analyzed before harbors a large amount of coral colonies, which are giant. And if we look at data that we have received from our citizen scientists, we can see this coral colony that was identified in 2018. So we're not only looking for corals which are presently alive, but also looking from photos from the past, because we can do basically a search of them and see how they are doing today. So for example, this one, we are on a mission to find this colony again and see how it's doing 6 years after it was first identified. And again, we have corals from another part of the world, Seychelles, which have only been estimated to do incredible photography, but we don't know much about them yet. So let's look into a little bit of the numbers. So of the colonies that we have reported, the vast majority of them ranges between 5 and 10 meters. Few of them, 16 colonies, are over 10 meters, and just few of them are even over 20 meters. Yet, as I was mentioning, some of them have been reported less than 5 meters in one of their lengths, so not technically giants for us. Porites seem to be the most discovered colony, which is very, very important data, because Porites, I was mentioning before, are slow growing organisms. The vast majority of the corals reported are not fully alive, but very, very healthy, with over 75% of the live tissue covering their skeletons. And as I was mentioning before, some of them were bleached, but for some of them, we have also data that report their recovery after the bleaching event. So after we have discovered these colonies, we want to perform studies, and that's where a meeting like this one today is very important, because we have scientists from different wealth of expertise which might be interested in conducting studies on these different colonies. We are open to any type of information that these colonies can harbor, not only studying them from a biological point of view, but also studying the legal aspects, for example, that could help us turn them to real marine monuments. So there's opportunities for everyone. So, and how do we plan to do so? We have launched, and we are you hear this for the first time before anybody else. We have a scientific expedition coming up at in the first half of 2025, in a scientific expedition which will happen in the Maldives, and it's open to anyone to study these giant coral colonies. We will not only try to discover them, but we will also allow scientists to study the colonies that we have already identified in this area, in the central part of the Maldives. And this brings me to the last slide of my presentation, where I kindly ask you to get in touch with us if you have any information about the giants, but also follow us, because citizen science means network, and that's basically what we are trying to achieve today. if you have questions, I'm here. Thank you.
Thank you, Federica, so much for our amazing presentation. There are a couple of questions in the chat. What is the preferred in Coral 3D model creating, drones or GPS or something else?
So for recreating 3D models, we basically use photogrammetry. So we go underwater with cameras and we collect photos. We're talking about sometimes thousands of photos from different angles. And then we use softwares to reconstruct them. So that's something that we are still working on. We have models already, but we have not put them. We are building the section of our website where this will be put in later on. Basically, the aim is to take the corals out of the ocean and bring them on land to people that have not the opportunity to see them underwater. So yes, we basically for 3D modeling, we need underwater photos.
Thank you. Another question is, are any of the models open source?
We will make them. As soon as we have figured out basically the computer technology behind it, we will put the models hopefully on the website for people to be able to see. The data are currently not the 3D models, but the data are currently available on the website. So if you type Map the Giants on Google, you should be able to see all the colonies that have been, or most of the colonies that we reported so far, with some information about them. But it's very important for us to let everyone know that if they do not want for any reason to have their data shared publicly, we do not share them. So if they feel that there is a sensitive area or a sensitive coral that doesn't need to be displayed on the website, it will not be displayed.
Perfect, thank you. Another question from the side. First the question from the chat. Could these models help us restore any giants affected by coral bleaching?
So that's it's a great question. So the models can be at time zero. So it's very important. When we were talking about, for example, the colony that was identified in Chile in 2018, there is a time zero that it's 6 years ago. So if we were to have a time zero of these giant coral colonies today, maybe we'll not be able to restore them, but we will have information for the rest of our days that we can use not only to portray, but also to study the coral colonies. And yes, if we know about the giants and we know where they are, first of all, we will hopefully avoid the destruction of them. So not only thinking about restoring them, but also protecting them from anthropogenic pressures, which might kill them. Let's make it simple.
Thank you. So the question from the side is, as a marine biologist, what do you think is the most important thing in preserving the coral reefs and protect them from all of the anthropogenic stressors?
Knowledge. So it's definitely knowledge. So the more people know about the importance for people of coral reefs, the more people know about corals in general and fall in love with the coral reefs, the more there's a chance that we can protect them. I do not think that we are doomed. I think there's still hope, and the more we are active and proactive about knowledge and conservation, the more there's a chance that coral reefs are making it to the next century.
Thank you. And another question relating to the nurseries of coral reefs. Do you have any experience with those, and do you think they are a legitimate method in restoring coral reefs?
So we're talking about restoration here, I guess.
Yes.
So most of the time when we are talking about restoration, we're talking about different types of corals. Although there's a branch of restoration which is working on slow growing corals. So working on corals that are taking a long time to grow. The vast majority of restoration works on fast-growing species. If we could compare them to the pioneering species of a forest, so we are talking about restoring the first stage of development of a coral reef, and there's studies which are also proving that when a reef is healthy, so when there's life on a reef, and that's what restoration can do, it attracts new corals to settle down. So even though we are not restoring the complexity of a coral reef through restoration, we are definitely working on a stage of development of a coral reef, which is important for the survival of coral reefs in general.
Thank you so much. Are there any other questions from the chat? Oh, yeah, perfect. One more question from me. Do you work with, not from me, from Eddie, do you work with any student association, marine biology student sections, or is it more individual volunteer based?
Sorry, can you say that again? If the project is on, yeah.
Do you work with any student association or marine biology student sections, or is it more individual volunteer based?
So basically, our project is from the University of Milano Bicocca, so from the Mare Center team. So we are a group of professors, scientists, and researchers and students from the University of Bicocca, but we are open to anyone because the project is on citizen science based. Anyone can collaborate, and the scientific expedition will actually host not only professionals, so people that have already been in the field for several years, but also students. So we are going to have also scholarship for two students to participate in the scientific expedition. So there's open to there's space for people which are in the field have been in the field for a while, but also there's space for people that are new to the field.
Question from the chat is, what is the recovery process for the bleach corals?
What is the recovery time or the process? So the process, I'll answer to both. So the process depends on the changes. So the conditions of the oceans getting back to normal. So bleaching occurs when the conditions, so let's make it simple, when the temperature of the water rises above average thresholds, and it rises above threshold for a long time, so the corals suffer, they lose their symbiotic relationship with zooxanthellae, and they basically both starve and also die of heat, not die, risk of dying of heat. But if the temperature of the water goes back to normal values, the coral can recover. It can take a few weeks up to several months for a coral colony to recover. So it really is a reversible process when the conditions are getting back to normal thresholds, but it can take a while.
Thank you. Any other question? Oh yeah, I got a question. So related to volunteer students that can join on your expeditions and your work, what are the requirements of their knowledge? I suppose they need to know how to swim and maybe how to dive or something like that.
Yes, there will be. So basically, we will make it public in a few days. They will need to have an open water dive certificate for sure. They will need to be enrolled in an MSc or a degree, BSc, MSc, or PhD students, and they need to have a CV which has some topics related to what we are doing. So basically, we would like to have people on board which want to move their career into studying giant corals in any aspect that may be relevant. So it needs to be something relevant to the topic of ecology, biology, photogrammetry, anything that relates to the study of giant corals. So you can you'll be able to find information on that on both our website, our social media page, and Mare Center website in a few days. I will leave the links in the chat.
Please do, and after you share it, Yeb will share it on their social media, of course, as well. Are there any other questions for the lecturer? Perfect. If not, thank you, Federica, so much
Thanks
for
a
this
lot.
amazing lecture. And I
Thank you.
hope that we can meet soon and collaborate
Yes.
on some other events as well.
Yes, please get in touch if you have questions or if you want to collaborate with us. Thank you.
Yeah, there's another question for you, I suppose, or just a comment of colleague Antonia. So her colleagues at Kotor Montenegro are starting a core restoration project, working with Gorgonias. Maybe there could be some student opportunities there as well. So yeah, we are 4 minutes ahead of time, but the next lecture is here. So I would just like to ask Eva to, I hope that I'm pronouncing that right, to try sharing her screen and checking everything works technically.
Okay, I don't think I have an option to share my screen.
You should now, Federica.
Oh yeah, I do. Yes, now it's fine. Right. Let me know if you can see my screen.
I can see the first page. Try second page just to see if it's rolling.
Yeah, all good.
Yeah, works. Perfect. So could we wait 3 minutes just to be on the agenda time?
Yeah, sure, no problem.
Perfect. Right, so we start at 35. So I'm just going to write down that. Perfect. See you in 3 minutes. Okie dokie, it is 11:35, so we can start with the next lecture. Let me just announce you. Feel free to share your screen. Right, so now we move on to our next lecture, titled Things Every Young Biologist Should Know, Insects for Aspiring Researchers. This presentation will provide invaluable guidance and advice for those just starting their journey in the world of biology research. Whether you're new to the field or looking to sharpen your skills, this lecture is sure to offer valuable insights. I give the stage to Ieva Andrulite. I am so sorry if I'm butchering your name and last name.
Actually, you pronounce it perfectly, and it's like a very rare occasion when someone pronounces my name correctly. So yes, hi everyone, and thank you so much for the kind invitation to speak at this conference. It's a real pleasure to be here, and I'm Yewa. I'm a PhD candidate in computational neuroscience in my last month of my PhD, based at University of Liverpool. And as I'm nearing to the end of my PhD, I'm really excited to share some of the key lessons I've picked up along the way. And I really hope that what I share with you today is going to be useful for young biologists. So I'm going to start with just talking about my academic journey before delving into advice. So I graduated my school in 2016 in Lithuania, and then I moved straight after my high school to the UK to do my genetics degree in University of East Anglia. During my genetics degree, I tried to be proactive. I did a couple of internships, joined biological societies, both in university and societies such as British Science Association, and kind of like navigated my way during my undergrad, also working as waitress because things are tough when you're a student. And afterwards, it's a very strange probably for people to see that I transitioned from genetics to neuroscience, but I always loved neuroscience. And during my genetics degree, I picked up that I really love bioinformatics. And that's why I did a couple of internships in bioinformatics and microbiology using computational tools. And only then did I realize that there's kind of a field growing in UK where I can utilize computational tools to study neuroscience. So I applied for the degree and got in the University of Manchester to do neuroimaging degree. And it was a COVID time. So my proactivity kind of dropped because even though I wanted to do many things, I couldn't. But the one great thing that happened is I got into PhD degree, which I'm going to focus on today because I think that I did quite a good job at picking up a lot of activities and learning a lot of extracurricular skills. So just to talk about my PhD, because I'm almost finished with my PhD, I did a few things. So I finally published my first paper. So I'm going to talk here about academic achievements more than any other ones. So in academia, papers are very important. So I was so glad I was able to do it in the end of my PhD. I gained some teaching experience, applied for some grants and got in. I went to France, for example, for 3 months to work with my collaborators. I presented at international conferences and attended a couple of prestigious summer schools during my PhD. But more about what is actually fulfilling, right? So during my PhD, I achieved a lot of things in terms of actually seeing the world and learning how to do public speaking and communicate science to people outside my field. Also building the community all over the world with researchers, because researchers are also people. They're not just someone who you can work with. Some people can be your friends and you can have a great time with them. And also science outreach. I did multiple projects where I just basically was teaching science to different people in different audiences, from children to adults, and I had a lot of fun with it. So I class them as my personal achievements. So before I delve into the whole kind of what skills are very useful for young biologists, I want to give you a metaphor first. So imagine yourself in a small boat drifting in the middle of the ocean at night. You have no paddle, no map, and no compass. If you're in a boat, you might eventually reach an island if you wait long enough. But what kind of island? Will it have what you need? Or will it just be empty, a lifeless place? Now picture two people in boats. One is focused on their laptop, sticking to the basics to stay afloat, and just basically floating. And another one is actually thinking ahead about their next steps and the tools they'll need to basically make the journey. So now imagine that the second person equips themselves with paddles, a map, and a compass to keep them on track. Those people are in control, or that person is in control. They can choose their destination and navigate towards the islands rich of opportunities and create a journey that aligns with their dreams because they have more freedom since they have more tools. This is what it's like in academia. If you stick to the basics, the essential niche skills, you might eventually land somewhere, but your options will be limited and your path will be shaped more by chance than by choice. So what happens if you actually try to utilize the resources that you have? Like in this case, you have paddle, you have compass, you have map. These are your resources. They let you steer your career towards exciting opportunities, like working with policymakers, communicating with clinicians, collaborating with businesses, learning how to code to be able to work on exciting projects, for example, brain-computer interfaces. In a fast-changing world, researchers who adapt and grow their skill sets are the ones who thrive. They are the ones who navigate their own journey and can actually have more freedom to career choice. If we like it or not, the world is changing and we need to adapt to it. So I'm going to start my presentation by first of all talking how to utilize research funding as young biologist. So first of all, what is research funding? Research funding is essential for driving projects forward. So research funding, essentially, the first purpose of it is for you to make your experiment work. But also, a lot of funding can be allocated, especially if you have a generous funding body, to your traveling expenses, because you want to go outside of your country to present your research, to maybe make some useful collaborations, get some great feedback. And also it can be utilized for training. So the first obviously purpose is to do your experiment, but it's more than that. You can do many more things with research funding. And it comes in a form of grants, stipendships, and bench fees. However, a lot of people have limited funds and they have to prioritize things that matter the most. And a lot of people, what happens is they have funding for their experiment and nothing else. So I'm going to talk about those cases and how can you work your way to get more money from external resources. So what is the best way to utilize research funding if you have it first? So if you are working with someone who is quite transparent about your funds, because a lot of PIs are not transparent with how much money you have in your bench fees, and those things happen. But if they are transparent, you can think ahead and be strategic about it. You can plan your experiment and think how much money you have left to dedicate that money to maybe present at the conference or develop the skill that will make you a better researcher and maybe make you will be able to have another chapter working on more sophisticated method. So try to think ahead, how much resources do you have allocated to that? And if you only have a very tight amount of resources, try to think about the grants you can apply for to help you with that. And for early career researchers, I very strongly advise that you also apply for grants even if you have enough money, because the way academia works, they see that someone is giving you money, so more people want to give you money. So I know it sounds ridiculous, but having on CV that you like having few grants on your CV, even though you had enough money in your bench fees, will actually help you get the job, because people will be like trusting you more because you know how to handle small amounts of money. And one of the most important things is no one is going to tell you what to do. You must be proactive. So your PI is not going to come to you like, oh, you have to learn the skill. I mean, some of them do, but majority of them don't have time for this. So you have to do your own research and propose the idea and try to convince them to fund that specific course or conference or project. If you have a limited funding, there is a lot in case of UK, we have some funding bodies that you can apply for to get money for conferences, for example, or for collaborations. If you don't have enough money for the data, for example, I realized in the middle of my PhD that my data is not good enough to answer my research questions. So I try to go above and beyond to find collaborators who can borrow me data, and we work together on a co-authorship paper. So basically, we work together and publish together. So it's a win-win situation. So that case, both parties win, and you also develop collaboration skills. Is this something to think about? And lastly, there's loads of free online resources. You just need to know what skills to work on. And in terms of skills, what are those skills that are extremely useful in this fast changing world? So I have a lot of skills here. I'm not going to read all of them. I think what I'm going to do is I'm going to try to talk about my personal example how I picked the skills outside the niche research world, because in a research world, they expect you to be a good researcher, writing, reading, maybe analyzing some data and knowing how to do things in your niche area, but you have to make sure you work on the skills that are more transferable, because in real world, those things do matter. Even in research, you are going to have to know how to present your research if you want to be a successful researcher, because you need to apply for grants, you need to go to interviews, like you need to master those skills. It's just how it is. So I want to just touch upon one of those, because some people might be skeptical, knowing how to utilize AI, because a lot of people demonize AI, but I think what we need to demonize is universities that don't teach you how to use AI ethically, because AI can be extremely helpful, not into finding research, no, but AI can be helpful in identifying patterns. For example, if you have a little mistake in your code, which is a consequence of a capital letter, ChatGPT immediately finds it, whereas you can spend 3 hours not understanding what's going wrong. So knowing how to ethically use AI will make your life much easier. It's not gonna think for you, but it's gonna help you get things done much faster. So my personal example, by default, as computational neuroscientist, of course, I would be learning how to write, read papers, use some softwares to analyze the tools, but I tried my best to think about it a bit more broadly and more applicably. So I tried to learn coding. Coding is something that we as computational neuroscientists have to know, but if it was all about me and my supervisor, it would be just R and maybe Bash for some certain things. But I tried to learn Python. I tried to work on MATLAB. I tried to kind of learn a bit more, to go above and beyond, to make sure that I have more opportunities to utilize different statistical tools and packages if I want to do that. And another thing is science communication. I'm super passionate about it. And I try to just seize all the opportunities to do it, try to participate in workshops, talks, and collaborations to basically stand out as a computational neuroscientist, because a lot of us, we are actually very shy and we just want to just do our research and don't talk to people. So just me talking to you makes me stand out a little bit more, because I'm able to do that. But it was never easy to me, to be completely honest. I was struggling a bit, but I tried to work on it. So I know I have 1 minute left. So I have 2 slides to quickly go about how can you plan and be strategic about the skills. One thing that's extremely useful, no matter if you're a PhD or postdoc, is put priority lists. So maybe if you have a dream job, go and look at the description and try to look up the skills those people have, and you can work a little by little on them. Or if you are someone who just don't know what's going to happen after your postdoc or PhD or master's, I don't know what level you're in, you can try to think about competitive skills in your field and try to work on them more. Skills that always useful in every single sector's leadership, collaboration. They're always going to be useful. Those 2 soft skills are something that you can always work on. But hard skills are a bit more specific. So just try to work on the skills that are in demand in your field. And talk to people. People are your friends, and you can just approach them on LinkedIn. Obviously, don't write long messages, because those things are not really going to help and no one is going to reply to you. Just be nice, approach, hi, I see you're working this company. I want to learn more. And people are happy to talk about their experiences. So key takeaways: be proactive. Science about building bridges, not walls. So you need to make sure you're equipped with those skills to be successful. And yes, try to look for funded PhD or postdoc programs or fellowship programs, because those are the ones that give you least amount of stress. And those are the programs that actually care about your self-development. And self-development is important because that makes you a better researcher. It's not selfish. So yes, wrapping things up. Thank you so much for listening, and I hope that was helpful.
Thank you so much, Yeva, for a beautiful presentation. I believe that it's something that a lot of the participants, especially master students, are thinking about now. So I think this was very helpful. Now it's a Q&A section, and there's already questions. So first question is, do you think gaining academic and technical skills through online courses with certificates that we didn't get a chance to work on in uni is relevant for international positions? What would be an alternative?
So if you're talking about hard skills, I think having to be honest with you, if you can have any project, that would be much better than a course. So it depends on technical skills. If you're talking about coding, you can try to maybe form a team and work on a project on GitHub. That's much more valuable than you just putting a certificate from a free course. But again, it's better to have the course than having nothing. But you having a project, even if it's your friends or a group of community basically trying to learn how to code, you can still use the courses, but you are basically working on a common project, trying to code something right now. And that kind of project is going to stand out much more. So I really recommend projects, but I also think that those courses are really helpful to shape your foundations. I personally have a few, when I apply for jobs, it really depends on a job nature, but I do have those courses in my CV if they are competitive by invitation, or if they are free, then they have to be very specific to that specific job. So I wouldn't just put all courses I ever attended. So I hope that's helpful.
It sounds like it was to me. Next question is, what do you recommend to start? What do you recommend to start a social media account about scientific divulgation?
I don't really know much what you mean by scientific divulgation. I'm not sure if it's because my language barrier or my scientific gap, but I don't know. But generally, when I'm talking, scientific accounts, Instagram, and or you can do it on LinkedIn, on Twitter, I think for me, it came very naturally. I was having my personal account and I started posting it and then just naturally found my community. So it wasn't something like I started to create it to get followers. In the beginning, I just posted my stuff, my life, and my life became my PhD at some point, and I found my community. But I think it's maybe thinking about what you want to share with people. Because if you find that community, it's much easier to create content because you are here together. You kind of support each other. So if you, I don't know if you explain to me. No. But for example, if you do content about neuroscience, try to find other neuroscience people. Or if there's no one, just try to find someone doing science, and then you kind of find each other and do basically have this community together and see what other people post and what's of interest. But at the same time, you have to be authentic. You just don't post the same thing. So for me, it came very naturally. I didn't have I still struggle because I kind of treat it like a personal account, not like a business account a lot of times. So it's hard for me to give an advice.
Thank you. Leila says that your presentation was really motivating and thanks you for it. Her question is, what would you recommend to people who do not have resources to go to courses, summer schools, etc.?
So it depends if you're a PhD student or a postdoc, because if you are, you must be eligible to apply for external funding for those things. Those competitive schools, for example, the one I went to University of Washington, they pay for your accommodation for 2 weeks. So all you have to find is the money to fly there. They pay for your food. They give you food tickets on campus. So some schools are competitive, but they pay you to get there. You just have to obviously, and then I applied for funding to get my tickets. So essentially I didn't use a single penny from my wallet because I tried to just get that money outside. But I don't know which country you're coming from, because that really depends on a country, and those things probably work different between the countries. Because I'm in UK, and in UK you can apply for a few charities that can help you out with that.
Thank you. while we wait for another question, hopefully one of my questions. So I have no experience with grants and funding in the academia, at least not yet. So my question is, after you get money for your PhD, for your research, what are the conditions? Obviously, there's probably going to be some report, but how do you deal with possible failure in the scientific aspect if you can't prove something or something doesn't have expected results? Does that affect the grant? Is that any type of problem to deal with?
Well, to be honest, you can't fake the data, and I don't think there is failed results. Well, it depends on the situation. What do you mean by failed? Is it because there is no significant result? Because non-significant results are also results, and we would be a terrible scientist if we just try to make them significant if they're actually not. But if there is any other circumstances, like in wet laboratory, things happen, and sometimes resources get wasteful. I mean, with grant money, this rabbit hole in academia is that the more you get, the more grants you get, the more you're gonna basically the more you can get, the more you will get. So this is a rabbit hole. So if you basically got a big grant and can have some even if you, for example, got no significant results, and that happens, if you can still write up a good thesis explaining why this happened, because non-significant results can be still great results and you can publish them, that's fine. I don't think PhD people gonna be very judgmental about it, because PhD is a learning opportunity. You are a student, so I don't think that should impact. But again, academia is toxic in that way because they want to see a lot of money on your CV so they can give you money, which doesn't make sense, right? If you have no money, how can you get money? Like, make it make sense. So that's why I'm telling, like, even if you get funded PhD, please apply for grants, because it's toxic and you just need them. My toxic advice to toxic culture.
It's adaptation. Yes, thank you so much for that answer. Are there any other questions for the lecturer? Right. So another one of my questions is what would you define as kind of the most difficult part of your career so far? You can say anything, but like just generally in your path of career, what would be the hardest?
I think the hardest times were so far was my master's degree, because I had no money. I had to work for my rent, for everything. I had no support from family, so that was hard. And COVID made it worse, and I got ill. So I had pneumonia for like 6 months. So that was very difficult to navigate. But when I'm talking about PhD, so that's why I'm so positive about PhD, because I'm like, I went through that hell. So even if it's hard for me, it seems like, it could be worse. I could be just having pneumonia and feeling bad all the time. During PhD, I think the hardest thing is loneliness. I think it's fine. You have to find your community, and if you can't, you can just feel so lonely and no one understands you, because if your friends are not doing PhD, they just think you're a student, right? So they don't really understand the struggles you're going through. So if you can't find academic community, and for example, I'm so glad I have my Instagram community, because yeah, I find my academic community abroad more than at my local place. So it's every time I travel, I love people, but in my lab, I think maybe personalities don't align. Sometimes those things happen, and you just can feel a bit lonely, especially if you don't make good friends in your lab. So that's probably a challenging thing, I would say, in doing PhD in generally.
Yep. Thank you so much for that answer, and thank you so much for this amazing presentation. Hopefully we'll work together soon on some other events in the future. So yeah, thank you so much.
No problem. I'm going to stop sharing now.
Perfect. And I believe that our next lecture is already in the meeting. So Mislav, if you can just check the sharing and all the technical things.
Yes, I'm here.
Perfect.
And this cringe moment now, sharing the screen and asking whether you can see it. Still after 4 years, I find it a cringe question. Wait, I'll put it in the presenter mode. Can you see the screen?
Yep, yeah, we can see it in the presenter mode. Can you just do the second slide so we can see if it's moving? Yeah, okay, it's moving.
Great.
Okay, let me just announce you. Okay, so we move to our next exciting lecture titled Not Too Basic Career Advice for Early Stage Researchers, Things I Wish I'd Know Sooner. In this session, our speaker, Misal Basic, will share invaluable career advice for early stage researchers, offering lessons learned from personal experience. Whether you're starting out or navigating the next steps in your career, this lecture promises to provide practical and insightful guidance. Mislav, you have the stage.
Thank you, Davor, for a very kind introduction, and thank you so much for inviting me and allowing me to choose what I'm going to present. And as you already kindly introduced, I will talk more about my personal journey, reflecting on challenges like imposter syndrome, perfectionism, comparing myself to others, how I tackle them, and how I grown on this academic journey. And this is definitely, for me, exiting my comfort zone. My comfort zone would be talking about either my master thesis research or a PhD research. But I think as this symposium has really people coming from different disciplines, students coming from different disciplines, that maybe cancer research is not for everyone, and that this will have more value. And definitely, I think that it will be relatable to everyone, and it is transferable to any career stage you are currently facing, whether you're a bachelor student, master student, PhD student, even if you have some postdocs, if you're in industry. So I hope you will enjoy this talk. And I would first like to briefly introduce myself with this scheme that I made so that you understand why am I here today and why can I talk about certain challenges and how which academic journey I had to overcome them and to actually understand what are the challenges. So originally I come from Croatia, and it's where I did my bachelor in University of Zagreb in molecular biology, and I always, since my high school, knew that I would like to go abroad for the masters. And especially studying science in Croatia, I wanted to go abroad because my wish was to go into the places where the science is done on a cutting edge level, because I was reading about it throughout my masters, and I wanted to understand how these people behave in these labs that have basically limitless funding, and how they decide which fancy experiments they will do, whether they just do experiments and then they see whether they get results, and so on. And that's why I decided to move to Heidelberg in 2020, the best year to move abroad, because that's when the world stopped. So this was sarcasm, but I'm really glad I made this decision. And my bachelor was more basic biology, I would say. And for my master's, I wanted to more specialize into some field. And I really found love for cancer biology. And that's why I came to Heidelberg and joined the major in cancer biology in the Molecular Biosciences International's master program. And then throughout my master's program, actually, I realized that just doing this science and research is not enough for me, and I have additional questions that I want to ask myself. So I want to get additional skills. And then basically I became very, very interested in startups and startup world, and became part of this Nucleate. It's an international organization for startup development, and I co-founded a German branch and served as a co-managing director for a bit more than a year. And during my time in this organization, I actually realized that I don't know a lot about entrepreneurship in life sciences. And as I'm a nerd, I wanted to learn it in an organized way, and also where I get a piece of paper for it. That's how my parents taught me. You always need to have a paper for things, for skills. I mean, it's not necessarily always, but I was still thinking in that way. And that's why I joined this postgraduate diploma in entrepreneurial project management, which is organized by Sorbonne University and For You Plus Alliance. And basically in the same year, last year, in the same year, with one month difference, I finished my master's program and my postgraduate program in entrepreneurial project management. And after I finished my master's, I worked briefly in industry for 5 months because I wanted to see how it's there. I was always listening horrible stories about working in industry from academic people, and actually it was very fun. And now we are so last year I started my PhD. I stayed in Heidelberg because I love the city, and I stayed at my institute, German Cancer Research Center. And for my PhD, I investigate therapy resistance mechanisms in acute myeloid leukemia by doing single-cell multiomics. A lot of very fancy, expensive experiments that are coming from clinical trials. And so basically, I fully shifted from wet lab to bioinformatics, and I really enjoy world of bioinformatics, especially now with all developments with AI and machine learning. And today I'm here, and it's 2024. I finished first year of my PhD, and I would say for myself that I'm more than just surviving as a PhD student. These are all pictures from this year, because everyone was talking to me when I started PhD, you will see during the first year that you will suffer a lot and you will have no life. But I was like telling everyone, I won't allow for that to do it, because PhD is just one professional aspect of my life, and I want to have something else. Okay, this introduction was a bit longer than anticipated, but okay. And let's go to see what's on the menu for today, what's on the agenda. So I decided to have 3 different parts. And in first part, we will talk about being uncomfortable about imposter syndrome, about perfectionism, about comparing yourself to others, and why is that a trap? And then we will become comfortable by becoming self-aware, uncovering our hidden potential, understanding that not everything what we do has to be splendid, perfect. And also we will understand how to compare yourself to others and to whom compare yourself to. And in the end, we will decide, although we got comfortable, to choose growth over comfort, and highlight that magic happens outside of our comfort zone, understand why soft skills are power skills in any career path. And when it comes to these soft skills, I represent what I use as hacks for effective time management and personal branding and authenticity. So I did a test talk for this talk. Normally, I always present in a different way, and I hope we will be able to go through everything. But if not, we will just tell me to stop, and the lecture is designed in the way that it doesn't need to reach the end. And first, so I would like to talk about imposter syndrome. Here I asked, and imposter syndrome, in my opinion, is elephant in the room. And if you are not familiar with this phrase of elephant in the room, so elephant in the room is something that we all see, we all experience, but we behave as if it doesn't exist. So we don't talk about it. And I ask here ChatGPT to basically depict an imposter syndrome as an elephant in the room. And it's a room full of academics, and this giant elephant is standing in the middle of the room, and no one is talking, no one is doing anything about it. And we want to change them. And when it comes to imposter syndrome, I would like to share my personal experience with imposter syndrome, and really had big troubles with it back then. So this is me here in front of my institute, and in the lab practical in November 2020, when I just moved to Heidelberg. And basically, I was very insecure during my bachelor's, and then I thought, after being admitted to one of the best universities in the world, Heidelberg University, that suddenly I will be amazed by myself and feeling this self-love, self-awareness, self-worth. But during this November, it was this very harsh realization that these checked boxes in a CV don't build a feeling of self-worth and self-love. And actually, then I felt worse than before coming to Heidelberg, because I looked around myself. I came into this amazing international community which is taking most talented people from all over the world, and everyone seemed far more experienced than me. And then I really started doubting the decision of selection committee of university. I thought that university made a mistake by selecting me. For example, in this practical course, we were doing ELISA essay, and I have never done it before because I never had a such kind of immune practical. And I totally messed up the experiment, and that was the end of the world for me. And also I started thinking that maybe they selected me just to fulfill the quota because it's the international program and they needed to have someone from Balkans or from the Eastern Europe, because the science in these parts of the world is kind of not the best funded, and then they want to kind of have someone to show how good their program is and how good they are. And also a big problem back then was my perfectionism, because I was feeling huge imposter syndrome, but I was also in this vicious cycle of perfectionism. So nothing that I did was good enough, and essentially to start anything, basically to start preparing a presentation for like a journal club, a seminar or something, to start even writing my to-do list for the day, I needed a lot of time, and it was super unproductive. And also, even I mean, I had quite some academic successes back then, also before that November of 2020. But still, my approach to it was that all my successes are just routine, and it's something that I planned and expected. And when I would get this thing that I want, I would not be really happy. But on the other hand, if I would fail at something, which would rarely happen, but would happen, then it would be the end of the world. I would not speak to anyone for 7 days because I need to be perfect. And now everyone is thinking of me, how imperfect I am. And that was really a struggle. And the third thing that really was very difficult was this comparing myself to others. So this is the picture of my master's program, 22 very talented students. And when I was comparing myself to other students in the program, I was thinking everyone is better than me, everyone is far more experienced, they all did fancy bachelor internships in the States or in the institutes around Europe before the masters. And also I was thinking if only I could be like them, then I would be happy. And I was avoiding using LinkedIn or any kind of science-related social media because that would be just making this feeling of me being not enough 10 times worse. But then I kind of decided through different workshops that I attended and also a lot of reading and so on to get comfortable about myself, because I realized that I could not go further in that state, and it was really difficult. So when it comes to getting comfortable, I think that becoming self-aware is very important. And self-awareness is termed as understanding your strengths, weaknesses, motivations, and values, and how they influence your decisions, productivity, and interactions. And here I would really like to highlight these strengths, because that's what's the most important. And also, when you become self-aware, you also see others in a different way. So you see their strengths, but you also see that these people have weaknesses. And then you understand that it's balanced out. So for example, I can have some weaknesses and other person has strengths in these fields, but some other person will have weaknesses in fields in which I have strength. So it's balanced out. We are all human beings in the end. No one is a superhero. I mean, still, I think for some people that they are superheroes, but they are rare. And I think that's really a key thing to mitigate feeling of imposter syndrome, is building your self-awareness. It literally kills imposter syndrome. And how to become self-aware? So this is a very good method. Is this Johari window, where basically you have different areas. So you have known to others, known to self, that's your open area. Then you have something that's not known to others, and it's known to you, that's your hidden area. For example, some hidden potentials or hidden weaknesses. Then you have this blind spot. Blind spots are things that others know about you, but you don't know them about yourself. And you have this area that is not known to others, not known to me, that's unknown. And basically what's the aim of this exercise? It's to expand the open area by filling it with your strengths and consistently refining and developing them. And how you do it? So first of all, you need to reflect on your achievements and abilities. And then really, by reflecting on your achievements and abilities, you can really understand what are your strengths, and you can decide to actively work on them and actively showing them to other people. But also what's kind of very interesting, it is blind spot, because here are the strengths that other people see in you, but you are not aware of them. So they're already there, but they're not known to you. And here, basically, what you just need to do is encourage feedback to uncover these blind spots, and you need to be open-minded about it. And then basically, in my opinion, is the best way in the beginning to focus on strengths in your blind spot, because it's sustainable. It doesn't need a lot of energy. Afterwards, I will talk about this unknown. But yeah, let's move forward. Then when it comes to perfectionism, so perfectionism kills productivity. That's and full stop. That's like some people are claiming they're perfectionists and think that it's very good, very desirable to be so, but that's not true. And everyone who wants to be productive knows it. And basically, the thing is that not everything we produce has to be perfect. And there's this cutoff between perfect and good enough. And for example, in my case, for me, in the beginning, everything had to be perfect. Like even my to-do lists had to be perfect, because if by accident someone would be reading my to-do lists, which is impossible because they're on my private Notion account, but you know, when you are when you want to be perfect, you don't care. So I would spend so much time in the morning to do my to-do list, to phrase the tasks, and that just, I mean, doesn't make sense. For example, example of something that should be perfect is if you have a defense talk for bachelor, master thesis, PhD thesis, there you should really seek for perfection. But on our daily tasks don't need to be perfect. They can be good enough. And then, so also when it comes to these comparisons with other people, it's basically this scheme from Lukas Stengel. It's a LinkedIn creator. It's the zoom out, where you basically think that life is very good up there, but then you actually, when you zoom it out, realize that the person that you are comparing to yourself to is also comparing themselves to someone else. So when you become that person, you will be, there's again someone better than you, and you will never be happy. And that's really this vicious cycle, and that's why comparing yourself to others is trap. I think you should use others as inspiration, but basically you need to compare yourself to person who you were before, and that's when you will be seeing growth and changes, and that's honest comparison, because you are comparing the same person through a sequence of time. Okay, and we are coming to our final part, which is choosing growth over comfort. And then when I become comfortable about myself, I understood that it's my comfort zone, and basically I wanted to understand, to gain new knowledge about me, to gain new knowledge in general. And that's when I decided to start stepping out of my comfort zone. And this is, for example, a depiction that we have in not my office, but in the office where I sit for a PhD, that the magic is happening outside comfort zone. And I really think it's the case. And also by exiting your comfort zone in this Jahari window, you will uncover this unknown area, your strengths that are unknown to you and unknown to others, because you will expose yourself to other situations in which you need different areas of your brain, you need different areas of your personality, and you will understand your new strengths. Also some weaknesses, but we are focusing on strengths. And when it comes to getting out of the comfort zone, it was also very nicely said in the previous lecture, it's about the soft skills. And I think they are really power skills because they are transferable to any career path. And unfortunately, that they're often not really covered in academic education. So, and in the end, the case is that someone with average performance but very strong soft skills will quite often be seen as a better candidate for a job than someone with high performance but poor soft skills. And soft skills are confidence, time management, self-management skills, ambition, focus, empathy, enthusiasm, public speaking, networking, leadership. There's like an endless list of them. And for example, one very important soft skill, especially to me, because I like to be efficient, is effective time management. For example, let's say you have this to-do list. For the sake of time, I won't go through I won't read all of the examples, but you can read them. And it's basically a list of, I think, 12 to-dos on that day. And here we really need to define what needs to be done first, and also what doesn't need to be done by us. And a great thing to help with that is this Eisenhower matrix, where you basically project your to-do list in a two-dimensional way. And you have this important and urgent area. That's something that you need to do that day. You need to focus on that immediately. Then you have important, but not that urgent. That's to plan and prioritize. You have things that are not important to you, but they are urgent to others. Those are things that you don't need to do. For example, those are some administrative things that in your lab, your secretary, whose job it is, can do. And she might pass it on you because, I mean, it's easier, but you can kindly push it back to her. But always be kind in a nice way. And then there are things that are not important and not urgent. These things we don't do. We delete them from the to-do list. For example, here it was this spending hours scrolling through academic Twitter on LinkedIn without a purpose. We always need to have a purpose for our interactions. And then we solved our to-do list. So we know what we need to do that day, what we need to do next week, what we will give to someone else, and what we won't do. And also another example is this 80-20 principle, which is basically proven thing. There's this great book. I just recently finished reading it, but I've been practicing this principle for longer already because I learned about it in this entrepreneurship project management postgraduate program. But basically what it states that 80% of your results will come only from 20% of the efforts. And basically you need to focus on small actions that will deliver the biggest impact and remove the noise. This is this 80% which doesn't give you any goals. And for example, in research, 20% of your experiments will lead to 80% of your key findings. And you really need to focus on doing these 20% of experiments to put a focus on that. But example of wrong usage of this principle would be, for example, I assume reading literature is not part of the critical 20%. So I skip thorough research because it's it will not help me achieving my goal. And then I spent a year on a project that's already published. So that's terrible. My whole time was used in vain. And we will finish with this aspect of personal branding and authenticity. So here my advice would be, and this is very uncomfortable because here you are showing yourself to the outside world. But I think it's important because it will help you with networking. It will help you with finding your positions. You will be able to get a job offer without applying somewhere or get an academic position without going through selection. And it's really amazing. So my advice would be to perceive yourself as if you were a brand. And then in your brand, you need to identify your strengths and unique skills, and that is the core of your brand. For example, in my case would be, so I'm very knowledgeable about all the single cell omic techniques and getting knowledgeable about it, and that's not so often still. And also, I don't know, I think I'm good with public speaking, with networking and so on, very open to the world, very open to the growth mindset. And that's how I'm branding myself. Also, you need to have at least 2 elevator pitches. So elevator pitch is basically a up to 1 minute talk or up to 1 minute explanation of what you do in your research. And one should be more for people that are in science, that it's more scientific, and another one should be more for people that are outside science. And also you need to use social media strategically to showcase your expertise and build connections, but you need to remain authentic and remain humble. And when it comes to that, for example, being authentic, it's these two different captions on LinkedIn. So one would be for me that I'm a PhD student and German Cancer Research Center. Here, I'm not authentic because there's 2,000 PhD students at German Cancer Research Center, and most of them have this description on their LinkedIn. So in your LinkedIn bio, you put the core of your brand, your skills. Also for me, for example, is that I have a degree or diploma in entrepreneurial project management. And it will basically help showing it will help the algorithm of LinkedIn showing your profile to the people that are either seeking for someone with those skills, with that brand, or others that have the same. So, and this description, the first description being a PhD student, it doesn't help the LinkedIn at all. And second thing is these posts on LinkedIn. So I think a lot of people see LinkedIn as a very, very toxic network because you always see this, I'm thrilled about, I'm announcing, I'm the best, and so on. I put here example of bad and a good post. And so two rules, if someone is taking notes, first, you are never thrilled on LinkedIn because this is always used. This is like a default thing. If you ask ChatGPT, it will write that you are thrilled. And if I think most of the people that see, or at least me, when I see that someone is thrilled about something, I don't read it. Or I just read it if it's people that I know because I can be a bit biased. And second rule, you are never announcing anything because you're not a Buckingham Palace in UK that's announcing a birth of new heir to the throne or something. You go straight to the point. You need to, in my opinion, write. For example, here in my case, it was the master's degree. What you do as a master thesis, give some context, what you learned. You need to definitely thank the people that helped you on the way, because that's how we are humble. We show that we are human beings. You need to acknowledge people, and also you need to write about what's next, because maybe if your next step is working in academia or in industry, maybe someone from academia and industry will see this post and they will contact you via LinkedIn and offer you a position. So that would be about LinkedIn. And basically, I think I'm a bit over time, so this is the end of my talk. And I'm really glad. First, I would like to thank the Young European Biologists Initiative for inviting me. And this really helped me to finally start my Instagram account, Not Too Basic Scientists, which is like an extension of this talk. I had this idea for past year to do it, but also as a recovering perfectionist, I keep procrastinating. And now I thought it's the perfect idea to do it. So it will be like a retrospective diary about completing my master's degree in Heidelberg, more detailed extension of this talk because there's a lot more things to discuss, and also live tracking of my PhD journey. So if you would like to stay connected with me, you can have a look. There are already some posts there, but I have endless ideas for the posts. And I also plan to also shift to LinkedIn at some point and so on. But for the beginning, Instagram is kind of my comfort zone because there are not the group leaders and PIs of the labs in Heidelberg and so on. And basically, I'm at the end of my lecture. I want to thank you for your time because your time is precious. It's our most precious resource, and I'm open to questions.
Thank you, Mislav, for a really, really amazing presentation. I loved it. I think all the colleagues did as well. Everybody, feel free to write your questions in the chat. Until then, my questions. So I love the presentation. It's very heartfelt, and it's very like it hits the right spot. Both yours and Yeva's presentations are really, it hits home. So my question is, as we brought from Croatia, and I think a lot of Croatians are thinking about going to study outside. What would be, and that is obviously a hard thing to do, living home and a new area and everything. What would be your advice to minimize all of the negatives of leaving home, going to a new place, starting your PhD there or master's and so on?
I mean, that's a good question. And especially, I think quite often this going abroad, especially in my case, is seen by my friends and colleagues from Croatia as something amazing, as something that has there's no bad things about it. But the fact is you are far from your family, and you are far from your initial friends, childhood friends, and so on. And that is difficult aspect of it. But nowadays, I think it's easier with, for example, social media to remain in contact, or especially also I use with my parents WhatsApp video call. I think we have a video call every 2 days. And then after some, it gets you get used to it after some time, and also you need to focus on the positive aspect. So you get so much in return for making this sacrifice of going abroad. And also you don't need to stay there forever. You can go and do a master's degree abroad for the sake of your experience and come back. I decided to stay abroad for a PhD. What I will do after a PhD, I don't know. And also it's, I mean, you can in between always return home, and you need to plan that you spend, I don't know, Christmas, Easter, this big holiday, summer at home, and that's really what helps you. So I think it's a journey, but it's making sacrifice, and you get a lot of return, and in the end you will overcome the sacrifice.
Thank you. We have a question in the chat from Nitkos. Excellent presentation. Since many tend to try to achieve a perfect results, how would you recommend escaping the trap of perfectionism? Aim for the best possible outcome? ?
Yeah, that's a really good question. So, I mean, I don't think if we have perfectionists here that necessarily this talk or my answer to this question will fully mitigate this problem, but could open your mind about solving it. So for me, first thing would be definitely that, I mean, if you're a perfectionist and if you do some things in a way that's not, that you don't think is perfect, others will still see it as perfect. And it's like, I don't know, for me, it was about how the pictures on the slides are aligned, and even I shift slides that the pictures are in the same field and so on. But then, I mean, no one notices that. Or like, I don't know. That's first thing. So still, if you do things, if you're a perfectionist, but do things in a way that's not perfect to you, others will still see it as perfect. And that's kind of a hack. Then other thing would be this identifying what's good enough. So, I mean, perfection is something that's limitless, and we need to have some cutoff. And identifying what's good enough is really good, because then you have some aim that you want to achieve. And the third thing, I really suggest reading about this 80-20 principle, because there you can see that you don't need to use so much time and resources. You can basically allocate 20% of your time and resources to get 80% of the results. So basically, by allocating a bit more than 20%, you will achieve 100%, which will be perfect or good enough. I hope it's very philosophical, but I hope it helps.
Thank you so much for that answer. And another question from the side is, how do you deal with failure, specifically in academia and science, regarding your results or just simple mistakes?
Yeah, it was also something I struggled a lot with, especially back then when I was hardcore perfectionist. The way I approach it now, I'm learning. I'm on a learning journey. I'm a PhD student, so it's like a trainee, and I have right to fail. I have right to make mistakes, and that's how you learn. And I made some mistakes in the past, and I'm grateful now that I did them, because they made me understand how to solve them, how to get away from them, and I learned a lot about how to approach them. And failure is not bad, and I mean, when you still have energy to rise from it in the end. But when you listen to all these greatest scientists and so on, I've been last year to a symposium where there was a Nobel laureate, and he was talking about failing. And I mean, it's very normal, very human, and especially in science where you cannot predict the outcomes.
Thank you so much for that answer. Are there any more questions for the lecturer? Going once, twice. Right, perfect. Thank you, Mislav, for this amazing presentation and for collaborating with us. We hope to meet you in some other event in the future.
Thank you.
Thank you. Now, the next lecture is either not coming or she's going to be late. Sadly, she's from Ukraine. No, not sadly that she's from Ukraine, but she is from Ukraine, so she has an excuse for not being here. Maybe she will join us later, but there's a possibility of another power outage, so most likely not. Until that, we will welcome the next lecture afterwards. That is Emrah Handimovic. Emrah, you can just check the sharing of the screen and presentation.
Okay, can you see it? I hope it's working.
Yep, can you just do the second slide? Yep, perfect, it works. Right, so let me just announce you. Right. Right, so next we have an incredible, fascinating lecture titled A New Method of Making Antidote and Adequate Manipulation of Venomous Snakes. This session will dive into groundbreaking advancement in the development of antidotes and the safe handling of venomous snakes, a topic crucial for both medical science and wildlife management. With venomous snake bites continuing to pose a significant risk in many regions, this lecture promises to offer new insights into effective solutions and safety techniques. Welcome, Emrah, you have the stage.
Thank you. First of all, I would like to greet everyone present. My name is Emrah Hamidovic, Doctor of Veterinary Medicine from Sarajevo. I'm a member of a snake farm South Africa, where our main job is to make antivenom who will save lives of human and animals. And through this short presentation, I would like to give a closer look at how it all works. Of the 3,000 described snake species, 600 are venomous and only 50 species are deadly to humans. Venom is a specialized secretion produced in modified salivary glands used primarily for hunting prey and then also for defending themselves. Snakes, they don't perceive humans as a prey, and in most cases they will deliver a dry bite. That's a bite without injecting venom because they want to save their venom for their prey. And it's estimated that up to 50% of snakes' bites are dry bites. That is significant reducing the risk of the humans. Although snakes are, how to say that, considered less popular animals in the society, they are incredibly important for our ecosystem, as they are the first line of defense preventing the spread of all diseases transmitted by rodents. It is up to us to raise awareness about the importance of snake in nature, to break the many disinformations we offer here today about snakes. Understanding snakes, both non-venomous and venomous, is not important just in veterinary medicine. Also, it is crucial in human medicine, biology, pharmacy, and many other fields. Studying the composition and effects of the snake venom is important due to its diverse applications. Some of them are analgesic effects, where peptides from venom can block pain signal from a nervous system, and they are being researched as potential painkillers for human and animals. Also, anticoagulant effect, where venom contains components that influence blood clotting. Antihypertensive effects, certain components inhibit enzymes that raise blood pressure. Anti-cancer effects, peptides from snake venom have shown the ability to target tumor cells. But why is searching about venom and venomous snakes important for us is to make antivenom and help patients who get bitten by venomous snakes. The process of venom extraction requires specialized training. Every approach to venomous snakes must be highly focused and also carried on with a high level of confidence. Snakes, they can sense our emotions, so it is crucial not to project fear into the animals, as even the smallest mistake can cause the venomous bite. During the procedure, our primary goal is to protect ourselves and also to minimize the stress to the snakes. Snakes do not seek interaction with humans, and our presence already causes them some level of stress. There is some some equipment that we can choose to avoid these like bites or also stress to animals. First, we will have protective footwear or boats if we know that we will be approaching snakes on certain terrain. Then protective gloves, which are resistant to punctures from sharp fangs. Protective glasses. If we know we will work with snakes who can spit their venom, such as wrinkles, Mozambique spitting cobra, or some other species of cobra. Also snake hooks, snake tongues, and snakes tubes for quick and safe immobilization of a snake. Measures that must be followed before and during work with venomous snakes are crucial for both. I said personal safe and protection of the animal. Here are some of the most important steps. Number one is knowing the species and the way that the snake give a warning behavior, as well as observing a snake in terrarium or a transport container. This provides insight into the animal's current state and allow us correct preparation. Number two is being informed about availability of antivenom in the moment of when we work with snakes, and also into procedure for providing first aid in case of a bite. Number three is maintaining the safe distance from the snake. On this photo, we will use an example snake of 1 meter long, whereas the zone within 1 meter is a dangerous zone. Up to 2 meters is relatively safe zone, and beyond 3 meters is considered as a safe zone, which allows time to react if the snake moves towards us. Number four is avoiding stress factors that for snake in the moment of working is the most important is the right temperature. If we work in a room with a low temperature, it can cause lethargy or shock for the animal. But if we work opposite in the really warm room, it can cause dehydration, increase stress and aggression. It's important to strictly follow all the safety rules and to approach venomous snakes only if it's really necessary. Only when we, after ensuring all mentioned safety measures, we providing proper working conditions, we can begin the process of making antivenom. Antivenom is filled with antibodies that perfectly match the proteins in the venom, binding them to and preventing them to causing harm in the body. Currently, there is a global antivenom crisis due to a lack of data and demand and distribution, as well as the complex production process. The snake farm model, our model for antivenom production, aims to simplify the process and expand coverage to as many countries as possible. Most people are familiar how the antivenom is making today, but I will just explain short. Large animals, the large mammals, mostly horses, are injected with small doses of venom and prompting their bodies to produce antibodies. Then blood is drawn from the horse and plasma is extracted using specialized instruments. This plasma is used as antivenom, but this method is really long, more than 1 year, sometimes also more than 18 months for the horse's body to produce a right number and quality of antibodies, which is risky if we have a patient who need a antivenom quickly. Also, this method is expensive and risky for experimental animals, mostly horses, because incorrect dosing can easily result to the animal's death. Another drawback of this method is that horse plasma, after we take a horse plasma, it must be purified and to remove unnecessary protein components that could cause adverse reaction in humans. The snake farm model seeks to replace this method by producing antivenom in just 1 hour and using just one snake. Venomous snakes are naturally resistant to their own venom, which protects them from accidental self-bites or bites from other of the same species. These snakes already have antivenom circulating in their blood, and their blood can be used to create antivenom for humans and animals. This process involves drawing blood from the snake, then waiting 1 hour for plasma to separate, and we use that rich plasma, who is rich of antibodies, as a final product, and that is antivenom. This method also can be further simplified. Everywhere in the world where venomous snakes live, there are also not venomous snakes that prey on venomous species without any adverse effects. Blood from such non-venomous snakes can be used to produce antivenom for venomous snakes. A prime example is the king cobra in America or file snake in South Africa. They can eat almost all venomous snakes. This approach eliminates the need for direct contact with venomous snakes, avoid using another animal such as big mammals, and skips the waiting period for antibody production. There is a video of like 3 to 4 minutes that all of this method of snake farm method making antivenom is explained in more detail. So I hope it will be more clear after this video. I hope it will play.
Antivenom was developed by the Pasteur Institute in 1895. Venomous snakes are milked for their venom, which is then injected in small doses into a large mammal, most often a horse, over the course of a year or more. This stimulates an immune response, leading to increasing antibody levels in the blood. That blood is drawn after the allotted time has passed. The antibody-rich portion is separated from the rest of the blood, purified to be safe for human use, and then bottled as antivenom. However, it's expensive to maintain a horse for over a year, dangerous to milk snakes regularly for venom, and ultimately too time-consuming to be cost-effective. As a result, a number of pharmaceutical companies have simply stopped producing antivenom. That's what we're here to change. Snake Farm has developed a process that takes advantage of natural venom resistances, allowing us to develop an antivenom product at a fraction of the time and cost. Anywhere that venomous snakes occur, there are non-venomous snakes that prey upon them. These non-venomous snakes have an impressive venom resistance that allows them to safely feed on their deadly cousins. We use these snakes to make our antivenom, immediately removing the need to keep anything that might be deadly. These snakes require no immunization period to raise antibody levels in their blood. Our research has shown that certain snakes have antibody levels that actually far exceed anything a horse-based product could produce. And unlike horse antibodies, snake antibodies require no additional purification. Snake blood is then immediately centrifuged to become antivenom, with the entire process taking place over just 1 day. And while large quantities of snakes are needed to ensure we can produce a high volume, we can maintain a colony of more than 200 snakes over 1 year for less than what it costs to maintain a single horse over the same time period. Our facility in San Marcos is responsible for research and development, as well as antivenom production, and currently houses almost 400 donor snakes.
Our facility in Los Angeles is where our serum is fine-tuned and every batch of antivenom is tested for quality and efficacy.
We have published patents on the formulation of this antivenom in both the United States and internationally, and our antivenom has been independently safety tested to ensure it is not toxic. In short, we have developed a more potent antivenom, ready for use within 24 hours at a fraction of the cost, with no need to keep any venomous snakes. Clinical trials of our product have begun in nearby hospitals outside San Diego. 50 canine patients have already received our antivenom prior to any other treatment, and all have gone home healthy, some in under 24 hours.
The trials have proven our serum works better than that of our competitors, and we can produce it inexpensively and in large quantities anywhere in the world where we know customers are waiting. We intend to serve the veterinarian market, which will treat 50 to 70,000 dogs per year in the US alone, and then move quickly into the human market, which has an attractive top-line potential for our product. We are the future of antivenom. We are Snake Farm.
And we sincerely hope that this method will soon replace the now currently in use as and try to reduce the number of deaths caused by snakes, which exceed 130,000 per year. Also here you have email address of the snake farm and my email address for every other questions, or also internship opportunity at snake farm in South Africa. Thank you for your attention.
Thank you, Maya, so much for this amazing presentation. Now there's a Q&A section, so feel free, all the participants, to ask any question you have for the lecturer. Perfect. And until the first question, there's a question on the side and my question. So my question is, do you believe that as this method is something that will, develop more, do you think that as these farms will obviously grow, and obviously you need to have a certificate to work with snakes, but do you think there's going to be some mishandling? And it's something that happens with capitalism. Obviously at some point people were starting to get these companies started growing snakes and doing this, but in that time there could be a mishandling of snakes. So do you think that the concept that snake farm uses is so good that could create this type of situation?
Well, I think this method of the snake farm method of making antivenom, it can replace this method currently in use, because it's more easy to keep, I would say, like 500 snakes than 500 horses. It's way easy to feed them and have a snake farm than farm of large mammals. So like per year, the snake farm can make enough antivenom for all Africa, whole continent of Africa, and also it's sending to the main zoos to the Asia and America, which is good number of coverage of the at the moment. But for Europe, I'm not sure, because I think we have enough antivenom. We are not producing our antivenom, but we have enough. I mean, we don't need that much. We have just 3 venomous snakes here.
Thank you. And another question from the side is, how can volunteers, students, young researchers join snake farms? So what is the procedure to do that? And is there any requirement for previous knowledge?
It's not, because I also start like that. I was student third year of my university, and after I finished my university, I went there in South Africa. I had experience with non-venomous, but with venomous snakes I didn't have. And they have 10 days, 2 months, and 3 months program, which we basically start from beginning. What is a snake, how to approach a snake, how to handle, first non-venomous, then venomous, then more and more till the end of how to make antivenom. So the best is to send email of the first snake farm, South Africa, where is the director of the snake farm will respond and say every detail of, but no knowledge before is not necessary.
Thank you so much for that. We have 2 questions in the chat. So thank you for the great lecture. I wonder how common are snake bites in cats and maybe other companion farm animals. We know that a lot of people keep outdoor cats despite the negative ecological implications. So do you have any experience with that? And can the antivenom be used on the animals as well?
Yeah, I have information of my country, Bosnia and Herzegovina, where in human medicine, we have enough antivenom, but for veterinary medicine, we don't have any. And that is a huge problem because of domestic animals, mostly large domestic animals, are bitten by venomous snakes because they live during the summer, they are outside. Also, some dogs, like dogs, hunting dogs, they can also be bitten by venomous snakes. And what we're doing, we're treating the symptoms because we don't have antivenom. That is the main problem, but more often are dogs and large domestic animals.
Thank you. Another question is, do most life-threatening snakes have a predator with resistance to their venom?
I'm sorry, can you just repeat the first part?
Sorry. Do most life-threatening snakes have a predator with resistance to their venom?
Yes, like I would say black mamba, cobra, they have file snake, which is not venomous snakes, and they can easily eat all of them, like puff adders, cobra, mamba, and all other the most venomous snakes. Also, there is a Australian python. He is also, like, I would say, the only python who would also chase the snakes and eat. But our pythons that we use as pets, for example, the ball pythons, they are resistant to our venomous snakes here. For example, our Vipera moditis.
That's a cool fun fact. So Margareta asks you, thank you, Emma, for this insightful talk. Are all currently developed antivenom monoclonal, or are you aiming for polyclonal as well?
To be honest, I'm not sure about that. I think it's monocolor, but I'm not sure 100% because I'm more based off just how to make antivenom and mostly help animals, the patients, because I'm a doctor of veterinary medicine, how to help them in the way of a venomous bite.
And another question from Helena is, thank you for the lecture. Do we have to pay to have an internship in the snake farm?
Yes, but it's changed. Depends of the which time. Right now is summer. It's opposite than here, so it's more expensive in summer because it's more work. During the warm temperatures, snakes need more care. They are more aggressive. It's really more dangerous to make antivenom than during their winter. So I would say the best is to apply where is the less expensive, around maybe May, June, something like that.
Perfect. Thank you for that answer. Are there any more questions for the lecturer? If not, we can continue. Thank you, Emra, so much for this amazing lecture. Hopefully, we will meet soon in some other event as well. And I would ask Maya to, if she's here, our next lecturer, to try the sharing and technical parts.
Hello, everyone. Greetings. I will do the informal thing just as soon as I think I make this. Let me just, I've never actually shared screen on Zoom, but let me see. Okay. Is it good, or can I move this somehow a little bit to the other side?
When you click on the sharing part, there's three options. If you click just share the screen, then we will see your entire screen, and it's more efficient. But right now, just click on the, if it works. Okay, we can see it. Try sliding to the next slide.
Okay, so it works.
Perfectly.
It works. Okay, great. Just let me know, do you see this like Mentimeter that I have just entered on Google?
I just see the presentation.
Okay, then, okay, just a second. Okay, how about now?
I can still. Oh, okay. I can see the Echo Brains there. So.
Okay. This would be just a short interactive part in order to not call out anyone by their names, just to like brainstorm a little bit for like a minute or two. Nothing spectacular.
Perfect. Let me just announce you and you can begin with your lecture. So we're going to our next lecture. Maja Ibricic will talk about the importance of youth engagement in the field of ecology, sustainable development, and environmental protection. This session will explore how the next generation of leaders is crucial in shaping the future of our planet. Maja, you have the stage.
Thank you very much, Davor. Hi, everyone. Now the formal part. My name is Maja Ibricic. And considering my educational background and the degree I obtained, I'm a Bachelor of International Relations and Diplomacy. But the part I actually love more is youth participation, youth engagement. And this is why I have decided to at least try to cover. There's a lot of topics, as you can see. And I think that we could have actually a whole seminar about these topics. And I am going to try to present the youth engagement perspective from my experiences, and I hope that you will be able to catch up with some useful advice. In order not to be so formal, I have sent you guys the link to the Mentimeter. I think that everyone who actually clicks on this link can enter the Mentimeter. It's a short brainstorm game. I just want to see your perspective. Basically, it's totally anonymous. And what I want to see, if anyone, let me know in the chat, if no one can access the Menti throughout the link, I will provide a code. I just want to see your guys' opinion on youth engagement, youth activism, advocacy campaigns, basically anything you think is important when it comes to. You can add up to 3 answers, I think. So I think it will be visible to everyone if the screen is shared correctly. And let me know if you were able to enter this Menti throughout the link. If not the link, I will provide the code.
I can enter it through the link, and there is the option for 3 words.
Yes. You don't have, it's not obligatory to put all 3 words. So yeah, let yourself go, write whatever you think, what actually reminds you when you talk about youth activism, being passionate about bringing positive change into your societies, anything literally that comes up to your mind. I just want to see what are we starting from. Great policy changes. I like this one. Don't be shy, just literally type anything you think it's important. Passion, great. Fighting, I love this. Nurturing, great. Community. Proactivity, yes, great. Interdisciplinary work, well done. That's also really important. Collaboration, great. I'm trying to catch up with all of these. Multitasking, definitely. Innovation, yes. Motivation, definitely. Okay, great. If anyone still wants to write any additional words, you're welcomed until I share my presentation on the screen. I really like these responses because this is something that definitely is narrowly connected to the presentation that I'm going to show to you today. Let me know if you see the presentation well. Now, is it visible?
Yes.
Okay, great. Great. Sorry if I interrupted anyone who maybe wanted to add anything else. So as Davor mentioned in the beginning and in this like introduction part, I'm going to talk about youth engagement importance in the fields of ecology, sustainable development, and environmental protection. What are we going to talk about today? Yes, like I said in the beginning, there are lots of things that I am aiming actually, my tendency is to cover all these topics that are written here, but I believe that you guys understand it is actually really hard to cover all of these and to be as concise as possible and to remain in the adequate context. What I want to share with you, I believe that you have heard so many useful things throughout this. Oh, sorry, something fell down in the room. I apologize. I think that you have heard many important things today, and something that you will advocate for in the future. And that's why something to start with is youth as leaders, basically leadership development, advocacy campaign elements. Then we are going to cover the empowering and decision-making processes, because we want youth to be actually included in future decision-making processes, not only to remain onwards. We want to be actually asked and to be a part of decision processes from the beginning to the end. I apologize in advance if this might actually sound a bit too political for you. Trust me, it is not. It is something that I believe all of us need in our work, to advocate for the topics that we are actually interested in. We definitely want the changes in many fields, and this is a crucial element of that. Paving the way to a successful activism. Some steps. I want to emphasize that this comes definitely from my personal experience. Something that I think is actually really important while building your activism and your path. Then benefits, obstacles, and how to overcome them. That's something not many people actually tell you. It happens a lot, and they don't tell you what to do. So you're stuck in your head. Oh God, what should I do next? I'm stuck. Nobody listens to me. What is there to do? And then shortly, because I probably won't have enough time for that, I will tackle the topic of engagement in green entrepreneurship, local community perspectives, and green business, in order to also tackle the topics of sustainable development. And we know that green businesses, green entrepreneurship, and these local community perspectives are really important. I believe that all of you have heard about the phrase think locally, work globally. Okay. When it comes to the leadership development, what I want to emphasize from the start is the definition of youth participation and why is it important. In advance, I apologize, maybe I have included too many memes in the presentation, but yeah, we'll get there. However, when it comes to youth participation, I consider it really important, and I believe that all of us, by attending such symposiums as this one, are already participating. And that's something that is truly important to have in mind. Basically, youth participation, I think it's really self-explanatory, but in order to be as precise as possible, there is the definition that you all can see, that youth participation actually refers to how young people can get involved in processes, institutions, and decisions that affect their lives. Of course, you can be both active and passive. So yeah, when it comes to this passive part, how about no? Definitely, that's not the solution that we want to go to when it comes to this. And you guys already being here means that you are active participants, that you are engaging, and that you are ready to engage in various fields. That's why I like when you mentioned in the Menti part of brainstorming that multidisciplinary actually approach, that you're engaging in any sort of field that actually is of your interests. Passive means you're willing to allow whatever happens to happen, and you're not changing or controlling the situation. Of course, as youth, we are not able to bring the laws to the table, but we cannot let things just pass by. That's why it's important to actually develop the leadership. I have put a few bullet points when it comes to developing successful leadership. Basically, the most important part for the beginning would definitely be identifying people with whom you have a good relationship or around whom you feel comfortable. I believe that throughout your work, from any aspect, you know how much this means. The social capital actually is crucial here, to have people that you're comfortable with and can work on certain problems, topics, whatnot. And then thinking about developing social capital in different directions, actually, That means basically targeting the group that you're most, again, confident with, that which you can collaborate with. And basically, I wrote this, that while more senior allies can remove roadblocks and provide valuable advice, you also need junior allies who can support you as a leader and a mentor, whether you are going to decide to be a leader in a certain group. And I think this is really important. The inclusion here, the inclusivity part is really important when it comes to this. We want to reach for the unheard. We want to be a voice for those who want their opinions to be stated in the places of decision-making processes. Also, being proactive, I think that someone also mentioned proactivity in the mentee part, is basically being proactive in reaching out to offer help with something even before someone asks you. So that means sometimes problems in the sphere of environmental protection, ecology, are not in the media, are not exclusive news, but that doesn't mean that the problems are not present. We are facing many problems nowadays. For example, in my country, Bosnia and Herzegovina, we are facing natural disasters, floods. Also, we had heat waves, but that doesn't mean that the other problems, such as air pollution, illegal waste dumps, and anything, are not present. That's why we need to be proactive and basically cover all this up. Be gracious also, helping and fulfilling someone's requests, but this is really important, being honest about what are you able to do. Communicating as well, staying in touch during times when you don't need anything. I believe that we have all experienced like amazing conferences, amazing seminars, and then the contact basically disappears. That's why it's important to communicate, stay in touch, maybe creating some sort of alumni programs and similar things. Also, being considerate of what you ask. Don't put an ally in an awkward or sensitive position. And thinking about who you consider your allies to be, as I mentioned in the beginning. How do you feel with certain people? Who do you want to be a part of your team in order to be proactive? Advocacy is something that's really important when it comes to environmental protection questions, when it comes to ecology, when it comes to sustainable development. What I want to emphasize here, that advocacy, must go beyond words. So it is amazing to have a plan, but I will show you what my experience has shown me throughout my activist experience and how to actually advocate. Basically, what advocacy is, it focuses on doing something to support, recommend, or implement actions that are linked to an idea or cause that you care about. I tried to keep this presentation as general as possible because I think that all of you, as I have been able to see from the agenda, are advocating and learning and researching about different things. That's why I haven't really I didn't want to stick to specific problems, just to like broaden this so that this can be actually applicable to all of you when you advocate for a certain topic, when you advocate for your research, when you advocate for preserving nature, when you advocate for the environmental protection in general. Basically, this is bolded for a reason, that all people are actually entitled to a say in the decisions that affect them. So yeah, like I said, being passive is definitely not an option, because we have to engage when something is of our interest. Maybe even if it's not of our interest, that's the altruistic part that we want to see here, helping others in need. Basically, when you advocate for an issue, you hold those in charge accountable to ensure that your rights are protected and upheld, that rights of others are also protected and upheld. Also, advocacy means that you make your voices heard as often as ideas and practices you want to change. And this is also important, like some things actually affect the most vulnerable people in society, and we have to advocate for them especially. What's also important, that you are not asking for favors. Actually, when we are advocating for something, we feel ashamed. We don't want to be maybe annoying or anything, but that's actually not the issue. If we are provided with enough information, we are not asking for favors. We are actually bringing change to the table. When it comes to advocacy campaign elements, everything starts with you and your personal background. That means you don't have to be a fast speaker and a person that talks a lot, such as I am. That's definitely not the thing. You can have various actually identities throughout advocacy campaign elements. That means you can be either a researcher, speaker, writer, or implementer. What I want to emphasize here is the fact that every position in the team is equally important. It doesn't mean that if someone shows up to the conference, to the seminar as a speaker, that the people behind him or her are not actually important. That's why you can be anything basically that suits you the best. That means that actually, like I said here, advocacy has a place for everyone. Each individual brings a unique set of skills and abilities that play a role in the processes. Maybe I'm not such a good of a writer because maybe speaking is my stronger thing to do, but that doesn't mean that someone else won't help me throughout this process. This is why teamwork actually is so important. You can gather a group of people who collectively provide all the skills and abilities needed to effectively achieve your advocacy goals. And I think that you have actually been able to see how this all is connected. Advocacy and leadership, like I mentioned in the beginning, you need a team, and then we are connecting it to the advocacy campaign. Basically, everything is causally consequential here, and really important to follow. When it comes to fact finding, I believe that all you people are actually researchers in different fields, and that you have been through this process lots of times. Basically, fact finding divides to additional elements, such as choosing your issue, gathering information, conducting research, and expanding personal engagement. 5W, I believe that you are also familiar with this. We have to be concise. We have to be clear to the public, actually, opinions. I know that you and I can understand others that come from the same branches we do, but we have to make our messages actually understandable for the general public. That's why whenever we work on a certain problem, whether that's air pollution, water pollution, building illegal dams in the rivers, hydroelectric plants, whatever that issue is, we need to answer these questions: who, what, when, where, and why. And this is connected to creating and generating our message to the public. And actually, nowadays, as a part of the Gen Z, we know that messages that tackle us the best are the messages that are kind of intriguing. So that's why it's important to, you can see this, I won't read all of this, because I think that you have also heard about the SMART method when we need to implement our advocacy in order to our goals to be specific, measurable, achievable, and to actually be in time, in some deadline. Also, what's important when it comes to advocacy is actually understanding, there's a typo, I'm sorry, in understanding there's a typo, understanding and engaging with policies. That means in identifying the right policy and reading and understanding policies, whether that's anything you're interested in. It's important to see what aligns with your topic, and whether there have been also present, maybe that you already have documents in something and you will use them. Maybe something when it comes to, for example, ecology, many things have already been ratified and are embedded in law but are not implemented. That is why it's important to like dig in and find what's the most useful thing for you. Also, what has actually been the most important thing for me throughout my activism experience is networking. And this online symposium and other conferences are a great thing to be for networking. At least that mailing list will provide you additional contacts that you can basically contact in the future. Like we saw in this previous lecture, people are already asking, how can we engage in some sort of organization. And that's the best part of networking. We are coming out of some seminar or a symposium or anything with additional acquaintances that are crucial or will be crucial in our future. I know that networking sounds intimidating. Not all of us are extroverts like I am maybe, and some other people, but it is actually important to maintain contacts that are useful for you. Also fostering allyship in any form. Of course, it takes commitment. People will knock the door in front of your face, but that doesn't stop there. You can actually find allies among other groups. That doesn't have to be youth, maybe some professor, media, whoever. But it's important actually to build partnership. This is something I have already been talking to throughout networking. Attending conferences and meetings with decision makers is definitely important. And what I think is crucial part here is identifying the jurisdiction when it comes to your problem. Not everyone works with a particular matter that you are working with. So you have to actually map the people that are actually responsible for the problem you're advocating for. These are some additional actually advocacy campaign elements. Finding space for engagement, whether that's a non-governmental organization, organizing events, engaging with media. You know that we have to be present in the digital space. I know that you all are familiar with this. We have to network. People usually make jokes about me because I always put things on Facebook and I write, oh, I was honored to attend this and this. I know that might be annoying to some people, but actually it's important. I and you guys can be a great motivational example for other people. Yeah, I think I don't have much time to actually cover everything up, but when it comes to youth and as decision makers, I will definitely provide this presentation to the organization actually team, so that you guys can go through it a bit more profoundly. There are lots of challenges when it comes to youth engagement in decision-making processes. That is definitely inequality, lack of mutual respect and trust, because people in the decision-making institutions or anything don't believe that we have enough expertise or anything, but that shouldn't definitely stop there. We should fight for voicing our opinions. Also, poor education and training, that has been actually an example in Bosnia and Herzegovina as my homeland. Cost, of course. This is actually something that I will show you guys throughout the QR code that you can scan later on. This is from a document I have been working with my colleagues from UNICEF, and this is actually how you can engage in municipal level, regional and national level when it comes to advocacy campaigns. Actually, who are the subjects that can help you and who are your partners in the PR topics. We call them stakeholders because they are actually important for something that we are advocating for. Of course, this basically wouldn't be without some motivation, and I believe that you all have witnessed youth and brave people actually changing things. That's, for example, who is from Bosnia and Herzegovina knows how small organizations from Bosnia and Herzegovina were able to gather people from the region, maybe even above the region, to protect our rivers, to protect our forests, to protect our environment as a whole. And yeah, come on, do something. Actually get involved is something that we want to share with our peers, because becoming involved in activism at a young age can shape how someone takes part in social and environmental issues throughout their life, because that will change actually the way people think, and they are going to be able to recognize problems from the start. Also, something that is dearly mentioned is creativity and innovation that youth have, and that how they make actually everything more mobile and how they make everything interesting. So that's some sort of positive weapon that we have among us. Yeah, very nice, great success. A bit Borat reference. This you can see like what are some steps to a successful activism. It's also connected to the previous topics that I have tackled, it's important actually to identify the cause, to set clear goals, to educate, to network, to be direct in the action, whether those are protests, lobbying for a certain law. And I think that also important part is monitoring progress and adaptation to everything, because when we formally finish some sort of project, that doesn't mean everything is over. We have to monitor what changes we have brought. Unfortunately, I think that the time is up. I actually won't be able to cover something that's connected to the engagement in green entrepreneurship, but actually that means building businesses, building actually public discourse in your local communities, because it's always easier to tackle the change among the people from your community, and then further develop some sort of policies when it comes to environmental protection and green entrepreneurship to a national level, and then of course to the even a regional and maybe a global level as well. Actually, local communities are a crucial part in green entrepreneurship and actually sustainable development, because not actually cities differ and local communities differ. That's why we have to adapt changes in our local communities. It's important for our economy, for our sustainability. And of course, we are facing many challenges that can be overcome. This is something that I just want for the end to emphasize. What if we get stuck? What if we think that change isn't near the horizon? It's not visible, it's not happening. Get support. Don't be afraid to contact people, to be even a bit boring when you have law, you have knowledge, you have research, you have everything behind your back, and that's why you can get support from everyone. Don't try to do everything by yourself. Have allies, have friends, have friendships, and tackle the change. Also remember that you're not alone. That's why I'm also asking you guys whether you need any sort of help with advocating for a change, whether you need contacts, feel free to contact me anytime. I think that my mail is also my social media is available, and turn to available resources and people. That's also really important. And actually some motivation that actually I'm sick of flosculas and the cliche quotes, but this is something I really resonate with. It's when young people have the energy and creativity to solve the environmental crisis we face, they definitely do, and empowering them is not an investment, it has become a necessity. Why is this important? So I want to motivate you guys, and I hope that I have accomplished my mission. I want to motivate you to stay active, to be resilient, to advocate for a change, to advocate for knowledge, to advocate for research, because all of this is, like you've seen maybe from TikTok, from Reels, very demure, very mindful, and very cutesy. For more information and the strategic documents that we have worked on through the UNICEF, through the Center for Ecology and Energy that I am a part of, are actually here on this QR code. When you scan this QR code, you will get access to my link tree, and there you have really important strategic documents that are covering something that I actually wasn't able to cover because the topics are really broad. You can actually access throughout this QR code. You can screenshot this, or when the presentation is provided to you later on. Thank you for your attention. Also, again, meme. Sorry guys. It's just professional deformation with memes, I guess. Please participate and don't think that you cannot change anything, because you can change things with the useful information. Please make your research visible, advocate for a clean environment, and always be brave when it comes to advocating and being an activist. Thank you.
Thank you, Maya, for this beautiful presentation. We already had a question in the chat. How can we, as individuals or as a part of an activist group, pressure the implementation of already existing documents, example on nature protection?
Okay, that's a great question. I think that, for example, Aarhus Convention, that has been really important. I'm going to picture this in a certain way. I hope that I will be able to provide a proper answer. People think that if something is not implementing, we don't have a right to it. We have rights. That's why it's important to maybe even address some people from the law, because you, as a citizen of a country that has ratified a certain document, whether that's a convention, whether that's some other sort of treaty, you have a complete right to use everything from that treaty. For example, you have a total right to ask a certain, I don't know, a minister or a member of the parliament for information. Let's see, that's something that comes up from Aarhus Convention. Gather a team, write some sort of letter to a certain person that has your problem under his or her jurisdiction, because they will have to provide you with information because they have ratified the documents. It's also important maybe to develop some sort of digital campaign to remind that we have these rights because our countries or any sort of other decision-making institution has ratified these documents, and these documents are our protection as activists. We have the right to transparency, we have right to defend ourselves, and I can actually tell you guys to maybe search Foundation ACT, and also see how they were able to actually implement these international regulations to protect our rivers. I hope that I answered this successfully.
Thank you for that answer. If there are any more, there's more question. Thank you for this thoughtful lecture with adequate meme choices. Do you have any advice about dealing with apathy among youth? Sometimes it can feel like people want to engage in advocacy only if they get like a special invitation of sorts.
This is an amazing question, and maybe I wasn't able to tackle this enough throughout the presentation. I have had this problem for years. Kids, young people, children, youth in general are changing, and their demands are changing. This is totally honest from my side. Trust me, you have to offer benefits to the youth because everyone wants to get something from being active. I know, first thing that comes up to our minds is money. No, that's not always the issue. You cannot pay people to be active. You have to awaken things in them by actually portraying a positive example. You being actually the positive example among your peers will definitely motivate them. That's something from my experience. People are contacting me on Instagram, on Facebook. Oh, Maya, how can I go to this and that conference and seminar? Please tell me, how can I also go to the spa center as you're going? I mean, yeah, this is a bit humoristic, but it's actually leading to a point that you have to offer benefits to the youth. You have to say to them that by being actually active, they are building their resumes, building their CVs, building their future, building their acquaintances, actually providing for their own future. It's not, this won't maybe tackle everyone, but if it tackles 2 or 3 people, it's enough for the start. And the next thing you do will tackle even more people and so on. It's important to actually, summa summarum, it's actually important to offer benefits of the work, to emphasize why is it important to be active member of the society and what are they actually going to get from that.
Thank you for that answer. Is there any more questions?
Thank you.
Thank you. Perfect. And just a few more minutes until the end of the first block. So here's my question. So YEB is also a network of young researchers. We are trying to expand the reach and obviously build a network. So what would be your advice on how we as a network of biologists, biology researchers, and biotechnologists could somehow basically do this, try to promote nature protection, try to work in that field as well? So what are our options and strategies?
Okay, these topics, actually topics of biology, ecology, environmental protection, sustainable development, at least in the region that I'm coming from, that's the Balkans, are considered as some sort of conspiracy theory. That's something I was able to see throughout social media, through people's comments. There's always lots of laughing reactions, but I think it's really important to first and foremost define the target group that you want to tackle with your engagement, whether that's other colleagues from the same branch, from other biologists, researchers, whether those are some other stakeholders, and then some sort of PR campaign should be created. What I have been able to see as a young advocate, actually real life experiences are something that tackles people the most. Filming what you're doing with, for example, I hope that she won't get mad at me for calling her out here, but my friend Leila Mujakic is from a different field that I am. She is a biologist and I am from international relations and diplomacy. And whenever she talks with me about her research, anything she has been doing, I'm always amazed. What is this? It's really amazing when someone shares their personal experiences, and that's something maybe I would like to see on your social media, actually young biologists and scientists doing something in real life and actually maybe simplifying the message to a general public.
Thank you for that answer and the advice. We will definitely look into it and try to adapt to that strategy. Thank you again for this beautiful presentation, and thank you for collaborating and being our lecturer.
Thank you. Thank you for inviting me. Once again, sorry for not being able to tackle all of this. It's actually a lot, but I hope that you guys are going to stay motivated, are going to actually keep up the great work, and please don't hesitate to contact me for anything. I'm here for you. I'm not saying this just because I'm a lecturer. I'm serious about this. Write me, ask anything that I might have not answered today. I'm here for anyone when it comes to advocacy and being active in the local community.
Thank you so much. So regarding the rest of the agenda, sadly, Daria, our lecturer, won't be joining us today. She might be joining us later if she responds, but we will definitely call her to other events in the future. Based on our agenda, we are supposed to end at around 11:40, but with one lecture or less, we ended it earlier. So we are going to have a lunch break now, and we will start the event again at 2 p.m. With BioBalkan Collab block. So the meeting will still go on. We'll put just a background right now. You can leave or you can stay in the meeting, leave and join us later, and yeah, whatever you choose. We hope that you will join us for the BioBalkan Collab initiative. We have 3 really good lecturers. One of them is a TED Talk lecturer as well. So thank you all so much for being here so far, and we will see you in 2 o'clock. Hello, everyone, and welcome back. I see that all three of our lecturers are already here. So, hello, Irma, Eva, and Milica. Right. So based on our agenda, we are supposed to start the lectures at 2:15. So I hope that's okay that we wait until then. While we wait, the lecturers can try their presentations, try the sharing, and to see if everything is working correctly. And the order is going to be like the agenda. So Dr. Milica is going to go first because of the time difference. It's night at her place, and then I think it's just a second. Then it's Dr. Eva, and then it's Irma Popovic. So when you get the time, try your presentations sharing, so to see if everything is working correctly.
Mine is working, right? Okay,
yeah,
great.
I saw it. Yeah.
Okay, great.
Is it a full screen?
It's full screen. Maybe try
Okay.
going to the next slide to see if it's moving.
Yeah. There. Is it okay?
Yeah, it's okay. It works.
Okay, great. Perfect. Thank you.
Thank you.
Hello, everyone, and welcome back. Today, now we are in a second block of today's symposium, and this is the BioBalkan block. And today with us, we have three lecturers, PhD Iva Njunic, MSc Irma Popovic, Dumjovic, and PhD Milza Stankovic. They will talk mostly about power of people, discovering new species with citizen scientists, community engagement in river protection and nature conservation, and of course, unlocking potential of seagrass meadows. How can they affect climate change mitigation? So I would kindly ask our lecturers to start. And first we have PhD Iva Njunic as Milza Stankovic, sorry. She will be presenting first. Milza, the stage is yours. Of course, feel free to write questions in the chat, and at the end of the lecture, I will read them out loud.
Thank you. Thank you so much, everyone, for having me here. Good afternoon to everyone here at this symposium. If you hear rain a little bit, I'm based in Thailand and we are in the middle of the heavy monsoon season. So you might hear some heavy rain, and I apologize for that in advance. Okay, so I'll just share my presentation. All right. Okay. I think you see the full screen right now.
Yes.
Yeah, okay, great. So first, I just want to introduce my lab a little bit. So we are based in Thailand, not in Bangkok. So originally I'm from Serbia, but I've been in Thailand for now 15 years. I did my master and PhD here, and now I'm working as a lecturer at the university. I am in seaweed and seagrass research unit. So we have many different topics related to seaweed and seagrass ecology, to the animals, from benthos animals to marine mammals like sea turtles and dugongs. We do blue carbon. We do also seaweed biodiversity, genetic biodiversity of the species, and so on. So every student or every member of our team has a different topic that he's focused on. Another affiliation that I'm part of is a dugong and seagrass research station, also based in the same university, where we specifically have two different sections. One is more scientific based in terms of ecology and biology of dugongs and seagrasses. And we have several students now. One more from Malaysia is coming. So currently we have students from Thailand, Indonesia, and Malaysia, and we have one more starting this semester. And also we have another part which includes veterinary care and research about marine mammals, specifically dugongs and turtles and dolphins as well. Okay, so in terms of the marine climate change so climate change is about to affect global sea level rise. So by the end of the century, it is expected that that the sea level will rise between 0.28, so from 30 centimeters up to 1 meter, depending on different scenarios and depending where you are on the globe. But also global mean sea surface temperature is also projected to increase from approximately 0.5 degrees Celsius to up to 4 degrees Celsius, depending on the scenario and again, where you are in the globe. All of this will have heavy consequences on the marine ecosystems, as well as animals that live there, and the whole collapse of the ecosystems. So we have a natural carbon sinks, that are naturally occurring in the world. So we have the fossil fuels and land use changes that are that are emitting CO2 emissions. This is the newest publication where we have very large numbers of fossil fuels and CO2 from land use changes that are being emitted every year. But we also have natural sinks such as terrestrial forests and oceans that absorb that CO2 and kind of create balance of CO2. Currently, this last publication is saying that we have a budget in carbon imbalance where more CO2 is being emitted in the atmosphere than the carbon sinks are absorbing that. So the natural carbon sinks, everyone is familiar with any kind of terrestrial forest, such as tropical forest, boreal forest, and temperate forest. But not many are familiar with coastal ecosystems such as salt marshes, mangrove forests, and seagrass meadows. Where if we compare the carbon burial rate, which is the accumulation of carbon in the sediment, it is much higher than what we normally see in the terrestrial forest. And that carbon in these coastal ecosystems that is being stored in the plants and as well in the sediment is called blue carbon. So it's basically carbon that is stored in the mud. And that carbon, as I mentioned, is mostly stored in the, just one more. Okay, so mostly stored in the sediment. So we see for seagrass meadows, salt marshes, and mangrove forests, this number is very high, and the green color represents actually the carbon that is stored in the trees, which is much lower compared to the tropical forest, where about 80% of the carbon is stored within the tree, but 20% is stored within the sediment. And when you have a natural disaster or you are cutting a forest, that 80% of the carbon just immediately gets released back into the atmosphere. And that 20% of the carbon from the sediment quickly, because you clear the forest, just emits back into the atmosphere. But when you have mangrove forest and seagrass meadows, that proportion of carbon within the within the sediment is much different. So we have about 60% in the sediment and 98% of carbon in the seagrass meadows. When you remove both vegetations, that carbon gets very slowly released into the atmosphere due to anoxic conditions of the sediment. So these ecosystems, so all these blue carbon ecosystems are globally distributed. So mangrove forests are mostly distributed in tropical and subtropical regions. Salt marshes are in temperate and subtropical regions, and seagrasses are distributed globally. And yeah, so if you look at the global map of seagrass distribution, there are about more than 70 species globally, and they occur in 159 countries on all 6 continents except Antarctica. But they cover about 0.1% of the ocean floor, which is very minimal coverage compared to mangrove forests and salt marshes. Southeast Asia is a diversity hotspot for seagrasses, where it can occur up to 15 species occurring together. In Europe, you have, I think, 2 or 3 species, or maybe, yeah, I think there are 2 or 3 species. The most common one that everyone is that you can see when you go to the beach is Posidonia oceanica. But in Southeast Asia, in one seagrass meadows, we can have multiple up to 5 species growing together. So the seagrass meadows occupy very shallow coastal waters. So this is the example from Thailand. You have very nice beach and hotel. And as you go deeper in the water, you have a huge seagrass bed. They occur in intertidal area and subtidal, which is a little bit different that compared to temperate region. So we have very high and low tides. So the seagrass meadow during the low tide gets completely exposed, and it's very easy to walk and collect the data. And as I mentioned, they occur as a single species meadows and as a multiple species. So in this picture here, you have the big Enhalus acoroides, you have Halophila ovalis, and Cymodosia. So this is the example of 3 species, but we have meadows here that have 5 or 6 species growing together. And they provide multiple ecosystem services. So this is a summary of all the ecosystem services for the seagrass meadows. They provide food for dugongs, sea turtles, for many type of fish, especially the ones that we are eating and we are relying as a food. And they also provide nursery grounds of many important fish, and other animals. They provide oxygen, they decrease water velocity, and so on. But today I will be mostly talking about their ecosystem service in terms of the carbon storage. So the carbon that actually comes to seagrass meadows can come from multiple sources. So in the reef-associated meadows, so when we have coral reef, when we have seaweed and seagrass meadows, up to 28% of the carbon can actually come only from the seagrass meadows. 26% can come from the seaweeds, and 18% can come from coral. So seagrass meadows act as a sink, as a trapping, and have a high trapping ability, where they trap all the carbon that is coming from adjacent ecosystems. In the mangrove-associated meadows, the situation is very similar, with much higher number coming from seagrass meadows, so approximately 40%. And between 30 and 66% is the carbon that is coming from mangrove forest. So you have the fluctuation and the dynamics between several ecosystems and carbon moving from one ecosystem to another, or the third one, or it is going all the way to the deeper ocean. So currently I am working on a project where we are looking the carbon connectivity between multiple complex ecosystems, where we are looking in terms of the carbon, macroalgae, seagrass, or mangrove and macroalgae, different type of macroalgae, as well as the corals, and to see how much the carbon is is connected between all these ecosystems, and how much carbon is actually being buried within each of these ecosystems, so we can actually quantify it. However, especially in Southeast Asia, the seagrasses are declining. Based on the literature review, it's about 2.82% per year. This is picture 1 of our study sites, where most of the seagrasses has been eaten. That's why it's so short, and it looks like it's clipped. It's being eaten by turtles, but as you can see, that there are a lot of boat activities in the seagrass meadows, which again disturb the whole ecosystems, and they are being degraded. However, the recent study from the whole region is saying that this number is much higher, which is about 4.7% per year. But as you can see here, that a lot of the meadows in Thailand, I think that is increasing, but in a few slides, I will show you that it's not really. And the global study for tropical Indo-Pacific is saying that our seagrasses are kind of slowly declining, but the whole region is very much data deficient. So I wouldn't really trust these estimates, especially for this region where the data is such sparse. You can see here that half of Indonesia, the whole Indonesia has one point for data collection. So the threats to the seagrass ecosystems are mostly coastal development, sedimentation and runoff, destructive fishing like trolling, where the nets are thrown in the seagrass meadows and thrown in the seagrass meadows to collect the fish and then scraped along the bottom. So destroy the whole ecosystem and aquaculture. And these threats have been the same for the last 30 years. So nothing really changed in the last 30 years, which is a little bit sad. And what happened to the carbon that is being stored in the seagrass ecosystem? So from a healthy seagrass ecosystem that is a carbon sink, we have a carbon source. So the carbon, it gets slowly emitted into the water column and then slowly into the atmosphere. So instead of the ecosystem that is sequestering the carbon and trapping all the carbon and helping mitigate climate change, you have completely opposite. And it is a carbon source. So in Thailand, it is estimated by 1990s, about 20 to 30 seagrass meadows have been lost. This number is very rough estimate since there are no records of seagrass distribution before that. There are some very rough estimates for some of the sites, but there is no national map. So the biggest, one of the biggest meadows in Thailand is in the national park. And so far it has been stable since 1973. And you can see that in 1995, 2003, and 2009, this is all seagrass bed. So even though this is very harsh environment with all the waves and the storms, the seagrass meadows are still there after 40 years. However, on the other island, we estimated that the loss of seagrass is about 3.2%, especially between 2004 and 2014, where a lot of meadows are lost. So that's the red color. So around here, it's very important. So both of these sites are actually dugong feeding areas. So that is a huge marine mammal that is only eating seagrass meadows, that is only eating seagrass. And the hotspot for dugongs in this region is actually here. So when you lose seagrasses, even a little bit in this region, the animals have to go and find somewhere else to eat. But on the other hand, we have some of the meadows that are actually increasing, where in this site, the local community realized the potential of these ecosystems for their food, and they made like eco tours, and even the monks, the Buddhist monks came and made, I don't know how I would explain that. They made like a fence of their robes, which are very sacred here, to protect, to make like a gate where the seagrasses are protected. And that is still today. So this seagrass in this island is pretty much stable and very well protected from any kind of boat activities, any kind of fishery, any kind of construction, and so on. So there are both positive and negative stories so far. And if we have, and along with the changes of seagrass meadow, the carbon that is stored within the sediment also changes. So for example, so this is the example from here, from Libong Island, where in the first years, the seagrasses were very healthy. This is the urban development area. It wasn't too big. And the carbon was actually coming from seagrass sources and from terrestrial vegetation. So as they cut the forest, there is a runoff into the coastal area, and the seagrass meadows are actually trapping that carbon. And then from 2004 until 2014, when the urban area started to develop more, and there were more cuttings of terrestrial forests, more eutrophication, more nutrient input. So seagrass meadows started dying a little bit, so decreasing. So there is less carbon from seagrasses. There is more carbon from terrestrial forest or mangrove forest that is being cut from the land. And in the last several years, where you have a huge, much larger urban development, the carbon is being very limited in the area. And this is the example that I was saying, that in 2016, it was very healthy seagrass meadow. In 2021, there has been there has been a lot of degradation. And then last year, there was basically nothing. And currently that huge loss of seagrass meadows is just expanding to all the other ecosystems. Yeah, so this is just the example from the drone image where we have in the dugong feeding hotspot where the seagrasses were very dense in 2018 and 2020, that in 2023 you have some very sparse, small size species of seagrass. You have dead seagrasses, or there is no seagrass at all. We still don't know the cause of the seagrass loss. So this was our stable area in the national park for the year. And in last year, when we surveyed, some of the areas were completely devastated. So in 2021, this is the drone picture from a mixed species seagrass. In 2023, it was completely gone. This is also mixed, two different species. Here you have some of the dead rhizomes of seagrass and small size species. Again, just small size species was available. But in the last surveys, my team members went several weeks ago, even the small size species completely gone. So what happens to the blue carbon? It doesn't matter what kind of threats we do to the seagrass meadow. Is it indirect pathways or direct pathways? When there is disturbance of the soil or disturbance of the ecosystem, that carbon just gets lost. There is increased soil erosion and remineralization of that carbon. So in the sediment of seagrass meadows, they are very anoxic, and that carbon is not degraded completely from organic to inorganic form. So that's why you have a lot of organic carbon stored. But then when there are threats or the complete removal of the meadows, that remineralization from organic to inorganic is complete, and that it just gets emitted into the water column. Yeah, so this is what I was saying, that from a sink, actually, it becomes a source. And I roughly calculated that if we continue with the current seagrass decline, that about 2.66 teragrams each year of new CO2 will be being emitted into the atmosphere, which is about extra 500,000 cars on the street, which is a lot every year. And when we lose the ecosystem, so we are not losing only the carbon that is stored, but we are also losing the ability to sequester the carbon to sequester new carbon, and that would be about an additional several hundred gigatons of new carbon every year, which comes to very large numbers of economic losses for countries. But the restorations are one of the pathways that you can actually, that you can restore the carbon storage and as well the ecosystem and other ecosystem services. So these are the current global declarations for restoration for mangrove forests, tropical forests, peatlands, and seagrasses. And these are the examples for the countries in Southeast Asia. And I roughly calculated how much countries can rely on, how much carbon they can rely on from the restoration. So you can see, for example, Philippines can rely on quite a lot of the carbon from seagrass meadows, much less from the mangrove forest, while Thailand can more rely on the carbon from mangrove forest. The green is the carbon from terrestrial forest. Yellow is the peatlands. Yeah, so for example, Indonesia has huge areas of peatlands and mangrove forests, but also seagrass meadows, so they can actually incorporate the combinations of ecosystems. So that there we come why this is all important for climate change. So if we do business as usual, where we have a decline of the ecosystem, and we are losing that carbon that is being stored in the ecosystem. But if we do conservation or restoration actions, we have the new increase of the seagrass area and new carbon that is being stored in the ecosystem. So there are two different pathways that you can actually take. You can do only conservation, or you can do restoration, or combination of both. Where if you are doing business as usual from the baseline, if you're just continuing with the decline of ecosystem, you are losing the carbon that is stored, but also the loss of the ability to trap new carbon. But if you are doing the conservation project, we are actually saving all the carbon that is being stored within the ecosystem and a potentially new one. While when we have the restoration projects, we are starting basically with nothing. So our baseline of the carbon is close to zero. And through the years, if we have increase, we have to wait for about 10 years before the carbon starts accumulating in the ecosystem. So this is the example of the global mitigation potential. This is in terms of conservation actions. So if we start conserving blue carbon ecosystems across the globe, how much of the carbon can potentially each country can rely on. And you can see that Southeast Asia has very high numbers, especially Indonesia, Malaysia, Thailand, and Myanmar. They have very high numbers for teragrams of CO2 per year they can account for. And if we look specifically for each ecosystem, blue color for mangroves, tidal marshes, and seagrass meadows, you can see that conservation actions account for about 1/3 or even less than that of the potential of CO2 per year compared to the restoration. So combination of both conservation and restoration would be ideal. So again, we are coming, why is that important in a global perspective? So we all know of the Paris Climate Change Agreement, and then based on that agreement, all the countries signed the Nationally Determined Contributions, NDCs, where they outlined what they want and they are planning to do in terms of they are reducing their carbon emissions and how much they are planning to do and how they will do it. So these are the examples for Southeast Asia, as I'm very familiar for this region. So, for these countries like Indonesia, Malaysia, Thailand, Vietnam, and Philippines, these are the examples of 2030 business as usual emission scenarios. This paper is a bit outdated, and I think the newer one is coming out soon. So this slide will be updated pretty soon. So from here you can see that their business as usual scenarios are quite different depending on the country and how much emissions they will have by 2030. So this is the dark blue one is the business as usual scenario. And more recently, all the countries in Southeast Asia agreed that by 2050, they will have net zero emissions. And by 2037, majority of these countries will be carbon neutral. So it means that all the carbon that is being emitted and cannot be reduced will be offsetted through the natural carbon sinks. And that's where the blue carbon can actually fit in. So these in pink squares are actually the amount of CO2 that each carbon has to offset by 2030. Now these targets are being moved to 2037. And so for each country, these targets are a little bit different. And depending on the country, the seagrass, the carbon within the seagrass meadows, plus the carbon from mangrove forests can contribute between 6 to 7% from these targets, from NDC targets, which doesn't sound very big. But when there are no options where you can reduce the CO2 emissions, you can start incorporating as many ecosystems as you can, and you have available in your country to try to offset all that to reach the carbon neutrality target. But seagrass meadows are most poorly represented ecosystem as the nature-based solution compared to the other terrestrial forest or mangrove forest, corals, and the other coastal and marine ecosystems. But we shouldn't forget that we also have climate adaptation measures with sea level rise and storm surges. Seagrass meadows reduce waves by about 2 times, and they also increase the sediment height by about 31 millimeters per year, which in most areas is more than enough to compete with the sea level rise. And in many of these countries, they provide food and livelihoods for many coastal communities, and which their lives and lives depend on, as well as their income. And this is kind of putting it all together, that all the benefits that we are receiving from the seagrass ecosystems. So not only climate benefits, but also fishery, global biodiversity, water quality improvement, pollution reduction, and so on. So we cannot only tie it with the Paris Climate Change Agreement, but also with SDG goals, which every country is trying to fulfill. But also the Ramsar Convention on the wetlands, and most recently with the CBD COP from several weeks ago, which changed, which kind of changed the Aichi biodiversity targets. So yeah, so that's from me. I just wanted to announce, if anyone is interested, that my lab is looking for postdoctoral fellow that is going to look into the current project that we are having. So we are looking at the coastal ecosystems as nature climate solution. We are working together with Indonesia and Japan. We extended the deadline to 1st December with the interviews on 9th December. So we are looking in terms of the blue carbon for mangrove, seagrasses, and seaweed beds. So if you have any kind of knowledge with that, you're more than welcome to send me the email if you want to have more information about the postdoctoral position, or you can use this QR to send your information and register for the position. But also we are having a lot of new internship positions, and we are going to look for new students in several months. And you can actually follow my lab and all the work that we do in all of these social media. We are very active because we are trying to push all the information out for all the local communities, policymakers, and so on. So whoever is interested we are more than happy to share any kind of information. And I left my email here if you all have any questions, or if you're interested, you can just send me the email. Anyway, thank you.
Thank you, Milica. We have two questions, and I have to inform you to be very quick with answering them since we are over time. So the first question is, are there any nursery-like organizations for repopulating seagrasses similarly to coral nurseries?
No, not yet. Seagrass restoration is very difficult to do. Globally looking, there is only one huge, large-scale successful restoration in USA. The other restorations are small scale, and it's only like on a single species, but we have 5 species, so it's not really feasible. There are some attempts, but not very successful.
Thank you. And the second question is there a commercial value to that carbon-rich mud?
Yes. So currently, as you have carbon credits and carbon financial mechanism for like REDD+ for terrestrial forest, you also have the same thing for seagrass meadows and mangrove forest. There are several projects that are currently registered for mangrove forest, and I think for seagrass meadows, Japan has already looked into the commercial. For making the carbon credits commercially available from the seagrass meadows.
Thank you. So if you have any other questions, please contact Dr. Milica Stankovic via email she provided. And now I call upon Iva Njunic to present her presentation on active citizenship and citizen science.
Hi, everyone. Just 1 second to... Yeah, can you see my presentation?
Yes.
Yes, okay, that's great. Just so, okay, now it's like a full screen view, right?
Yes.
Okay, great. So hi, everyone. My name is Iva Njunic, and I run an organization based in Leiden in the Netherlands called Taxon Expeditions. I like to say that Taxon Expeditions is actually a scientific travel agency. That maybe sounds a bit weird, I know, but actually what we do is combination of real scientific research and tourism. And we organize scientific expeditions with a mission to discover new species for science. But what is cool and unique about that is that anyone who is interested can join and travel with a group of scientists to some interesting place. What is citizen science? Well, citizen science is scientific research conducted in whole or in part by amateur scientists, or simply public participation in scientific research. What I personally like about citizen science is that it is bridging the gap between so-called, science world and the general public world. And I think it's important. It is making science more understandable to people. How did it all start? Well, it all started in 19th century in the US, where there was this horrible tradition called Christmas bird hunt, during which people were competing who is going to kill more birds around Christmas time. And this even drove some species to extinction. Of course, ornithologists of that time were really bothered by this, and one of them, famous ornithologist Frank Chapman, decided to change this. So he proposed counting birds instead of killing them. And everything started in 1900 with only a few observers, while nowadays this is really a big movement, and there's almost hundreds of thousands of people every year participating in traditional Christmas bird count. And this is a great example how we can change these traditions and do something which is useful for the science and local community as well. Here in the Netherlands, and there are many ongoing citizen science projects, and also all over the world, right? There are many citizen science projects. There are many apps that you can use as a citizen scientist to contribute your observations to the database, which is publicly available. You have probably heard of iNaturalist, right? Opsidentify, BirdNet, etc. And how most of these apps work is that you take a photo of a plant and animal in your surroundings, you upload it to the database, together with your location, and then the app using image recognition can identify this organism for you. So it also helps you learn about plants and animals in your surroundings, but also in some apps, other people and even scientists can help you identify. So this is really a cool way to, be a community scientist. But with taxon expeditions, we go one step further. And because citizen science is so cool, you can even go on a holiday as a citizen scientist. And that is exactly what we are doing. I'm going to play you a short video just to give you an impression of what our expeditions look like. I hope it will work. So often when I talk about citizen science and the work of taxon expeditions, I like to ask the audience, do you know how many animal species live on Earth? And usually they say something like about few tens of thousands or something like that, while the true number is probably around 8 million. And actually why they think that is because when you ask people, about animal species, they usually think of all the birds and mammals, right? And during all these centuries of biology, right, we have discovered and named only 1.5 million of species. So just a small fraction of the true number. And now in 21st century, we can say that 80% of animal species are still unknown to us. So birds and mammals are just a small fraction of biodiversity, and the true biodiversity is in the millions of tiny creatures, such as insects, spiders, snails. What is unfortunately happening nowadays is that human activities are accelerating rates of extinction. So this means that we are basically losing species before we even had the opportunity to discover them and to name them. And of course, job of taxonomists, which are biologists like myself, is to describe and to discover new species for science. I'm actually also evolutionary biologist. I study cave beetles. But one of the things I'm doing is also discovering and describing new species for science. Here in the Netherlands, we have something called the National Research Agenda, which is a public opportunity to send in questions they think scientists should answer from, the tax money. And one of the main things the general public were asking from biologists to do is to discover more species on Earth and to figure out how many species live on Earth. Unfortunately, at the same time, public funding for taxonomy is going down. You probably are aware of that, because taxonomy is more traditional while governments give priority to cutting edge science. So quite likely, if I would now write a grant proposal that I would like to go, for example, somewhere in the jungle, to stay in the rainforest for a while, and just to collect organisms and to discover and describe new species, quite likely my project would not get funding. And this actually made me think of finding some alternative ways to fund my research and research of my other colleagues, taxonomists who are working with me. And this is how we came up with the idea of starting taxon expeditions. At the same time, we also realized that people do think that our job is cool, because whenever I would travel somewhere, whether in the tropics or to a caving expedition, I would post photos on social media. And very often I would get comments like, wow, you have the best job in the world. Can I join you? Like, I would love to go to Borneo and stay in the research center for a week and just do whatever you biologists are doing. So I also realized that people love this kind of work. And yes, this gave us an idea to start taxon expeditions. What do we do on these expeditions? Well, expeditions usually have, I would say, kind of two parts. First part is the field work, where we do this hands-on work, such as placing all kinds of traps to collect insects and other invertebrates, also doing some manual collecting, hiking in the forest. And then the second part of the expedition takes place in the research center, in the field lab. Usually we bring all the equipment ourselves, so we have microscopes and everything that is needed to be brought in the research center to build a lab, which we're using there for a week or so. And in the lab, we teach our participants how to sort and study specimens, also how to mount insects, how to dissect them, right? Because to be able to distinguish species, you have to often look at insect genitalia, how to make an insect collection, etc. And what is also very important to mention is that we publish all our papers with descriptions of new species exclusively in open access journals, because we believe that science should be public knowledge. It should be available for everyone. And we also tend to deposit the specimens that we collect always in the local institution, so that local conservationists and biologists can also study this material. Where do we organize these expeditions? Well, one of the most popular trips that we organize is expedition to Borneo, where we get to work in a small country called Brunei. We get to really work in the heart of the rainforest. It's literally in the middle of nowhere where we are going to. There is this research center that belongs to the local university. There is no road leading to this place, so you have to go by boat on the river, which is usually very nice, also part of the whole adventure. But sometimes the water level is quite low, so then we have to carry boats with all this equipment on it up the stream, and that's always not so fun. Also, there's lots of mosquitoes, etc. Likely there are no crocodiles, but that's also part of the adventure, and also reason why I like doing this job. It's always a bit unpredictable and adventurous. The research center has facilities such as accommodation for a group of people. They have space for the lab. They have someone who cooks for us. So it's very well organized. And of course, in Borneo, you can find the most impressive organisms on Earth, such as this caterpillar, for example, or this plant hopper nymph. And one of the highlights are night jungle walks, because usually most species are active at night, and the whole jungle becomes just incredibly noisy. And then after dinner, we had up the hill through the forest to look for animals, to photograph them, and to collect them. One of my favorites is absolutely violin beetle. This beetle is huge. It's like maybe even 10 centimeters long. And one way to find it is to go out in the jungle on the rainy nights and to look under these big mushrooms. So it's quite rare species, not easy to find, but we kind of got to know their habitat. So it's always special fun, like to go violin beetle hunting during the expedition. Stick insects are also favorites of many people, and some of them can be very, very big in Borneo. Also, we are focused on snails because we have a snail expert in our team. And we also have a team of geneticists from Verona with their mobile DNA lab, with which they can read the DNA of the new species that we are collecting in the field. And they're one of the first groups starting to develop methods for using this sequencing technology entirely in the field. And we are really lucky to have them in the team, because they just help us to to speed up the biodiversity discovery. However, my favorite expedition is absolutely the one in Montenegro. We were working in Durmitor National Park for a few years. I actually was lucky enough to grow up on this mountain, so I know it very well. I know every cave. I climbed every summit of Durmitor. It's really my favorite place, and I love going there and just sharing my love for this mountain with other people. But now we are focused from this year on researching Canyon of Komarnica River, also in Montenegro. This river, I don't know if you even heard about it. It's really not so popular, but this canyon is as beautiful and as valuable as Tara River Canyon in Montenegro, which is, as you may know, under UNESCO protection. On the other hand, Komarnica is at the moment threatened by a hydropower dam construction because the government of Montenegro wants to build a hydropower dam and to turn this beautiful wild mountain river into an artificial lake. As a cave biologist, I'm really very concerned about this project because this would also flood subterranean habitats, and we know that Montenegro is one of the world biodiversity hotspots when it comes to subterranean fauna. So together with my team, with Taxon Expeditions, but also with our nonprofit organization, Taxon Foundation, we are collaborating with local NGOs and trying to do a biodiversity assessment of this canyon so that we have better arguments to protect it. And this year we organized an expedition there, which was a huge success. We had the biggest team ever. There was like 25 people in total. And we also sponsored 6 local biologists to join us in the exploration of this canyon. We discovered many caves, sinkholes. We did rafting on Komarnica. We're placing traps everywhere in the canyon. And now I have a lot of samples to process. But yes, I also have wonderful colleagues who are helping me. We also have an expedition in Italy, in Tuscany, in a dreamy medieval castle called Castello di Potentino. Maybe it does seem a bit like an unusual place for an expedition, but we're really lucky to be able to work here because the lady who owns the castle has a huge private land, and she's managing everything on her land in an organic way. So she has organic vineyards and vegetable gardens. And we're also doing other kind of mini projects on her land, not only these expeditions focused on biodiversity exploration and finding new species. But I have to say, I really enjoy being castle biologist because we get to stay in this beautiful castle, you know, with fantastic Italian food, unlimited wine, and we get to build a lab in the castle, which is also always fun. Of course, the best accommodation is on this expedition. But yeah, so on every expedition, our goal is to discover new species, and here are some of our results. On our first expedition to Borneo, we discovered 3 new species of miniature leaf litter beetles. We discovered them by collecting leaves from the forest floor and placing them in these cloth bags, which are called Winkler bags. And this has been published in Biodiversity Data Journal. And often people ask me, you know, how difficult it is to discover new species. Can anyone do it? And I would say if you are in a biodiversity-rich place like the jungle of Borneo, it is very easy to discover new species. Basically, you go out in the forest, you take a handful of leaves, and it's almost guaranteed that you already found a new species. But the trick is to figure out which ones are new and why. And that is why you have to be an expert on certain group to be able to say if the species is new or not. And on our expeditions, we bring experts on various groups of organisms to discover new species together with us and our participants. We also found a new species of snails and semi-slugs. This, what you see here, is a semi-slug. It's called a semi-slug because it's not a snail. It doesn't have like a complete shell. It has just a tiny bit of shell under its skin. And we found this species just by manually collecting them from the leaves around research center where we were staying. We also found species of new water beetles, collecting them with this very fine mesh from a river in Borneo. And on another expedition, 4 more new species of water beetles. We also discovered new species of a snail, tiny snail, which we named after Greta Thunberg in recognition for her work as a climate change activist. But you don't have to go to the jungles of Borneo or to high mountains of Montenegro to discover new species. New species can be discovered absolutely anywhere, even in your own backyard. And that is exactly what we wanted to achieve when we announced that we're going to organize an expedition in Vondelpark, which is the biggest park located in the center of Amsterdam. And of course, people thought that we're absolutely crazy, right? Because there's no way you can discover new species in the center of Amsterdam. Netherlands is a country which is not so biodiversity rich, and it has a long tradition of taxonomies and just describing new species and just doing biodiversity inventories. So really, nobody almost believed that this is possible. But on this expedition, which we also invited local people to participate, we found not only one, but two new species for science. One was a parasitic wasp, Aferetta vondelparkensis, so we named it after the Vondelpark. And the other species was tiny leorid beetle, which we named Ptomophagus the Beatles, after the music band The Beatles, because we thought it's not fair that there's no species yet named after the Beatles. And this generated a lot of media attention in the Netherlands, but also in Germany, and journalists were coming to visit us during this expedition and to see what we are doing. And yeah, it was very interesting to actually see these, discoveries being shown in the media and getting some kind of public support for the work that we are doing. After that, the municipality of Amsterdam hired us every year to do a biodiversity inventory of various parks in Amsterdam. So they were not so interested in discovering new species, but more like doing biodiversity inventory together with the people from some nature clubs or people from the neighborhood, and just giving them, species list as a report. So since then, every year we have these urban expeditions in Amsterdam, which are also a lot of fun. And for people who are joining, they're, exciting, educational, and we get to learn more about urban biodiversity. Here are some of the papers that we published as a result of our expeditions in Borneo, Montenegro, Italy, etc., and also Amsterdam. So far we have described more than 20 new species for science, and here are some of them. And as you can see, they're mostly really small organisms, small beetles, slugs, snails, and actually the largest species we discovered is this slug that we discovered in Tara River Canyon in Montenegro, and it is about 20 centimeters long. And of course, we are very proud of our discoveries, but often when we tell people about them, they kind of, shrug their shoulders, because they always prefer about some new species of, I don't know, birds or some showy butterflies. That's why we have one trick up our sleeve: celebrities. New species are sometimes named after celebrities and their trademark looks. For example, this orange spider named after David Bowie, or a new species of fly with a big golden behind named after Beyonce. Or one of my favorite examples, new species of moth named after Donald Trump, with this beautiful white hair, but also very small genitals. And on our first expedition to Borneo, we discovered this beetle, which is 2 millimeter long water beetle. Many people would say not so important, ugly little animal, right? But we decided to name this species after famous actor Leonardo DiCaprio, not because they look similar or anything like that, because we wanted this way to acknowledge his work that he does for nature conservation and raising awareness about climate change and plastic pollution of the ocean, etc. So when we named this species after Leonardo DiCaprio, we were expecting it might get some media publicity, right? But we were absolutely not prepared for what happened next. And that is that Leonardo DiCaprio changed his profile photo on Facebook to the photo of our beetle, and he even wrote Photocredit Taxon Expeditions and Henrik Freitag, who was the first author of this paper. But what did we want to achieve? Black and Tiny Leonardo DiCaprio beetle may not win any Oscar for its looks, but in conservation, every species counts. And as you know, insects and other teeny tiny organisms are often neglected when it comes to conservation. Everyone wants to protect, polar bears and pandas. And I get it. I like those animals as well. I think they're cute. But we biologists, we know that insects and other small organisms, invertebrates, are much more important for the functioning of ecosystems, right? Because they play many important roles, such as pollinating the plants, aerating the soil, decomposing organic matter, and literally they are the little things that run the world. So if we would lose insects or their populations would decline significantly, really the life on Earth as we know it now would not exist. So with taxon expeditions, we are trying to discover and describe more species before it's too late and before we lose them forever, but we're also always trying to show people how beautiful and interesting these organisms are and to highlight how important it is to protect them. And that will be all from me. Thanks. And yeah, please take a look at our website and our social media accounts. I'm trying to be very active and it's a big job to do all of this. But yeah, it would be great if you could check out our work on our social media.
Thank you, Eva, for this insightful presentation. We have already 2 questions, so please, if anyone has any other questions, write them. First one is, what do you find hardest about organizing a field expedition?
What I find the hardest? That's an interesting question. And I would say the administration to obtain a research permit is always my worst nightmare, especially to do research in Borneo, even though we collaborate with local university, there's always some very long procedure, lots of paperwork. And also they're changing some things every year. So suddenly one year we have to register every participant as kind of a local student temporarily, things like that. That's for me very hard to deal with, I have to say. But often we do have some logistical difficulties like transporting equipment, also exporting specimens when we have to bring something home to study here in the Netherlands. This can also be all this lengthy paperwork, thanks to Nagoya Protocol.
Next question is, are there any grants available for participating in an expedition?
Well, we often do get some students on our expeditions who manage to find grants for students to do some kind of conservation work or to do like a mini project together with us during the expedition. So yes, there are some grants available for this, but you would have to look into that yourself. In Borneo, we always sponsor three local students who are joining the expedition free of charge. And yes, in Montenegro this year, we also sponsored 6 local biologists, which was very nice, to have them in the team.
Thank you. Next question is, do you think taxon expeditions could bridge off to other fields like microbiology and discovering new microbial species?
Right now, we are not thinking to do any work like regarding microbiology, but I'm thinking to organize expedition focused on fungi, probably in Greece, because I'm personally interested in fungi. And I will be a citizen scientist on this expedition myself. I don't know much about fungi, but it's kind of my job, my hobby now, and I'm reading a lot about it. I also want to do a course of fermentation, so it's a bit of my personal interest. So yeah, maybe fungi expedition in Greece is coming at the end of the next year or something like that.
Of course, here is your Dirk. He congratulates you. He says, it's a wonderful talk, Eva, and wonderful work of taxon expeditions. Cheers.
Thank you, Dirk. Dirk was actually a person who joined our expeditions 2 times, one time in Italy and one time in Montenegro. Yeah. So it's nice that he's here also. Hi, Dirk.
Then next comment / question is great presentation. For the expeditions, are people that come with you as citizen scientists are bringing their own gear? You said you bring equipment, but what about biospeleology gear, for example?
Yeah, so those who are already speleologists can, of course, join the expedition, and they can go in vertical caves together with me and my team. They do have to, ideally, they would have to bring their own equipment, but I can always borrow also equipment for them. But mostly people who are joining our expeditions are just members of the general public. There, I don't know, dentists, doctors, lawyers, like people who are not biologists, who don't have this kind of experience. But of course, people with some extra skills are always welcome to join, and I'm happy to have also speleologists in the team. Yes, we do bring all the equipment regarding the lab, but people do have to bring their own, like backpack and personal gear, I would say.
Next comment is, I'm studying a biotechnology degree and I have loved your lecture. I would like to participate in this project or a similar one in future. And next,
that's great, thank you.
A lovely idea would be looking at different fermentation cultures, plus many opportunities to try different wines.
Yes, yes. And beer.
And beer. Thank you. If the public doesn't have any more questions, I would like to call upon our next lecturer, Irma Popovic-Dujmovic. Stage is yours. Thank you,
Thank you,
Iva.
everyone. Bye.
Hi, hello everybody. It was really nice to listen to both presentations and to make me laugh about the names of these nice animals that you found. I'm a biologist by my vocation and by my heart, so I understand the importance of all these small creatures. Even they are not the most beautiful one, but still they are very, very important, and even I like them more than the big ones because it's a cliche to like pandas or elephants. So I like beetles and insects, definitely. So I'm going to talk from the perspective of, let's call it applied science, because I'm working in the Nature Conservancy, which is the non-governmental organization working on nature conservation around the world. And we rely a lot on science and on science results, but still we have a lot of scientists in our team, but I'm not the scientist, even though I was studying to be a scientist, but I have never worked as a scientist because I already started to work immediately after my studying, started to work in applied nature conservation and started to work for non-governmental organizations to save our rivers and water ecosystems. So I'm based in Zagreb in Croatia. And I'm going to talk about the thing that we are working on. It's a community engagement in the nature conservation because it's something that is definitely often neglected, but very much needed to do because people are the one that we have to work with if we want to save nature. So what is TNC, the Nature Conservancy, doing in the Western Balkans? We are preserving the wild beauty of the Western Balkans. We can say that here in the Western Balkans we have a rich biodiversity and that we have a lot to show to Europe and to world, and that's why actually TNC decided to select the region as one of the iconic region, one of the 5 in the world. So in here we are mainly working on freshwater and also on climate, mainly in terms of promoting solar and wind and doing the wind power plants in terms of sensitivity mapping, and that they are deployed on the places with least impact on the environment. I'm working for water. We have the a campaign or initiative called United for Rivers. We have partners around working on in Croatia, in Bosnia and Herzegovina, in Serbia, in Montenegro, and in North Macedonia. And in each of them, we have at least one partner and at least one river that we are working on. For instance, in North Macedonia, we are working on River Vardar and its tributaries, actually on one part of River Vardar, Demir Kapia.
Sorry to interrupt. Do you have a presentation? Because slides are not showing.
Not showing, really? Yeah, I
No.
have a presentation. I'm sorry. You should warn me before because I am showing it. I'm sorry. I was not sharing it. I'm sorry. Ooh, just a second. So share. Okay, sorry. I will start then. I will just show you because it was yeah. Okay.
It's
Just
okay.
a second. So do you see it now? Do you see it now?
It should. Yes. Yes.
Okay, great. So, yeah, I'm sorry. So just in short, this is the map of the region where we are working on. This is this United for Rivers, the initiative that we are having. And the partners are here listed below with their logos. So, main aim of the initiative and the vision is that we are working in the region where rivers are loved and people thrive. So it's kind of it's a vision. So we are working on that in with two goals actually. And primary goal is to protect, of course, and preserve these the region's most important rivers. And the second goal is to strengthen the awareness of local communities about the importance of river preservation and the role that communities play in that process. So it's not just about river preservation, it's nature, it's about people. It's about people living around the river, people using the river, and people, wanted to get some income out of the river. So, in the region, we don't have that much communities depending directly from the river for like food. Like in many areas or in like Southeast Asia. But we want to raise their awareness about rivers and how important it is to have healthy rivers. So what is community? It's it's not easy process. It's something that when you need to work collaborative with people that have the same interests, there are somehow affiliated, live in the same region, or they have some common interest, or they are in some similar situation, and this is some situation that is affecting their livelihood. And also it's a vehicle. Why it's not a tool, as you can imagine, it's not a tool. It's not that easy as a tool. It's a vehicle. It's complex. And there are many tools that you can use and many parts of it. So that's why it's called a vehicle for bringing people about some kind of environmental and behavioral changes. Mainly for their own benefits. So it's about joining people together to get their for their own good, of course, which is not easy. Sometimes people don't understand that it's for their own good. So you have to work a lot, educate them, and raise their awareness about it. Often it involves partnerships and coalitions because there are some resources that need to be mobilized. We need to influence some systems, and also we need to link and build relationships. It serves as a catalyst for changing policies, programs, and practices. It's all about change. It's all about changing behaviors. It's all about changing different kinds of policies, practices, and behaviors. So it's a long-term process, which is not easy, but still it's a necessity because it's in this world, in the changing world, when people are affecting so many so much the environment, it's more than a necessity. So I'm going to talk more about what we have been doing in River Mrežnica, because the Nature Conservancy is working in the River Mrežnica for several years now. It's one of our priority rivers, Mrežnica and Tončica, its tributary. This is a testing ground, let's call it a testing ground for us and our approaches. And the first time this community engagement was applied here in a case of Mrežnica river protected areas, I would say not just in Croatia, but also in the region. We started in January last year. So it's now already, it will be 2 years soon. In a few months, it will be 2 years that we started this working, and it's ongoing, as I said, as I wrote here, because it's a long journey. So what we want to aim, we want to preserve the River Mrežnica by shared responsibility with all stakeholders. Usually people like to say that someone else is responsible, of course not themselves, and this is something that we want to eradicate here, because it's common and it's joint responsibility, shared responsibility, and all the stakeholders are equally responsible for what is going on in the region, for the quality, for the health of the river. River Mrežnica has developed already tourism. There are some inhabitants around, weekend houses, rafting activities, diving, so different kinds of activities. People like to go there during the summer period. So it's a nice place to go. But if you want to keep it nice, everybody has to be a part of it. So what did we do? First, in January last year, 2023, we did an online survey. So just to repeat, it was the first time that someone was doing it. So we were piloting it and actually pioneering it. So we didn't expect anything out of that. And we asked 27 questions. Some were offered answers and some open-ended. And we got 324 answers. So we were first astonished by the number of answers because it was quite good. The survey was open for 10 days. And one of the main, I would say, results out of it was that almost 65% of subjects think that the protection of Mrežnica would have positive impact on their activities. Also, one thing very important, that we all started to do this before the river was protected officially by the local government. And this is, I think, one of the important parts to mention, because if you want community to be there, you have to involve it before in the decision-making process for them to feel valuable, to feel that they are really contributing to the system. So not that decisions are made somewhere else and they are just players there. Also, after that, so the colleague who was doing that, she analyzed all of this and she gave them an option that they could have a telephone interview, and 19 people participated in that, left their contact telephone number. So each interview was 30 minutes, and there were 7 questions for in-depth analysis of some of the questions from the survey. So she gave them opportunity to talk more about about their opinion about Mrežnica and different kinds of aspects. And these answers from the interviews actually served as a basis for focus groups and workshops that came later. So first focus group started already in March last year. So there were 3 working group meetings with 3 focus groups. One was tourism, another one was fishing and sports, and then another one was general population. And in total, 25 people participated. So it maybe look small, but as I said, this is the first time that we started it. And TNC was not very known by then in the region, so we were happy to get anything out of it. And as you can see, the main conclusion that we got out of that is that what we actually wanted to see, how do they feel about the establishment of a protected area, and they mainly support the establishment, which was, we clapped to that because we were like, okay, great, so we will not have a great opposition. Then we also they expressed that with these protected areas that there is opportunity to solve some of the problems that the river is facing with this tourism over development, for instance. Then it was visible that all these people, they want sustainable development, especially in sector of tourism, and that they also expressed that they need further communication with the management body of protected area, but also between themselves. Like all these stakeholders need to talk more about what is going on in the region. So based on that, we decided, okay, we need to continue the communication with them, and we had these workshops. They were organized from November to July, this November last year to July this year, and intensive workshops with diverse stakeholder groups. As you can see, stakeholder groups were more divided. So it was not just tourism, it was tourism service providers like kayak, canoe, and raft operators. Another one group was service providers, tourism service providers like restaurants, bar, and camp owners. Then also fishermen associations, and then private accommodation owners, and lastly, weekend house owners. There were in every month there were one or two workshops per month with part of them and several of them. So main outputs so far were that active parties that they felt, they are participating in creation of regulatory framework for tourism activities, because in March the Mrežnica was proclaimed as protected from March this year. So things have changed. There will be a management plan. They started this concession grant for the kayak owners for these tourism agencies, and also some things has to change on the ground. So somehow we open this floor to everybody in terms of they could be a part of it, and they could give their inputs to the whole process of regulation of the behaviors around newly protected area. Also, there was an idea to form an NGO with renters and weekend house owners, because they felt they need to be better represented and better heard in this management of protected areas, and to have a greater voice than they have now, because most of them don't even know each other. And first time we had communication between some stakeholder groups, and we are happy for that, because definitely most of the problems that we have on Mrežnica is because of lack of communication, and people are not discussing about things, and many things can be solved just with communication and dialogue. And this is the main value of these workshops that we are having, that we are trying to build a dialogue on local level between different stakeholders and local groups. So the main conclusion out of it is that we need to continue with this activity, with this community engagement, as there are more things to talk and discuss about, of course. So we continue. This year we started again these workshops, and we have the focus now on these private accommodation renters and weekend house owners, because we need them to create an NGO, and through NGO they could be more active participant on the ground. We want them to have a better wastewater treatment, mainly through the nature-based solutions like constructed wetland. Because now they don't have appropriate wastewater management, which is then definitely directly or indirectly affecting the river and the river ecosystem. And we want them to have a better riparian zone in terms of that they are not building any concrete beaches, benches, or something like that. So we want to build their awareness that rivers should be as natural, even though it's their own land. They are the owners of the land, but still that they are not able to do some crazy construction as they are doing now, like building some walls around themselves or building some concrete beaches and cutting trees around. So we want to make it as natural as possible, to look at the river and the riparian zone to look as natural as possible. So that's why we are continuing to work with these stakeholders and continuing the discussions. And this is the end of my presentation. Thank you for your attention. And if you have any comments or questions, I'm here. Thank you.
Thank you for this presentation. One question that we have already in the chat is, do you ever feel like working with local community is unrewarding, in sense it could be easy, but people make it difficult more than it should be?
Yeah, of course. People always make it difficult, more difficult than as it should be. The main reason for that is because everybody is looking only from their own perspective, like they are the only one on the world, and they don't have this feeling that we are a community and that we have to behave like a community, and that the community interest is the one, that we have to obey to. So yeah, it's not rewarding, I agree, but still, I'm working in the nature protection sector for 20 years, so it's not rewarding as well, and I'm not giving up. So I think it's more like some kind of I'm saying, okay, I will not give you I will stay. I will push you till the end. So if you don't like it, I don't care, So because I know why I'm doing it. I'm doing it for the good of the river, of the ecosystem, and then finally for the good of themselves. But maybe they don't understand it from the first side, but eventually they will. And then I will say, I'm glad.
Thank you for this answer. Next question that we have is, how can the success of the River Mrežnica project be applied to other river conservation efforts in similar or different regions?
Yes, thank you for the questions. As I said, we take Mrežnica as a case study, and we want to build experience on that and knowledge and, how to behave on certain, in these cases. And what we want to do, we want to copy this and make it as a blueprint for other rivers and for other rivers, not just where we are working at, but also for, wherever it's needed to be done. And I would say on all rivers, you need to work with not just with locals, but also some other activities, and build their sense of ownership, build their sense of pride about what they have, because I think this is also lacking. Everybody's taking for granted all these natural values that we have. They are taking them for granted, and this is something that should be definitely changed. And what we are doing, we are also developing this toolkit. It's an online database and a platform for where you can get all information that you want about river protection, and all results that will be posted there So this is something that will be for everybody to use. So we are hoping that it will be used as much as possible. It's still under construction, so as a web platform, but this is then where everybody can get useful ideas, useful data sets, and approaches how to work and what to do to help their rivers to thrive.
Thank you. Eva Nunch is also commenting, wonderful work. She's got so many ideas from your talk we could implement in our work to protect commons.
Yeah, that will be great. I liked that you work there because the community is definitely one of the the most valuable, I would say, and the most unknown river ecosystem that we have, and the canyon that is crazy. And definitely, I would not like to see this project being done, but for sure, some approaches could be related and applied also in Komanica that we are working on. So yeah, we can get into touch directly.
Thank you. And last question from me. And if you have any other questions, please comment. How do you address resistance or conflicting priorities from stakeholders such as developers, local businesses, or other groups?
Oh my God, this is one of the there is a lot of resistance in terms of they want to keep business as usual. Usually it's like that, and they are afraid of any kind of changes. I think it's a human nature, but that's why I think you need to give them time. You need to give them time to adapt to change. That the change is not per se bad for them. So they just need to have time to process this in their minds and to feel that, okay, it's not that bad. Okay, yeah, I need to change things, but nothing strange and bad will happen to me. So that's why, this community engagement is very, very long lasting and very, you have to be there. You have to be thorough. That's why we engaged with a local person that is there. So he is like a promoter of what we are doing, and he is like taking care that, the voice of Mrežnica and protection and our work is seen on the local ground. So he's we call him spider in the web. So he's like everywhere around, connecting the dots. Resistance, there will all be resistance, and there will all be someone saying, no, this is bad, la la la. But in the same time, I said to myself, yeah, of course, whatever you do, positive, negative, there will be someone hating that. So this is not something that will discourage us. It's something that will just, I would say, encourage us to keep on and to prove that we can have some positive results of working together. And I often like to say, yeah, I would like to see everybody playing in the sand together and happy, which is unrealistic, but this keeps me active and positive, that things can change, but you just need to give time.
Thank you. If the public doesn't have any more questions, I would like to conclude this block as well. Thank you, Irma, Eva, and Milza, who I think is not here anymore. Thank you for joining us today, and to everyone that stays here to Symposium, we will take a break, 10-minute long. We will be back at 16 hours.
Thank you very much, and bye.
Goodbye.
Bye-bye.
Hello. I apologize. I seem to be having an issue with my camera, but it's not that important right now. So yeah, as we get ready to start with the final block of lectures, I will be moderating, and I would like to invite Christopher to see if his slides are working. Hopefully not like my camera.
Yes. So can you see a black screen at the moment?
Yes, it's a black screen at the moment.
Perfect. So, and now you should see something.
Great, yes, we can see the slides. Can you try to switch to a different slide just to make sure it works?
Yep.
Perfect. Thank you so much. Okay, it is 4 o'clock in 1 minute. Once again, I apologize that you can't see my face. I hope I can fix that very soon because it
No
feels,
worries.
yeah, it just feels a bit awkward for me. I'm going to give a short introduction. So this lecture will be by Christopher Lance,
Yep.
and the title is Integrating Genomics and Single Cell Data Analysis to Understand Inflammatory Bowel Disease in Children. In this lecture, we can learn how data from single-cell RNA sequencing can be utilized to build a reference atlas of cells in the human gut, and what this can tell us about disease mechanisms in children with very early onset IBS, or inflammatory bowel disease. So let's all welcome Christopher Lance. The stage is yours.
Thanks a lot for the very kind introduction. If you paid attention, then I shortened the title a little bit just to make it fit nicer on the slide, but you're perfectly right. So I'm going to talk about single cell data analysis. I'm a bioinformatician, a PhD student in Munich, and I'm shared between the Children's Hospital in Munich and the Helmholtz Institute in Munich, so just an independent research institute. And I'm in my 4th year of my PhD now. And yeah, let me get started then. I would like to give you also a little bit of a kind of disease background before jumping into the single cell data. I'm going to talk about very early onset inflammatory bowel disease, which is a multifactorial disorder of the intestinal tract. Multifactorial is what you can see on the right, that we have usually a genetic predisposition, and then we have environmental factors, the microbiome or immune dysregulatory mechanisms that all contribute to a chronic inflammation of either the small and large intestine, which is Crohn's disease, or if it's only the large intestine, then it's ulcerative colitis. Or there is also this IBD unclassified where clinicians cannot really say whether it's one or the other. I mentioned those here because it's relatively common, and some people of you might actually know someone in the, like a relative or in your the people you know, and that have one of those diseases. Now, it's common in adults, but we are at the Children's Hospital look specifically at very early onset, which means that this intestinal inflammation starts before children get 6 years old. And it's quite interesting because VO-IBD, so this very early onset, is first of all a rare disease. It's familial, so there's quite some inheritance between from parents to children. Then it's often unclassified IBD from the clinical symptoms, and unfortunately also often intractable, which means it's basically hard to treat or does not respond to treatment. Now, what I want to give here on the right, what I want to visualize or kind of make you kind of aware of is that if you look at the x-axis, which is the years in age, like the years of onset, you can see, first of all, for Crohn's disease and ulcerative colitis, we have usually disease onset in the, let's say, around 20 years of age. And then below, you can see how much environmental factors play into the disease onset and how much genetics play into the disease onset. And the group of patients we're looking at is specifically very early onset. And you can imagine, like with a 2-year-old kid, there was not much environmental factors playing into the game. So the genetic component is much, much larger. Talking about the genetic component in the clinic in Munich, we have a large cohort over years and years collected, where collaborators from all over the world in the end sent us samples. We did whole exome sequencing, and then at the clinic and also in other clinics around the world, several monogenic forms were found. So the way it works is that basically BioIBD is a rare disease, but then among those rare diseases, we have many different forms where a mutation in a single gene can actually cause the disease. And a specific diagnosis of a specific causal gene can be done in roughly 20 to 25% of those patients. For 75%, though, we have no clue what's really the mechanism causing the intestinal inflammation. And as I mentioned before, in terms of intractable to standard treatment, or like intractable disease and like without like very good treatment opportunities. On the right, you can see that when you treat patients with biologics, so that's kind of like the more complex, very IBD targeted drugs. So not small molecules, but like antibody based, for example, that can be something very effective in adults, like only 25% of VOIBD patients respond positively. And also at the bottom, you see HSCT, which is hematopoietic stem cell transplantation. Let me quickly explain this. For certain monogenic forms, just like this IL-10 or IL-10 receptor deficient patients, this is an immune cell defect. And especially those IL-10 receptor deficient patients, it's a very severe form. And what you do in the clinics is when you can identify this mutation, you basically swap out the immune system of those children with hematopoietic stem cell transplantation. So a very drastic therapy, but it does work for about 75% of the patients where this is done. But then still for 24, when you decide to do this, it's still not working. So it's sometimes really, really hard. And if you look at disease progression, then you can imagine, as with multiple diseases, the earlier you can detect the disease and also the better you understand what's really going on a molecular level, the better you can treat those patients. And that's in the end, the goal of this project is to identify a subtype of that disease in a patient as early as possible. Now, if we look at the intestinal barrier, it's actually a quite complex system where we have our epithelial layer, and then we have the lamina propria, which is like, and altogether it's a very complex interplay of immune cells, fibroblasts, epithelial cells, all the different cell types that have to, on one hand, defend the microbes that are just in the lumen of the intestine, but then still, you can imagine they have to keep them out, but still they have to let the nutrients through. So they are kind of still the epithelial intestinal barrier needs to be basically has to have a very fine balance between defending microbes, but still allow nutrient uptake. And so it happens very easily that the immune system is, if it's slightly dysregulated, then there's a big inflammation going on, because like this balance of immune activity is kind of, it's a fine window where it works well. Now, what do we do to understand the system in VOIBD? And also other diseases to some extent, is we use single cell RNA sequencing. And if you think of bulk RNA sequencing, that's something that you probably have heard in your studies for sure. You can imagine that you look at a smoothie of cells, while in single cell RNA sequencing, you actually sequence each and every cell individually. And that means that you can also analyze all your expression patterns in a completely different way. So it's really looking at a cellular level when describing cellular activity and disease mechanisms. Now, single cell RNA sequencing by now is very commonly used, but it's actually not that old. So in 2009, there were those very first publications where like people literally by hand pipetted single cells and then like performed RNA sequencing. And then roughly in like so 2015 was the first droplet-based single cell RNA sequencing. And then you can see that in let's say around 2018, 19, those single cell RNA sequencing kits became commercial, with 10x being the largest company. And then it really took off in the last 5 years. Now, the way this droplet-based sequencing looks like is shown down here. So you have, in the end, an oil phase and a water phase, where you have gel beads that come here as those bigger dots, as well as cells, which are kind of a bit hard to see, but there's a cell always coming here from the bottom. And you have gel beads and cells that come together, and then they form a droplet where you have a gel bead and a cell together. And then there's basically a reaction environment where the library preparation happens, and then you can sequence like in a simplified way. Now, when you actually get this data, then here we go. When you get the data, what you get is a gene by cell matrix, a count matrix, and then you need to do several steps for pre-processing. I'm not going to go into everything here, but I just enlarged this quality control step, where you can imagine this is the droplet that we just saw on the previous slide, where we have the cell and this barcoded bead, which is then the reaction environment for actually doing the sequencing. And then we have all kinds of things. We have empty droplets, we have dying cells, we have doublets sometimes, and then there's just RNA floating around, which is called ambient mRNA. And we basically need to take care of all those quality issues to, in the end, get cells that are, like data that is of high quality. Now, once we have done all of this, we can actually do downstream processing. And just one note, what I want to state here is that projects, they are now in a scale where we're really talking about like several hundred thousands to, in some cases, millions of cells, especially if you integrate multiple data sets together. And this is from a blog from the NVIDIA developer blog, where they actually used standard processing tools, and they implemented GPU accelerated versions. And I thought that this I mean, me doing this analysis a lot, it's super exciting, because if you look at the speed up that you get with like modern tools now, it really, really allows you to analyze a whole different scale of data sets. And yeah, it's also kind of interesting how, a company like NVIDIA puts that on their blog. So they're also somehow interested in those research applications, and then probably also in the health sector potentially as well. And yeah, when you then have those data sets analyzed, I just want to show you a little bit of data here. One of the first or early steps is that you basically get cellular profiles that you try to cluster. And then here on the left, you can see it's called Leiden-2, which is just based on the method for the clustering. It's called Leiden clustering with the resolution 2. And not so important, the important point is you get clusters of cells with similar identity, with similar expression profile. And then you can look at marker genes. Here, this is an example from PBMCs, so basically blood cells. And at the bottom, you can see here those marker genes. The rows are clusters, and then you can actually just identify the cell type based on the marker genes. Now, when you have your cells annotated, then you can start with the more interesting analysis, and that's something that we looked at, for example, in inflamed intestinal tissue. We did a so-called neighborhood enrichment analysis. And what you can see here is basically all the cell states that are enriched in inflammation are colored red, and the ones that are rather lower are blue. And I just picked the fibroblasts here, but I can also tell you that, for example, here in the bottom, you have very, very classical like enrichment of CD8 cytotoxic T cells compared to, like, for example, here up here is the regulatory T cells that are like rather lower in the inflammation. And yeah, so you can really, for each lineage, you can go into depth and see what cells are enriched in inflammation. And then here's just an example for the fibroblasts, because in intestinal inflammation, the fibroblasts are like very much looked at the moment. And then you can actually identify kind of the subset of inflammatory fibroblasts and also look at the genes that are associated with them. So that's actually quite exciting and allows you to really go into depth of understanding what's happening on a cellular level. The other analysis that we're doing is for each cell, you can also quantify pathway activities. And here I show you a heat map where we selected just a bunch of common pathways that are used as also as drug targets. And what you can see is that for each patient, you have different pathway activity. And now this is, it's not something that we use in the clinics, but that's where we do a lot of targeted analysis, is that we look for patients that actually have a certain specifically high activity of the pathway that we can potentially also treat. But this will be greatly improved. That's why I put it here as open question as soon as we have longitudinal single cell data and before and after drug treatment. But that's very hard to get, and in children it's even harder to get. But data sets looking at this are coming out now at the moment quite a lot. I'm switching gears a little bit in terms of application. I just wanted to show you one more example of what else I'm working on besides looking at a specific disease. I'm working in a quite large consortium. It's called the Human Cell Atlas Consortium, and I'm in the team where we build the gut cell reference atlas, basically. And so what we do there is we take a whole bunch of data sets, basically all big data sets that we can find out there, and integrate them into one common reference. And it's like the interesting part is, or the tricky part is, that we have batch effects between different data sets. And we have a whole pipeline where we try to integrate those data sets together, that the cells of the same cell type basically come together and the batch effect is corrected. What we can do with this, on one hand, it's quite exciting because it's community, like also the cell type annotation is very community driven. And you can think a little bit of, we're trying to build, let's say, something like a reference genome. We try to build a reference atlas of cells of a healthy human tissue that you can then use to actually compare disease states to. And this is super exciting. And then also because we include so many data sets, we get a better coverage of the population variation, for example, across ages, and then also across all the anatomical locations in the intestine. And then we can do analysis. We can see, for example, how do immune cell activity change over age, for example. And so this is really exciting, and another project to keep an eye on. Now, I just want to mention. I'm not going to go into depth here, but I just want to mention that, of course, cells are not only out of RNA, but we have the whole from the dogma of biology starts from the genome and then epigenome, transcriptome, and so on and so forth. And I was actually also involved in a larger review where you can see the first figure here, where we try to basically find the best practices for processing single cell data that comes from all those different modalities. And yeah, I just want to, as one example, to see where the field is going, I want to just give a quick recap of basically regulation of gene expression, which is a lot connected to the openness of the chromatin. And this we can measure with attack sequencing. And there's also a single cell version of that, where we basically just have that TN5 transposase binding to the DNA, tagging the DNA at those positions, and then we sequence that, and then we actually get peaks of open chromatin regions, which we can then use for multiple things. For example, we can basically just understand how different regions in the DNA are linked in terms of openness. We can, for attack sequencing, it's a lot about trajectory inference, like if you have cell differentiation, which regions get open and closed, and then we can look very much into depth. If we include the motifs of transcription factors, we can really understand basically gene regulatory networks and how they change in different conditions. Yeah, with this, I'd like to wrap up with just a little take-home message that you've learned a bit about VOIBD as a rare, heterogeneous, and often intractable disease. We can use single-cell sequencing to understand, for example, compositional effects or insights on the gene and pathway level. Then, if we combine a lot of single-cell studies, we can actually build a reference atlas of all organs that's being done at the moment. It's an incredibly huge project, Human Cell Atlas, worth to check out. And then also the future will be a lot of multi-omic data. The hot topic at the moment is single-cell proteomics as well, because it was incredibly hard, but there's more and more coming out on that side. And with this, I would like to give a big shout out to the labs that I'm in. It's 2 labs at the Helmholtz Institute and 1 at the clinic. And yeah, since, as I understood, you're here also quite a lot of students. So if you have, if you are looking potentially for a bachelor or master thesis, and you can do it outside, your university, then also, I'm happy to, get in contact. And with this, thank you for your attention.
Thank you so much for your lecture. I feel like this is something that we don't get to hear often about. I got my camera to work. We have like 5 minutes for questions, for a little Q&A, so I'm just gonna dive right in. The first question we have in the chat states, how can insights from genomics and single-cell analysis be translated into personalized therapeutic strategies for children? And are there genomic or single-cell biomarkers that could be used for early detection?
Yes. So genomic diagnosis is, for example, for VOIBD, used on a regular basis when we have a new patient. Then we do whole exome sequencing to see if we have known monogenic forms. So that is actually done in the clinic on a regular basis for a few years now. Because if you have a known form, then you sometimes know the best treatment already. But that's just a small fraction of patients. Now, for single cell data, at the moment, it's all on the research level. There are not yet, let's say, single cell data derived biomarkers where you do that for a diagnostic purpose, but it's a lot about understanding mechanisms and finding, for example, new drug targets. But I know that there is also studies and efforts going on to actually really try to get diagnostic markers basically from single cell data, but usually you would not sequence the entire, like you would not consider all genes, but you identify a gene set that is very distinctive for a certain disease subtype, for example. And then you could think of developing a test for basically measuring only that small subset of genes in a specific cell type that you then probably would filter or first using FACS, for example. Sorry, long answer for question, but it's exciting. It's a very
for sure
active field. Yeah.
Thank you so much for your answer. Let's maybe go over to Mislav. He would like to say that you did a very nice talk, and he also likes your research. Since you and him are both in the single cell field, he wanted to ask you whether you find it overwhelming that every part of analysis, like QC, clustering, cell annotation, can be done in many different ways with different packages, and there's no standard way of doing things, basically. How do you?
Yeah, it's a good point. So I mean, I was also involved in this best practices of single cell data analysis across modalities review, and we tried our best to basically find benchmarks to give kind of as clear statements as possible. Now, there's two things to that. First of all, there's a few methods that kind of in general perform well. So you have the two frameworks. First of all, do you want to do it in Python or do you want to do it in R? Ideally, you kind of switch a bit back and forth because sometimes there's, for a certain step, a better method in R, and sometimes the method is better in Python. So you build pipelines where you just kind of switch. And then what is quite exciting and which is, I think, a wonderful idea is called Open Problems in Single Cell Analysis Platform, which is a living benchmark. So for a lot of tasks, they have, like, it's quite new that it's like that it went live, but they basically have for all those steps a benchmark, and you can add data sets, methods, and metrics. And so things get evaluated live, kind of, live. And then you can basically from that derive pipelines, like versions of pipelines where you always take the best method based on those benchmarks. And so I think the field is going in this direction, but it's just getting there.
It sounds exciting. Maybe one more quick question with the quick answer, let's hope. So, could these diseases be circumvented by selective use of embryos during IVF? Yeah.
So, in theory, the ones where you have a very well-known, let's say, monogenic form, for example, probably. But the issue is that for the majority of patients, you don't find like the causal gene, and for those, then you can't.
Yeah. Then you can't use this method. Okay. Well, thank you so much for all of your answers. It was a great lecture. So next up, I would like to invite Vidak to try on his slides.
Hello.
Hello, Vidak.
And request.
Yeah, I think I need to stop sharing, but my laptop got a little bit stuck. Sorry.
Yeah, that's fine. I also had some issues, obviously.
I cannot see anything anymore. Ah, here we go.
Thank you.
Okay. Is it working?
You've started screen sharing? Okay, I can see the slide, and I think you can just switch a slide just to make sure it works.
Mm-hmm. Does it work?
It definitely works. Thank you so much. I'm just going to give you a little introduction. In this lecture, as we are nearing the end of this online symposium, we will hear from Vidak Lakušić, based on his experiences of studying fauna of arid regions of Saudi Arabia. Vidak is an entomologist. He has experience with working with various insect groups. And in his lecture, he will delve into the various methodologies and also the novel findings of his research in one of the world's most unexplored areas. Without further ado, let's learn about Saudi Arabian fauna.
Thank you, Eddie. Thank you for inviting me. So, as Eddie said, I'm Vidak. I come from Belgrade, from Serbia. I'm a PhD student at University of Belgrade, Faculty of Biology, and I'm on the model of entomology, where I'm studying aquatic beetles. But for this occasion, I will be talking about studying fauna of arid regions of Saudi Arabia. So how all this started? So 2 years ago, I became an external collaborator of CBIO, Institute of CBIO from University of Porto within the group of biodeserts, and with which I've collaborated on 3 projects in Saudi Arabia. The project that I will be talking about today was funded by Royal Commission for Alula. And the general idea of Saudi Arabia is here I put it's a Vision 2030, where they have idea to diverse income in their economy, so they don't want to be completely dependent on oil. They want also to develop tourism. So ecotourism is one of their main goals today. So we can start. We had 4 main objectives in our project. First, and the biggest, let's say, objective was to collect baseline information on the fauna in the Alula region, with focus on the 12 conservation areas, as you can see here on the map. We were working on various group of animals, so we covered scorpions, spiders, butterflies, and dragonflies among insects, and all groups of vertebrates. So we did fish, amphibians, reptiles, birds, small mammals, large mammals, and bats. Here you can see the map. In the middle of all the conservation area, there is a city of Alula, where was our base camp, from which we went to all other regions to explore. Second objective was to develop guidelines for the conservation, rehabilitation, and management of fauna and diversity in the region. This part I will skip due to the short time of the lesson. And just briefly, by the end of the presentation, I will go through the training and outreach that we also did in this project. So here, the study area. So we worked, here we can see the map of Saudi Arabia. It's obvious that it occupies the most part of the Arabian Peninsula. And one interesting fact is that the area of Saudi Arabia is almost as large as Balkan Peninsula. We worked in the northwestern part of the country. And here you can see in the yellow, it's a bounding box of Alula, and it's the area that we conducted our research, actually. We worked in different areas. So we worked in the oasis, in Alula Oasis and Al Khaybar Oasis, which are actually palm plantations with a high anthropogenical effect. We worked in desert regions in eastern part of the area. We worked in the mountainous regions in western part with the Jabal Shaykhub and Jabal Al-Ward, where water was quite abundant in some seasons. And we covered also 3 volcanic areas. Harat, actually in Arabic, means lava field. So yeah, we also covered 3 volcanic areas. That was super interesting as well. We had a large team. We had Jose Carlos Brito as the PI of the group and the boss. We had the administration part. We had 6 mammal experts. We had 2 bird experts. We had plenty of reptiles experts. 3 invertebrate experts and experts on habitats, remote sensing, and spatial analysis that conducted all the things on the computer that needed to be done. So all our missions we had 9 members that were core members of our team, and in some missions we also had other people joining us. For example, here we have my sister. Actually, she was there collecting venom from venomous snakes. Here, this is the invertebrates team, known like that. I was working, as I said, with butterflies and dragonflies. Martina was working with spiders, and Yuri was working with scorpions. So we started our project in at the end of 2022, and it lasted until end of 2023. We had 4 expeditions, totaling 216 days. So each mission was longer than the previous one, with first lasting for 1 month, for 1 month actually, and the last mission lasting just a bit more than 2 months. Initial plan was to have 5 missions of 50 days, but due to some bureaucratic problems, administration, we were not able to do it that way, so we had to adapt. So we had 4 missions with a different number of days. Here on the right side, you can see the routes that we covered in our project. Is almost 11,000 kilometers we driven in those days. Most of the sampling, almost everything we did by car, of course, and generally our field worked like that. So we have 6 days in the field, and then we have 2 days in a Lula house, and then again we go for 6 days to another protected area, and etc. in the expeditions. In those 6 days, of course, we were sleeping in the tents. We had few sampling points that were very isolated, and we were not able to reach them by car, so we had to do a helicopter ride to get there. It was very interesting. So in total, we sampled 121 sites. Of those, 68 we called general sites, which means that we spent 24 hours on that site. We would arrive there around midday. We would have afternoon transect, then we would have dinner, then night transects, then early morning transect, depending on the group that we are working with. And around 11 or the midday, we would leave to go to the next point. Besides that, we also had 42 fast sites. Fast sites are the sites that we visited only once and spent less than up to 6 hours at those points. Usually those sites were at the beginning of 6 days period as well as at the end of that period. At the end, we also have 11 reference sites. Those are the sites that we visited, that we stayed 24 hours on, and we visit them in each mission because we wanted to see the difference in the seasonality of the abundance and species richness in those points throughout the year. And also we left the camera traps there, which means that even when we were not in Saudi Arabia, we were able to collect data throughout the whole year. And we also have 421 ad hoc sites. Ad hoc sites are the points actually where we would usually stop when changing the points, and it was all usually collecting either bird observation or roadkills. Okay, of course, sometimes also the reptiles, but those 2 groups were the main things we were collecting at ad hoc points. We had also different types of sampling. The main methodology in active sampling was visual encounter survey, where each expert for its group would do a transect in the site, recording the track as well as the time, and we will count all the species and all the individuals of the species that we found in that transect, because then we were comparing the abundances and richnesses in different sites. Besides that, we had point survey, which was mainly done for the birds. But for example, here is an example where we found a water body, which is very rare in the region we worked. So I did a point survey for dragonflies, for example. And the last one is the road survey. Although here I put the car during the day, actually the road survey we were doing at night. So we were catching the small mammals, particularly the rodents, with the car, if the conditions on the point allowed us. So if it was not too rocky or it was not too sandy on the other hand. We also had passive sampling, so we used the light trap for the moths and beetles, Sherman trap for small mammals. For large mammals, we were using camera traps and cage traps. And for bats, we used bat detectors, heart trap, and MISNet. MISNet we also used a few times for the birds, but it was not the main thing we did. So eventually, when we collected all the sampling efforts we done in this 1 year project fieldwork, all of our experts, team members, we did in total over 3,000 kilometers on foot, which lasted almost 2,000 hours of sampling just by foot. Here also you can see almost around 40,000 hours of camera trap surveys, then 20,000 hours of small mammals trapping surveys, etc. Here we have one of my favorite illustrations where you can see how actually our day works in a field. On the white point, you can see it's our camp spot, and the green circle is a buffer of 1 kilometer in which most of our sampling was conducted. Of course, not for every group is possible. For example, in blue color, you can see the bat survey where Diogo was walking much more than 1 kilometer, putting audio mods or the bat detectors, which are the blue stars. On the other hand, you can see a red line, which is a mammal transcend, putting the camera traps, and then you have birds, reptiles. There is one problem with this, but it's okay. It's not too bad. That invertebrates are put in one group, which is not really, should not be like that, because of course scorpions, spiders, and butterflies, dragonflies are very different organisms. But never mind. Still, you can see how our work day looked like in the field. So at the end, we did mapping species distribution. So we had points that we observed in the field, which are these black dots that you see on this map. And on the other hand, besides that, we also did the potential species distribution, which you can see as the orange on this map. So this is one example of one lizard. We produced this map. And besides that, we also did the mapping of habitats. So the colleague was collecting control points on the type of habitats and vegetation cover to build the database on the habitats that occur in the Alula region. And then we come to the results. So in total, we gathered almost 38,000 observations, from which 26,000 are from our fieldwork and 11,000 are from bibliography. And here I just summed everything together. There is it's a bit weird that there is much more vertebrate observations than invertebrate, but first of all, most of these bibliography observations are related to vertebrates, so that explains a lot. On the other hand, you can see that there is a lot of bird observation, 20,000. There is, of course, a lot of birds, but our bird experts have a different approach there, where they almost every bird count as observations, not every, but majority of time is like that. While on the other hand, you can see that there's only 700 observations of butterflies, for example. But that doesn't mean I've seen that number of butterflies, but actually I've seen more than 2,000 of butterflies. So if I'm doing a translate and I see 15 butterflies in 20 meters, I'm not going to put 15 observations. I will put one observation of the same species, but then I will write that I've seen 15 individuals. A similar example you can see for the beetles as well. Although for dragonflies, it's very correct, 92 dragonfly observation, that it makes sense working in arid regions without water, it makes sense. So yes, here you can see that the number of observations, it can be doubled if the approach is different. So after our research, we got to conclusion that there is 664 taxa for the group that we worked in Alula region. And this is not only species level, we also include genus level here, because with some taxa we had problems with identifying them, and it's mainly with spiders, but I will come back later to that. Here I separated vertebrates and invertebrates groups, so you can see how many species actually we found of each groups. I put 100 beetles, but there is many more of them. So when it comes to large mammals, we observed 15 species of large mammals in Alula region. There is a wolf, there are 3 species of foxes, there is wildcat. Those things we observed. There is a sandcat, which we were not able to see, as well as hyena, which we know that occurs there. There are also some old records of cheetah and Arabian leopard, but for them is known that they are extinct from this region. And there is one interesting finding also, because the way we worked with mammals, it's not just camera traps. We were also collecting scats and we were doing metabarcoding. And with that, we found one scat of caracal. So we know that caracal also occurs there, but we were not able to see it actually. And in the lower photos, you can see gazelles, capra, and oryx. All these species are cannot be found in the wild, except the capra. They are found in one of the nature reserves where they are doing strong efforts to with reintroduction. So Orycleucoryx is the endemic species for Arabian Peninsula, but in Alula region is extinct from the wild. So that's why they are doing reintroduction of it. When it comes to small mammals, we found 13 species of rodents. Here we also included one species of hedgehog, hare, and rocavi. I'm not sure if they're defined as small mammals, but due to their size, I put them in this group. When it comes to bats, we had 18 species of bats. It is important to say that most of the data regarding the bats was collected with audiomods and actually the bat detectors. Catching the individuals was very rare occasion because of the lack of water and lack of caves. So if we couldn't find caves, if there is a water, colleague was putting mist nets, so he was able to catch. But even in that situation, there was not many individuals that he was able to catch. But all these things you can see, they're all from our projects actually. Super cute. Birds, we in our project we registered 173 species of birds. They were like kind of a main thing for our project because of their importance for the tourism, and people like to see birds and they're easily identifiable. The most common species of bird is this Amamanus deserti. It's like everywhere. And here we can also see Arabian woodpecker, which is endemic for Arabian Peninsula. We had, I don't think, only 2 observations of this species, actually. We had 29 species of raptors and 5 species of owls in the region. Then when it comes to reptiles, we registered 26 species of lizards, including skinks or the sandfishes, some agamids, normal lacertid lizards, 2 species of uromasting, and also the monitor lizard or varanus, which we were not able to catch. We saw it a few times, but the animal is super fast. It's unbelievable. And we also recorded 15 species of geckos. Here you can see one super cute Pristurus, which is the only diurnal actually species. All the other are, as these Tropiocolotes, they're all nocturnal, but super cute. When it comes to snakes, we had 18 species of snakes recorded. From these 18 species, the 4 you can see on the lower part are venomous, highly venomous. The first one, Equus coloratus, I think it was the most common snake we found. It was rare, but still the most common one. Ceraces was the sand viper where we would find it. And here we have also 2 black venomous snakes. And for example, what is also interesting, this Walternesia, we found only one and single individual in this project, but also in a second project we had this year, we haven't found any individual. So super rare, but super cool snake also. Only 4 species of amphibians. It makes sense. We are working in arid region. We found them usually in the oasis where there is either standing or flowing water, but we could also find them in the wild, in the mountainous regions where there is seasonal water. So they were also there because they're able to live through the dry as eggs in eggs. Then, when it comes to spiders, with spiders we have the biggest problems because there is probably the biggest gap in knowledge regarding this group. There is no literature for this area. There is, because also I didn't say, but one way of IDing species we conducted using DNA barcoding, but there is no DNA barcoding library for spiders of this region of the world, so it was problematic, but we managed to ID some to the species level, some to the genus level, but it was okay. Then when it comes to scorpions, it's a similar story. We had 17 species of scorpions, much less, but they're also a bit problematic with there is also a gap in knowledge. So we are developing on that at the moment. Here on the upper part, you can see some highly venomous species, while on the lower side, you can see a mildly venomous and scorpio being completely, well, not venomous. Then when we have butterflies with 37 species recorded in Alula region. People are always, what do you mean you're doing research on butterflies in deserts? They do not expect butterflies to occur there, but there are actually, and quite a lot, 37. It's not a small number compared to 150 in whole Saudi Arabia, which has some very nice southern areas. And the most common butterflies is definitely this Pontia glauconome, or the desert white, is literally everywhere. But on the other hand, there is only 12 species of dragonflies that we found. They are very rare, which is again normal. They need water to complete their life cycle. 3 species we found only in one oasis with the flowing water, so they only occur there. And for what is, for example, interesting, that this Pantala, this species here, it was everywhere, literally everywhere, but except where was water. Where was water, I didn't find it, but where there was not water, it was there. It's a cosmopolitan species also, so it's a very good at flying. And although beetles were not part of the project, because the beetles are my biggest love in the world of animals, I was also working with them. I was collecting data of them. I was collecting them. So this is just a small show to see what beetles occur there. And I had to give one slide for Tenebrionids, the most famous probably insects in, or at least beetles in desert regions, with a huge diversity and fantastic shapes and fantastic animals in any sense. For example, this Adesmia cancellata lives everywhere. Whether is it a desert, is it a rock, is it a lava field, is it oasis, you can find it wherever you step. It's unbelievable. So then we come to maybe the most interesting results of our research. Our colleagues from the herpetology team, right, herpetology experts, they described new species of snake for science. They named it Rhynchocalamus heliasicus, or Hedges black-collared snake. And the name comes from the Hedges mountain range where we actually worked. What is interesting for this species is that it has 2 morphotypes. On the left, you can see the orange one, which occurs in the sandy areas where we found it, and the black one occurs in the harats, or in the lava fields where those black rocks occur. When we found them, we thought at the beginning there were 2 different species, but using barcoding, we got to the conclusion that it's actually a same species and it's a new. And besides that, we are at the moment, there are 4 new species of geckos waiting to be described. 1 species of rodent, probably. 1 species of butterfly, which you can see here. It's a Melitea species. We are doing a morphological study at the moment to try to find some traits that would differ it from the most a close relative, which is Melitea deserticola. We are extracting genitalia at the moment to find something there, because mostly it's in those structures, because the wings patterns are almost the same. So it's super hard to, why we know it's a different, because the DNA barcoding show us high divergence between those 2 species. And then when it comes to scorpions, that is, we are still waiting for some sequences, so who knows what will come out from that. We expect at least 3 or 4 new species to be described, and potentially even 1 more new genus, but we will see in the near future, I hope. And as I said, I will quickly just go through the 2 more objectives. One was training, so we conducted we had a presentation that we gave to the rangers of the conservation areas that we worked in, and besides that, we also did field work with them, so they need to learn how to handle animals, which animals they should, which animals they should not handle, etc. And by the end, the last objective was to produce the field guide of all the groups. Here I showed only reptiles and invertebrates, because those 2 guides are already finished. They are published and they're printed already. And we are still waiting for mammals and birds to be finished as well. They include the information about the area where we worked, about the topography of the groups that we worked, and we have their distribution. So this is a potential distribution with the description of each species and their illustrations. And that's it. Thank you for your attention. I'm very happy to be invited to talk here. If anybody has any questions that we won't be able to answer now, you can ask me directly. You have my Instagram I was tagged on the post on Instagram as well. If you want to maybe collaborate with the Biodeserts or Institute of Sibiu, you can either contact them through the Instagram, they have Instagram page, or you can just go to Google and Google Sibiu or the Biodesert, and you will be able to find it. Thank you.
Thank you so much. That was a lot of fun. I had so much fun as a zoologist. So I'll just there are so many questions we can ask here. I'll try to make this run smoothly. So first of all, let's maybe have two questions in one. So what did you find most challenging about working in a desert environment, and did you maybe have any unwanted encounters with dangerous venomous slash poisonous species?
So there are two biggest problems for me. The one that was the hardest, it was that when the temperature would reach over 50 degrees and we were in the sun in the tents, we had to drink warm water. I think if the water was cool, if it was cold, everything would be much easier. But this way, it was hard. The second problem for me personally, I have to say it, I have a bad knees, so going to the toilet was the biggest problem for me. But besides that, everything it was not hard. You get used to it very quickly, and it's okay. When it comes to dangerous animals, probably. There were some situations potentially dangerous, but nothing ever happened. So sometimes because the way we survey scorpions, for example, we are using UV lights. And when you use UV light, you do not see where you're stepping. So you just see scorpions, but you don't see anything else. So there is a high chance that you might step on the viper in that situations. But luckily, those vipers, they make noise, so you can hear them actually. And it never happened that anybody stepped on that. I was like 1 meter from it, but it made noise, and I was aware that I should stop. So there was not really any dangerous situation.
Nice. We are happy to hear about that. Next up, we have a nice question from Lena. So how diverse is the region when it comes to habitat types, and which would be the most biodiverse or dense with species habitat according to your findings?
So the region is quite diverse. So we have sandy areas, as I said, in the east part, with not many dunes in the area where we worked, but it's sandy with a lot of sand rocks. Here you can see on this photo. Then in one part we have volcanic areas are super interesting because they seem like a rock. It seems like or bare rock, but then you have huge valleys, huge wadis with lot of vegetation, with rich soil, with elements, with huge acacias. And then you have the probably most the places with the least any biodiversity were mountains. Mountains on the west are closer are the lower altitude, and there was not too much diversity. I'm not sure now if I'm correct, but I think that Haratu Airid, which is a volcanic area, has had the highest diversity. At least in butterflies, I'm pretty sure that's the area with highest diversity. It has a high altitude. It has huge wadis, as I said, and it looks ugly when you see it, but then when you enter in the wadis, it's just fantastic what you can find there. It was unbelievable, really.
Awesome. We love to hear it. I'm gonna go with two quick questions regarding methodology. So, did you do some night walks in the desert with the UV light? Maybe. If you did, please tell us more about that experience. And also, what methodology did you use for catching snakes? Was it just like hand searching?
So as I said, I already said that we were using UV lights for catching scorpions. So we had huge pincers, UV light, and you just walk around and collect everything you can find. It was super cool, and super easy. The thing is, UV light is not good for eyes. So we had actually I was using the glasses with the yellow filter, so I don't harm my eyes. When it comes to the snakes, we were doing the transects, so we would have a snake catcher and some super thick gloves, and it was so all the venomous snakes were nocturnal. So at night we were chasing them mostly. You can find them under the rocks, of course, but mainly they're active during the night. So yeah, it was just a night transect with a snake catcher in the morning. So we would, not me, but herpetology experts, they would collect the snakes, pack them in a bag. In the morning, they would extract the venom from them, and then take some nice photos and then release them back into the wild.
Very nice. Okay, let's do the final question right now, real quick. So how challenging was the making of a field guide? I can just add, because I've had experience making a field guide for Coleoptera. It was challenging for sure because it's a lot of info, but I would love to hear your answer.
It was super challenging because of the lack of information. For some groups, it was okay because for I don't know, for birds was quite easy because there are already a lot of books of them for them. Then when you come to the butterflies, also they are known, you know what species they are, but there is no information about their life cycle, about the host plants. So it was quite challenging. It took some time to finish it. Also, IDing some butterflies is challenging, but luckily barcoding helped. So it was challenging, I must say, but at the end, the result is super nice. So I'm very happy with it at the end.
Awesome, awesome. It was completely worth it. Awesome. Thank you so much for your presentation. Yeah, I would like to just a little bit of an apology to Rami for because we're a bit late with the schedule, but I think that we all really appreciated having to hear more about this very unexplored region of the world. So I would like to invite Rami to share his slides. Vidak, can you please stop sharing yours?
Yes, just a second. Thank you very much. Bye bye. I mean, I will stay, but bye bye.
Hi, everyone. Can you hear me, first of all?
Hello, Rami. Yes, we can hear you.
It's clear, or I can, it's better to use headphones?
I think it's super clear.
All right, perfect. Let me share the slides and we start.
We can see the slides. Just try to go full screen.
Oh. Yeah, sorry.
Okay, yes.
Yeah,
Thank you
all
so
right.
much. I'm just going to do a quick introduction for you. So last but certainly not least, we have a lecture from field herpetologist Rami Khashab with the title of Field Herpetology in Lebanon. This lecture will present the importance of Lebanon as a biodiversity hotspot with an overview of Lebanese herpetofauna. We will also uncover the state of reptile and amphibian conservation in the region. Rami, the floor is all yours.
Hello, thank you, Eddie, and thank you, Vic, and thank you, everyone, for this opportunity. Happy to be here, and sorry I couldn't be earlier due to personal issues. And now I'm gonna start talking about a very big subject that usually takes much more time. I'll try to be as brief as possible, try to sum up the main info about Lebanon, about the reptiles and amphibians we have here, the challenges, the threats, and the conservation efforts that are very challenging as well. My name is Rami Khashab. I'm studying biology because herpetology is not an option in Lebanon. We don't have this at any university here. So I had to learn the hard way ever since I was very young. Actually, in my hometown, I just went outside and used to catch snakes and lizards and then try to identify them because as well in Lebanon, we don't have any good references, any good studies done on reptiles and amphibians in many, many years. So there is a lot of outdated data, and even this data is very rare relatively. Lebanon as a country, it's a very small country in the Middle East. It's found on the eastern Mediterranean Sea, in the east of the Mediterranean Sea, near Syria and Palestine. It's just 10,452 square kilometers, which is super small. Yet this country has a lot of special characteristics, such as a very unique location. It's in the center of the world map. It has very unique climate and topography as well. If you see the map of Lebanon, there is two mountain chains, and you go from the coast 0 meters above sea level to 3,088 meters above sea level in like 2 hours maybe. And then you have a huge valley between the two mountain ranges, Bekaa Valley, which also creates a very unique and special kind of habitat. This created a large biodiversity, a large diversity in habitats, microclimates, and everything in Lebanon, which allowed large diversity of species. The species here are a cocktail between species from Europe, from Balkans, from Africa, from Asia, from the Arabian countries nearby, from desert species. So Lebanon, in a nutshell, has more than 3,000 species of plants that we know about. This is, of course, the numbers are not stable because there's always new species being classified and species that gets merged with other species, etc. So around 3,000 species of plants. There are a few hundreds endemic to Lebanon or to a certain mountain range in Lebanon. When it comes to birds, Lebanon hosts like 400 something species of birds. This includes migratory birds, nesting birds, like the birds that stay here all time, summering birds, and etc. etc. When it comes to mammals, we also have more than 60 species. Some of the species like the Syrian bear and Arabian leopards and other species mentioned by Vika actually got extinct here because they are big animals and the habitats are shrinking real fast and the country overall is small, so such big animals find it hard to adapt and survive in such environment. When it comes to reptiles and amphibians, I didn't go through all the animal categories because there is a lot like marine life and vertebrates. We go a few thousand more individuals there. So when it comes to reptiles and amphibians, we have more than 60 species, including some species that were mentioned in literature but were never confirmed here, maybe because of some mistake in museum labels or because of some misidentification or so. Those are very few, but so far we have around 63 species of reptiles and amphibians with expectations of up to 70 when the field work is done better. The herpetofauna in Lebanon, so as I said, we have almost 60 something species. 55 of those species are reptiles and only 8 amphibian species until the day. This includes frogs and toads, salamanders and newts, snakes and lizards, and turtles and tortoises. We don't have any crocodilians here, except an occasional invasive Nile crocodile that gets thrown into a river here, and it survives for a while until some organization picks it up. So all the species here, all the reptiles and amphibians and other species actually are understudied and data deficient here, which makes it very difficult to preserve them or work with them. We have to do most of the field work from zero and visit new areas just to find out more about them. So more than 10 of the species we have here are threatened internationally by IUCN, on IUCN Red List, including sea turtles, like the sea turtles, for example, and few snakes, few lizards as well. Locally, we don't have red listing for the reptiles and amphibians simply because we don't have enough data and we don't even know the exact number of species that exist in Lebanon so far. By assumption, if I want to guess, and from my observations, I think half of our reptiles will be nationally threatened if we did the right red listing. So when it comes to amphibians, there are 8 species of amphibians. Those amphibians are mainly frogs and toads. We have 3 species of toads, 3 species of frogs, including 2 tree frogs and 1 water frog, Pelophylax, very similar to the European water frog. We have 3 toads, including the very common Buphotis and a rare Bufo species, Bufo verrucosissimus, which is very similar to Bufo in Europe, but it's only found in 2 or 3 places in Lebanon, and they are just river valleys where it thrives, and then nowhere else in the country. It's near threatened on IUCN Red List, international IUCN Red List, and their numbers are declining because of the severe water pollution we have here. And we have Pelobates syriacus, or the Syrian spadefoot toad, which you can see here. It's a burrowing species that burrows like underground and spends most of the year there, and then emerges during the rainy season to reproduce. These guys are very unknown in Lebanon, and we actually found a few very interesting new population of the species here in Lebanon, and we are working on publishing them very soon, including some ecological details. When it comes to turtles and tortoises, we have 6 species. 5 of them are threatened internationally because like 3 of them are sea turtles. We have the loggerhead Caretta caretta, the green sea turtle, Calonia medes, which nest on Lebanese shores, and we get hundreds of nests every year here. And the leatherback sea turtle only visits Lebanese waters, but we are still counted as Lebanese herpetofauna, although it doesn't nest on Lebanese sandy beaches. The only tortoise we have here is the Greek tortoise, Testudo greca, Eddie's favorite probably. And this species is vulnerable, and it's doing well in Lebanon, but still many people collect them to put them as garden pets or to sell them because they think they just eat lettuce and they can survive without food for very long, which is one of the threats that our herpetofauna actually facing. When it comes to freshwater turtles, we have one very common Maurimis rivulata, which is the only non-threatened testudine we have here in Lebanon. Maurimis rivulata, or Balkan pond turtle, found in almost every river, every lake, every water body, permanent water body here in Lebanon. They can be seen thriving even in very polluted river, although probably carrying some diseases and parasites that we still cannot investigate properly. And the last species is the one you see here in the photo, the lower photo, is the Trionyx triungus, or the Nile softshell turtle, or the African softshell turtle. It's vulnerable internationally. In Lebanon, it's almost extinct. We have less than 10 records in the past 20 years. This species is big. It grows up to 1 meter. It reproduces in rivers that exit to the sea mostly. And most of our records are made in the sea because this turtle, to survive, it goes to the salt water, to the sea, and hunts there, lives there for a while before returning to the rivers to reproduce. Unfortunately, we don't have any good nesting grounds for this turtle. We don't have any nesting records for this turtle. We just have almost like less than 10 records of it in the past 20 years or so. I've seen it once in my life, and it was accidentally caught in a fisherman net in the sea. I managed to take a few photos, DNA sample, and we let it go in a place we assume will be safe for it. So this is one of the species that is a priority for conservation, and it's probably critically endangered to almost extinct in Lebanon. When it comes to lizards, we have 21 species belonging to 8 different families. This includes true lizards, lacertids, which most of us are familiar with. We have skinks, the second biggest family here. We have legless lizard, including the big guy you see in the photo. This is the Levant Sheltopusico Levant glass lizard, which is Pseudopus apodus levantinus. It's the largest legless lizard species in the world, actually, and they are dark. Apparently, in Europe, they come are very yellow and very brown. Here, they are very dark, which is normal for us, but usually people get surprised by that. This species is feared in Lebanon because people think it's some kind of super snake. They kill it. It has very rough skin, so whenever they try to kill it, it's difficult to do that. So people think it will be some kind of deadly dinosaur that will eliminate the entire village. Other species of lizards include the geckos, agamas, like Lodakia vulgaris you've seen in Vika's PowerPoint. We have worm lizards and chameleon, and a single chameleon species, of course, the Mediterranean chameleon, which is common throughout the region. Species of lizards in Lebanon are mostly, like 4 species are threatened. We used to have 5 threatened species, but the Acanthodactylos species became classified differently, so it's no longer considered internationally threatened. One of these species is the one you can see here in the photo. It's endemic to Lebanon and only to Lebanon, and we know it from only a few mountain ranges of the Mount Lebanon range. It's called the Parvilacerta frazi, or the Fraz lizard. It looks very similar to most lacertids you're familiar with, but it's only found here in highlands of Lebanon. When it comes to snakes, and snakes are my favorite because they need the most work, because they are the most misunderstood and most mistreated animals here in Lebanon. We have, until today, we have 25 recorded species, 26 now because we are working on a publication for the 26th species and species of blind snakes, actually, that was found in Lebanon recently. Like all reptiles, there is always a chance to discover more species here because the border areas are not explored. So we expect species like Natrix natrix and Dolichopsis schmidti to be found in Lebanon. These snakes belong to 7 families. Most common of them is the Colubridae, which includes the genera non-venomous snakes like Dolichophis, Spalirosophis, Hemerois, Platyceps, etc., and some venomous Colubridae like Telescopus phallax and Telescopus nigriceps, both which are present in Lebanon. Lebanon also home to 3 highly venomous viper species. One of them you can see here in the photo. It's the Macrovipera lebetina, or the blunt-nosed viper, which is also present in parts of Europe. We have the Boya palestinae, or the Palestinian viper, which is considered the most dangerous snake in Lebanon. Although snake bites in Lebanon are very rare, and most of the reports of snake bites turn out to be false. We have a third species called Lebanon mountain viper, Montivipera bormulleri, which is a species that lives above 1,700 meters in the mountains. It's endemic to the region, and it is endangered. It is ovoviviparous. Yeah, it gives birth to live young, and it is threatened by global warming because the habitats where it exists are declining from global warming and from shepherds mainly. Two species of these snakes, the Montivipera bormulleri and the Elaphidrusia, which is the colorful snake you can see here and in my profile photo, are endemic to the region. Elaphidrusia was actually described as a separate species last year by Daniel Jablonski, and it is a very enigmatic species. We still know almost nothing about the species other than where it lives, and we're trying to check the northmost distribution of the species where it's found in Lebanon. And this year we actually managed to find the very first juvenile of the species, and we finally know how they look and how they behave. So we know nothing other than that about the species, and it's still under investigation and requires a lot of research and fieldwork. When it comes to threats to herpetofauna, I think it's similar everywhere in the world. So urbanization and habitat loss for urbanization, deforestation, overgrazing is a big problem everywhere. In Lebanon, it's even more problematic because there's no civil planning. People just build houses anywhere. Rules and regulations when it comes to environmental laws are very weak. And species like reptiles and amphibians are almost completely overlooked, even by ministries like Ministry of Environment and Agriculture, who are responsible for this kind of actions or protections. The global warming, we don't have direct studies on the effect of global warming on reptiles and amphibians in Lebanon, but through field observations every year, we can see how the snow in the mountains melts earlier, or there is less snow now and then you can find gravid female mountain vipers, for example, much earlier. For this year specifically, for example, when we always visit the spots for the Lebanon viper in early spring in April. And usually that's when they start breeding, because that's when there is some snow melting in the mountains. That's when they start going out to bask and mate and everything. To our surprise, we found some already gravid females, and you can actually visually see the gravid female, bulkiness of the female. So it was gravid for a while, which is a bit concerning because this is one of the signs that might push them to breed or to have maybe 2 breeding seasons per year or something, which can be very traumatic to their bodies and can lead into affecting the entire population, or it might affect them in other different ways, including affecting their hibernation cycle, for example. Lack of awareness is the root of all the issues here, of all the environmental issues here, not only when it comes to herpetofauna. Lack of environmental awareness, information about these animals here in Lebanon makes it very difficult to convince people not to kill snakes, for example, or to let some green areas be for snakes to reproduce, or let the beaches alone for turtles to reproduce. No need to put all these projectors and lights, avoid the light pollution, and all these things we keep talking about with very little results, unfortunately. So the awareness is not planted in people here from young age. It's not very common in the culture here, which is very sad because that's how animals and reptiles specifically are suffering. Invasive species, luckily, when it comes to reptiles, we don't have any invasive species other than the occasional crocodile I mentioned, or Trachemis red-eared slider that was released in some place, but nothing catastrophic. The main invasive species, or the most concerning invasive species, are the house cats or the domestic cats, and dogs, of course. But cats are the most damaging hunters in this case. Cats are a big problem here and in many places in the world. They hunt reptiles, birds, mammals, and everything, and we try to convince people to keep their cats indoors. But people think that these cats are part of the ecosystem, so it's unethical to keep them at home. And plus, we don't have any proper organizations or shelters that euthanize these cats. if they don't find homes, or catch the stray cats and control them. So in every way, cats are thriving here, and they are affecting all kinds of wildlife, and lizards and smaller snakes usually suffer the most in this case. Illegal pet trade, although reptile keeping is still not very developed in Lebanon. Very few people actually keep reptiles, snakes, and exotics. You can often see species like the Greek tortoise, the Pseudogrecca, or the fire salamander, Salamandra inframaculata, in pet shops in big numbers, because they're easy to find, easy to collect, and they look nice. And people think because they are reptiles, amphibians, they don't need much feeding, much care. So usually these animals get confiscated and released back to the wild, but it's still an ongoing issue. And I hope people don't show interest in keeping reptiles here in Lebanon anytime soon and start catching other species. Pollution, of course, it's an issue, either land pollution, water pollution, air pollution, chemical pollution, use of pesticides. All of these are big issues. They pollute all kinds of habitats here. Our rivers are very polluted with sewage. There is solid waste everywhere. Chemicals and pesticides are being used without control as well. So you can just imagine the damage that's being done. And like sometimes when you think about it, it feels like there is no time to save these animals compared to the damage being done. But we try. So when it comes to conservation of these animals, protection, public awareness is the first thing, because I believe like if people understand why they should protect and save these animals here in Lebanon and why they are important, and that they are not as evil and harmful as they think, and snakes won't chase you and kill you, and all these myths. If you break the ice and you make people understand even a little bit about how these animals are important and why they shouldn't fear them, people might change their mind and at least stop killing snakes, or at least think twice before, I don't know, put their house sewage in the river or throwing something on the street. Reptile rescue, it's only done by me and maybe a few others, which is still very new in Lebanon, because people don't understand why snakes and other reptiles are important. They won't call us for that, but sometimes some people who like animals, who like nature, they do call, and we managed to save some of these animals and release them back to the wild. Treating of these animals also, sadly, we barely have any vets experience with wildlife or exotics at all. So we have to count on ourselves, on contacts abroad, to help us with rescuing these reptiles that actually suffered some major damage, like, I don't know, tortoise that got run over by a car, or a snake that was hit by a shovel, or I had a case where a tortoise was chewed by a dog because someone decided to bring it home for the animals to play together, and ended up killing the tortoise. The tortoise didn't survive the dog bite. So we try to rescue and rehabilitate these animals as much as possible in order to save as many as we can. Fieldwork and data collection is a big part of the work. We encourage citizen science a lot, because most of the country records, because at first it's very difficult for me alone, for example, or for us alone to do the fieldwork by ourselves. The season is very brief and there is many areas to visit. Although Lebanon is very small, there is a lot of small microhabitats that need proper investigation. So citizen science helps a lot. People start reporting these animals on Facebook groups, or they just ask if the snake is venomous or not after killing it most of the time. Then when we tell them the snake is non-venomous, in some cases they stop killing, and next time they send us a picture of a live snake asking about the species. So when it comes to data collection, every year we do tons of field visits, personal field visits to different areas, especially unexplored areas near the borders of Lebanon and Syria, to try and find and collect data, as much data as possible about the reptiles and amphibians we have here. And this led to us finding some interesting discoveries to be published as well. One of the publications we made 2 years ago, where we recorded the diadem snakes, Biologists diadem for the first time in Lebanon. Now we are working on a checklist for Lebanon for all the species we know exist in Lebanon. In this checklist, we try to fix the errors, or like comment on the records that were made before and add all the new taxonomic evolutions or new taxonomic changes that happened, and add the species that were not published in the previous checklist, which was made 22 years ago in 2002. And direct protection, we try to protect the species directly on the field by saving them on the spot, removing obstacles, removing anything that might harm them. And when it comes to sea turtles, there is some actions in Tyre in the south of Lebanon, which is now a war zone, major war zone. The area is famous for its wide sandy beaches where sea turtles lay eggs in large numbers. So my hometown in the south has a beach that received up to 100 nests of sea turtles, both loggerhead and green turtles. So we used to work there and try to protect these nests from predators because we have overpopulation of red foxes there and dogs and all that. So we used to try to protect these the eggs, then make sure the babies make it to the sea, collecting data, etc., etc. This work continued until recent events because the war intensified now there. So the work, it is impossible to do the work. And because of lack of funds, we couldn't evolve that work. We would just make sure these babies made it to the sea and hope they survive and come back to this beach in the future. So my project is called Herping Lebanon, which is mainly shown on Instagram. I try to post everything there to have like a portfolio for everyone to see and to understand what we do, and to make local people curious most of all, and show them what we have in Lebanon, because most of them don't even know we have sea turtles here. Although they can have a walk on the seaside in Beirut and see these sea turtles, but they just don't pay attention. So Herping Lebanon is a personal project dedicated. It's the only project actually focused solely on reptiles and amphibians in Lebanon. Unfortunately, when it comes to this subject, there are no proper museums, no natural history museum in the country, except in a few universities where specimens are not taken care of, and the data there is all chaotic. So there is no way to do good research based on these specimens. And so we have to count on ourselves to collect all the data, to do all the work, and then try to publish it in the most beneficial way possible to everyone. We do a lot of awareness lectures in schools, scouts, and any public circles, actually, even pubs and clubs, to try and raise awareness. And we try to work with kids most of all to make sure the next generation is curious and knows that something called herpetology or reptiles and amphibians actually exists. Because when I was young, I didn't have this chance. No one told me there is this until I started looking for it. So we focus on the kids, on next generations for this. We're hoping there will be more experts that work with reptiles and amphibians and other categories of animals here, wild animals here, because it's much needed in Lebanon, and we're getting very close to a point where it's too late for many species. And we already spoke about the diadem snake, so I will not go through it. Thank you so much. This is all I wanted to say. If you have any questions, I'm happy to answer. And apologies if it was too fast, but I think we have a time limit. So I didn't want to keep you waiting.
Thank you so much, Rami. I think that was a great 25-minute lecture. Thank you for delivering the final lecture of this online symposium. I would also like to quickly thank everybody who is here listening to all of the lectures. Some of you have been here from the very beginning, and we really appreciate you. Some of you joined later, which we are also very happy to have you here till the very end. And now we can move on to the final Q&A session of the symposium. So let's see what we have in the chat. So is there any international cooperation in regards to conservation efforts and between researchers, NGOs, and the government?
The government is always out of the picture. As I said, they care very little about this, unfortunately, here. So, but we do a lot of collaboration, and most of my collaborations actually, when it comes to reptiles and amphibians, are with people abroad. So I collaborate with, for example, with Daniel Jablonski in Slovakia. Czech Republic. His university is in Slovakia. Actually, I collect DNA here actually, and we send them he visits every year, so we send the DNA with him, and we actually work on the DNA of reptiles and amphibians here through that method. And most of the upcoming papers are usually collaborations between here and experts abroad, because there is a lot of missing information here. So I kind of have to count on international people to do that. And another sad thing is that people in Europe, like researchers and herpetologists in Europe, most of the time they know more about the species here than people here, and even ourselves included, for example, because they have access to some data, some old museum specimens that were during the French occupation, for example, or that were collected many decades ago, that we don't have, and we don't have this access here, and our museums are useless, as I mentioned before. So there are collaborations. These collaborations are growing for sure, and I'm positive about it, but it takes time, and I'm hoping there are more interested people to do this work, because it's very difficult to do it alone or within a very small team.
Yes, of course. Yeah, it's a shame about how things are to some extent, but yeah, but we do remain hopeful that things can change for the better. Okay, so next up we have a comment. He says, "Really nice work," and I agree. We have a question from okay, so it says, "I wish you all the best in further research, and I'm really looking forward to that." Okay, so it wasn't a question. I'm sorry. He's looking forward to your publication about Syriacus. What was the species again?
Pilobatis syriacus, the spadefoot toad, Syrian spadefoot.
Yeah. Yeah, we also thought we had that one here in the Balkans, but then the name changed to Balkanicus.
Balkanicus, yeah.
Okay. So is there a good methodology for catching fast lizards? So a colleague of mine is asking this question.
Well, honestly, I enjoy chasing them. I'm always careful not to, like, how I catch them, because they always lose their tail if you grab them the wrong way. But some people use pit traps, or there's a way called noosing, I think, where you use a fishing rod, like a small fishing rod, and you basically use the fishing rod and a small rope to catch them while they're sitting. I don't really like this because it can be a little bit harmful, but this is one of the accepted methodologies in most cases. I often flip them when they're still cold and they're easy to catch, or I try to chase them to catch them. And most of the observation can be done in situ, like while they're sitting, unless you're targeting to collect DNA or morphological data.
Yes. Yeah, I also know some methods we could try, Philip, like someday. I learned something from the Slovenians the last time I was with them, but okay. Yeah, like with sticks and like little nooses, and it's a safe method. Okay. Okay, so
if you do it right, like if you do it right and carefully, it's supposed to be harmless, but like you have to be very careful. I don't recommend it because many people get too excited and end up like breaking the lizard's neck or something.
Yeah, so Agapi says, "Thank you, it was a great presentation." If there are no further questions, then maybe I think it's a good time.
Anyway, for any questions, they can reach me out on Herping Lab on Instagram, or you have my contact information as well. So happy to answer any questions or share any information, of course.
Of course, thank you so much. I think we can move on now to the closing remarks from Davor.
Thank you, Eddie, so much. Thank you to all the coordinators. Thank you, Leila and Eddie. Thank the organizers, obviously, and thank all the participants, especially the ones who stay all the way to the end. And obviously, thank you all the lecturers for your wonderful presentations and lectures today. This is an event that is icebreaker for our network, and hopefully it is a foundation and start of much more events and a bright future for our network. So with this in mind, I'd like to thank all of you once again, and we would close Naturalist Point event at this moment. Thank you so much.